engineered microbiomes. I will not go into the methods, but some of the methods include root exudate collectors, root chips, root arrays, plant chips, K chips, and other standardized fabricated ecosystems, which will help us to evaluate the effect of the engineered microbiomes. The highlights of the today's talk are: in view of the complexities of microbial interactions, micro-friendly plants or genetically engineered edited plant genome will lead to field success by strengthening plant health and preparedness against environmental fluctuations. This microbial fortification will enhance gene expression, enzymatic parameters, nutrient uptake, and biocontrol response. Strategic manipulations, therefore, they help in inoculation of microbial multispecies, will have more impactful response on plant growth performance by minimizing chemical farm inputs. In this scenario, microbiome engineering offers exciting opportunities for understanding and engineering individual organisms to the entire ecosystem. This envisioned technology will reveal a vast diversity and elegance underlying natural microbial ecosystems at a frontier of nutritional and ecological demands. The transcendental role of fungi in the field of basic sciences, human health, and agriculture is worth mentioning at the frontier of synthetic biology strategies. However, many of the associated technologies of bioengineering are still in infancy and require a regulated framework for examining in future the crop improvement programs. This is one of our papers, Engineering Host Microbiome for Crop Improvement and Sustainable Agriculture, which, if somebody is interested, can get into the details of this paper. Giving some, you can say, examples of the work that we are doing, I'll just briefly take a few minutes for this. We worked on the diversity and biological activity of fungal endophytes of Gingiber officinale with emphasis on Aspergillus terreus, which was producing a very important bioactive compound, that is styrene, and that was used as a biocontrol agent for causing the leaf spot of ginger that are caused by Colitotrichum gliosporides. Similarly, in another study, the production of gentisyl alcohol from Phoma herbarum was carried out. This Phoma herbarum was endophytic in Curcuma longa, and its antagonistic activity towards leaf spot pathogen, again Colitotrichum gliosporides, was established, and it could be used as a biocontrol agent for this particular disease. Then we also have worked on saffron. This is a slide which is showing the culturable bacterial diversity of endophytes on saffron, but we have also been working on the fungal component of the endophytes of the saffron in Kashmir, and we have a DBT project now, a network project with 5 network partners, and we are trying to use these fungal endophytes for establishing the saffron in the non-traditional areas of the North Indian region, and we are trying to see that how these fungal components, they can help in establishment of this saffron, which grows only in Kashmir and in few areas of Jammu, how we can establish in them in other areas which are non-traditional in nature for their cultivation. And then we have another study where diversity and bioactive potential of culturable fungal endophytes of Berberis aristata has been carried out. And of course, here the motive was to study the bioactive components of the particular endophyte, but then we are also working on the biocontrol abilities of these fungal components as well. For all this kind of work, I would like to, at the outmost, I would like to pay my gratitude and full thankfulness to my mentor and my guide, Professor Geeta Sumli, who has been instrumental in, as I said, in enthusing in me the spirit of working with fungi, which I'm carrying forward for the last 30 years. And I'm spreading the message and telling my students to have passion for working with fungi. I am thankful to Mycological Society of India for giving me this opportunity to deliver this prestigious lecture, that is Geeta Sumli Award lecture, and also for bestowing in me the bestowing with me the honor of this award. I am thankful to my collaborators, Professor Manoj Dhar, and my chemistry partners, Professor K.K. Kapoor, Professor I.P. Singh, and Dr. Prasoon Gupta from IIIM CSIR, the funding agencies, DBT Government of India, DST, CSIR, UGC, and of course the PERS, FIST, and RUSA grants which have been given to our department, and a lot number of students who have worked hard to what we are today and whatever publications and research we have. Thank you very much for a patient hearing.
Uh, Thank you, madam. Your soft copy of your award is in your Gmail, mail, and the award will be sent you by speed post to you.
Okay, sir. Thank you. Thank you so much, sir.
Okay, madam. Congratulations, madam.
Thank
Congratulations.
you. Thank you. Thank you.
Rupam ma'am, final words to sum up, please.
Professor,
Rupam ma'am, your mic is off.
no, there is no need to sum up in the memorial awards, please.
Okay, okay.
I mean, award lectures, please.
But just congratulations.
Congratulations, you can do
to
that.
you,
You can always do that.
Sanjana. It was a
Yes.
lecture we really thoroughly enjoyed listening to.
Yes.
Thank you.
And
Thank you.
we don't think the second speaker awardee is here, sir, for
He is
this
here.
session.
He is here, madam. He is here. Professor Mulani
Is
is
he?
there, madam. Madam, Rupam Kapoor, madam, he is here online.
Yes. Yes, madam.
Yes, sir. Can we have your
Yes. Good morning, madam. Good morning. Good morning, sir.
Morning, sir.
Professor
Good
Mulani.
morning.
everybody.
Good morning, sir.
Professor Mulani is being awarded with Professor P.C. Jain Memorial Award Lecture. May I request him to please share his slides and
Yes.
limit his presentation to 20 minutes.
Yes, madam. Let me share my slides.
Yeah, you can share, sir.
It is not allowing.
Dr. Professor Madhivanan, ask them to admit the slides, please. Permit the slides.
Can you see the slides?
Not yet. Not yet. Professor Madhivanan.
So you open the presentation and then share. Yeah, now I think it's going to come. Yes, it has come in, sir. Go full screen. Yeah,
Can you see, sir?
Yes, sir. Go
Yes,
ahead.
sir. Yes, sir. Please go ahead.
Yes, sir. Sir, good morning to all. Respected Chairman Professor Yashpal Sharma, sir, co-chairperson Professor Rupam Kapoor, madam, the Professor Atri, Professor T.N. Lakhanpal, then all others, other executive council member, Professor Raman, sir, and all the organizing committee member, executive council member for conferring me this PC Jain Memorial Award lecture. Thank you very much, sir. And at the outset, I am thankful to my mentor, Professor A.R. Kulkarni, then Professor D.J. Bhagyaraj, Professor C. Manoharachari, Professor D.J. Bhatt for regular guidance during my research work, and respected Honorable Vice Chancellor of my university, Swami Ramananda Tirth, Marathwada University, Nanded, for allowing me to participate in the conference. Thank you very much. I can go with the slides. And once again, I'm thankful for PC Jain Memorial Award Lecture. And I can just go ahead with, see, I am proposing a very different type of concept, which already we have worked on it. It is a rocky, grassy crevices on the mountainous plateaus of Western Ghats as AM gold mine, arbuscular mycorrhizal gold mine for diversity of arbuscular mycorrhizal fungi. I am in short telling you, like till date, even after our work, see the scientists, basically Professor Mukherjee and Kapoor, Gupta and Mukherjee, then Rodriguez and Bukhari, Khan from Pakistan, and number of workers like say Professor Rahman, Professor Mohan, then Raghupati, Kanan, Parmeshwaran from Tamil Nadu. They have worked out the mycorrhizal spore diversity and population variation in forest and various types of cultivated crops as well as the other plants as such. And the rhizospheric soil spore number, which people have shown, may vary from say like 10 or more up to 200, 200, 200, 50 in 100 grams of soil. Some have analyzed in 25 grams of soil. But let me tell you, sir, our observation shows that in 1 gram of soil, more than 100 spores we get on a rocky, grassy crevices. So basically, rocky, grassy crevices in which it is a plate-like crevices present on the rocky surfaces on mountainous plateaus of Western Ghats. What we investigated, these we investigated 13 sites for mycorrhizal spore colonization, along with we screened number of hosts from each locality. These sites are distributed in about say 7 different districts along the Western Ghats, and the sites are basically rocky one on which one can see these crevices in which a number of years the soil gets accumulated, and during summer season it immediately dries up. Even when water stops, the rain stops, it immediately dries up, and as the soil remains, plants get immediately the the it is opening for the drastic conditions, and the plant whatever colonization is there in the roots, it is being converted into spore form, and these spores are accumulated there itself, and it is accumulated for number of years also. So as a result, large number of spores have been observed there in the localities of such rocky, grassy crevices. Okay, and these plateaus we have checked, 13 plateaus we have checked for mycorrhizal spore colonization, in which one of the plateau which we want to explain in detail is present in Canary Caves in Western Ghat, Borivali forest, where you can see a plate-like thing in which endemic plant Dipcadis saxorum you can see in a fruiting. So these plant species which are present in as rocky crevices, one can remove with the hand entire plant along with the mass of soil, it comes out. And this soil is about say like 1 kg, 2 kg or 500 grams or sometimes more, little more. So when we analyze the host, we get say like more than 75% colonization, in which Dipcadis saxorum shows 100% root colonization as such in the rocky crevices. Along with that, whatever grasses we have reported from the rocky crevices shows also a good amount of mycorrhizal colonization, and spores remain there, which we have analyzed in the soil as such. This work has been published in Journal of Soil Biology and Ecology in 2000, and it was appreciated by Professor DJ Bhagyaraj. Okay, and in that journal we published this work, and later on we analyze. This is a normal, method which we use for separation of the spore, Jardam and Nicholson method, and it is being identified by Seneca and Peirce. Okay. AM colonization in the roots is what done by whole mount of Phillips and Hyman method. Okay, we did the physicochemical parameter analysis of every site. Okay, micro elements, even the nitrogen, phosphorus, that also we have analyzed in different season. But one thing I want to tell you is these rocky crevices, immediately the water stops, the things get dried up. And the plants you can see over there, some of the, so what host we have screened for AM colonization, I am showing the root whole mount, say like a number of plant species which we have screened from various localities. I am showing some of the plant species over here, mainly the grasses and small plants, they are there. Okay, of Liliaceae, Poaceae, Euphorbiaceae, the small one, no doubt, even Commelinaceae, they are there, but the percentage occurrence is less as far as Commelinaceae and few other families they are concerned. Okay, so these, like say Panhala site, we have screened from Kolhapur, then Amboli site from Sindhudurg, and Bhima Shankar site in district Pune. So, and some of the selected hosts from these mountainous plateaus, the rocky crevices I am showing over here. You see the colonization, the whole mount of root shows presence of various types of vesicles in the whole mount of, this is in Iphigenia indica, then Crinum latifolium of Amarlidaceae. You can see a lobed vesicles, they are there. Then we have, say like Erivochilon, then the other plants, lantana, even lantana, though it is a shrub, bigger one, but some of the in crevices, the seedlings, they are there, and that we have analyzed for. Okay, so the Commelina, even Commelina also we got the colonization. Dipcadis axorum, we got very good colonization as such. See, various types of such, most of the host which we screened, either they show mycelium or say like vesicles within it, and the Dipcadi montanum, Ipsimum pilosum, this is a grass species which we have screened for. Inside the whole root mount, you can see the vesicles, they are there. So such vesicles we have analyzed from different plants. Arbiscles are over here in oxalis. This is cynodon, and oxalis you get the the vesicles. Okay. Then okay, and next is arbuscular mycorrhizal fungal population. We have screened the population in rhizospheric soil collected from rocky grassy crevices. What many types of the various, the mycorrhizal species which we observed. And let me tell you, where it is being checked for, we in general got 6 genera. Out of that, Glomus was dominating at every place. Various species, about 23 species of glomus we reported in different rocky crevices over there. And these are the localities. This is from Kas Pathar, which is very important place where for more than 40% endemic plants have been reported from this locality, Kas Pathar near Satara. Then we analyze from rainy, winter and summer season. But still the Achilospora, Glomus, then Gigaspora are the dominating genera from these sites. Okay. Raigad district, we analyzed the Raigad place. Then Karla from, this is from Khandala site, Bhima Shankar. And what also this is the population from it is from Borivali. We have also analyzed, say, like spore wise number of spores in different season. So we got a more number of spores in the rhizospheric soil of various localities, and it is very high number what we observed from every locality. And in winter, followed by summer season, we analyzed along with the the physicochemical properties of that soil from Kas, from Amboli. It is more than 100 spores has been observed. Even in rainy season, we got good number of spores. This is in 1 gram of soil. I just want to tell you, in rainy season also, if we get the soil, because crevices, entire, as I told you, entire crevices, the plants, they come out, and these plants can be checked. We get the spores, but a high number of spores we observed during the winter season. It is more than 100 Karla site. Then seasonal variation in Borivali. Borivali is one of the very important site, undisturbed. These rocky grassy crevices are undisturbed areas where the even after grazing animals they move around, they cannot remove the entire this thing crevices as such. They cannot remove the plants, so the spore which is there, it can be very easily analyzed. Chlamydospores, what we observed in rhizospheric soil, I am just showing some of the pictures from the chlamydospores. They are Achilospora, Archaeospora, then Enterospora, various species of Achilospora. We get Gigaspora. Then we have various species like Gigaspora, then Glomus. Glomus is dominating 23 species we reported, we observed from various sites of from the rocky crevices, out of which large number of species which we recorded. Glomus fasciculatum, very typical one, which you get always in bunches. This, then we have Glomus other species, then Cutlospora, various species which what we observed. And this my work actually, it is based on one of my student, Professor Dr. Vijay Sathe. He did work in the Western Ghats. Along with that, Rajendra Prabhu has done excellent work in Canary forest. Along with my other students, Ashok Kanade, Mustafa Contractor, Pushpa Andale, Solankar, and Savita Wankhede, they contributed in arbuscular mycorrhizal research. Okay. So again, I thank once again, I thank the Professor Raman sir for giving me an opportunity to present whatever work I have done in this regard of mycorrhizal, arbuscular mycorrhiza. I just want to just tell you one thing is that let the young people, because these mycorrhizal crevices, rocky crevices are there throughout India. Though the climate may be cold, or say like temperate climate, which is a high temperature at distributed in various places, such sites need to be checked for further observations. And I tell you that one will get very good number of spores at even at your locality. So with this, once again, I am thankful to organizers for giving me an opportunity to present my work in front of you. Thank you very much.
Thank you, sir. And your award is soft copy is in your mailbox, and the award will be sent to you by speed post. Congratulations, sir. Thank you very
Thank
much,
you,
sir.
sir. Thank you, sir. Thank you very much.
Over to Professor Yaswal Sharma, please.
So thank you very much. And
Thank you, sir.
we wish to conclude this session by just once again uh, informing that we have had three very good lectures in this particular, award lecture series session, wherein Dr. T.S. Murli yesterday presented his talk, and today in the morning session we had a wonderful lecture from Professor Sanjana Kaul on the plant microbiome and its relevance in the sustainable agriculture. And then it was followed by a lecture on AM fungi by Professor Mulani. So with this, we congratulate all the three speakers, and we wish them a wonderful future ahead. And I'm sure that they would be an inspiration for youngsters to follow up.
Thank you very much, sir.
Yeah, so thank you very much. And at the end, I would like to thank our co-chairperson, Professor Kapoor, for conducting the session very meticulously. And I'm also equally indebted to the organizers, particularly to the secretary and to Dr. Shahi, the organizing secretary, and all the senior mycologists present over here for giving us this opportunity to chair and co-chair this wonderful award lecture series session. So thank you very much and have a wonderful day today. Thank you very much.
Thank you, Professor Rupam Kapoor, as well as Professor Yashpal Sharma. Professor Yashpal Sharma chairing the session and Professor Rupam Kapoor co-chairing the session. Now I over to Professor Saye or the organizers for the conduction of the next session, please.
Good morning, Atri Saab. Atri sir, namaste.
Atri sir, namaste.
Sir, namaskar. Thank you, sir.
Actually, I could not join yesterday's meeting because of the network problem.
Congratulations, sir.
Really.
Congratulations. Congratulations.
Thank you. I missed both the meetings. Earlier, I never knew that I am part of the executive. I read it afterwards, the notice, and subsequently the general body meeting. I kept on trying for 20 minutes, but somehow it didn't materialize.
So, Madhivan? Next
Good morning,
session, you
sir.
can
Good
start.
morning, Dr. Atri.
Good morning, ma'am. Namaskar.
Hello, Dr. Sharma.
Sarma's joined. Okay, good, Sarma. Professor Madhivanan?
And the co-chair will be
Yeah, you can start. You can start.
the junior one, senior level, or senior one, or what percent? And we were that.
Sir, you are not audible, sir.
I think
Dr. Susil?
no other person in our country.
Nothing is audible.
So much work in the area of this area.
Dr. Sushil.
I think Dr. Aneja is. It is Dr. Aneja, I think he is speaking.
Dr. Professor Anja is going there.
ಇದು PDF ಇದೆ PDF
and I don't know, you will see yourself, and what are you
Professor Madhivana, it's not audible.
ये समय में हो जाएगा इसकी
Sir, Lakanpal sir, we are requesting you to restrict the lecture by 20 minutes, sir.
How much time, sir?
20 minutes, sir.
As it took by against 10 times. Let's try. देखो, one करो उसको वहां से। एक मिनट अब चलाओ आप यहाँ से
ये वाला
हाँ हाँ उसको देखो ना मिनिमाइज करो माई कंप्यूटर देखो बेटा माई कंप्यूटर पहले माई कंप्यूटर देखो क्या हो पीपीटी का नाम है इसी माई कंप्यूटर इस पीसी और this is new one चलो it will take time these slides अब खोलो बेटा अब खोलो इस पे हाँ प्रेजेंटेशंस में खोलो खोलो
एक करके देखना
and my friends from CS and Botany. पेन ड्राइव नहीं आ सकती पेन ड्राइव बेटा ये तो गवास this is copied from यहाँ से इसमें से डाल लग जाएगी सीधे नहीं ये लग जाएगा उसमें लग जाएगा इसमें लग जाएगा नहीं लगती
तुम्हारा पेन पे मेल कर मैडम एक इसको मेल करके मैं आपको दे रहा हूँ
Morning, Professor Raman.
Good morning, Professor Roderick. Good morning. Welcome, welcome.
I could not join yesterday for the general body. Something went off again with my.
No problem, no problem. Now it has your internet is restored. Good.
How much is it that we are feeling from most modern
कैमरा
Once again, he is not audible.
I think it is audible, please.
Good morning, Professor Rodriguez.
Yeah, good morning. Good morning.
Your auditorium, there was a lot of visitors in the university is a unique, for us is a university. World is a school.
माइक्रोफोन लगा इंटर कर
Education is the way.
सर
अभी तक and that all of us were engaged with research. Do we have to grasp and try to project the organisms as organisms? Do not compartmentalize. Do not pigeonhole them. Because there is significance. We have great academics. But that doesn't have significance in understanding the underlying technique.
सर कुछ नहीं सुन रहा है यहाँ मैं आप वहाँ बैठे हैं उनको बोलो साउंड सिस्टम ठीक करो बिल्कुल एक वर्ड नहीं सुन रहा है ऑडिबल प्लीज नथिंग इज ऑडिबल
Yeah. Okay. जी सर सर करेंट कनेक्ट करो हेलो प्लीज कनेक्ट हेलो
couldn't disconnect.
हेलो हेलो सर फिर ये वाला यूपीआई हेलो नहीं हेलो
Yeah.
हेलो
Now it's okay.
हेलो हेलो
Hello, sir.
Yeah, this we can see here. This mic testing we can hear.
I think
Hello, sir.
yesterday
I am
Dr.
audible, sir?
Bhuvneshwari presented nicely.
Sir.
If they can resume the same system. Yesterday, Dr. Bhuvneshwari's presentation
Hello,
was
sir.
very highly audible.
Hello, sir.
I think
Hello.
they have some technical problems. Let them resolve it.
Hello.
चार्ज है ना इंटरनेट चल रहा है
ईमेल हो जाएगा
हाँ ईमेल हो जाएगा
ईमेल करो लॉगिन
हेलो सर am I audible
Very clearly.
Yes. Yes, you are audible.
Yes. Yes.
now sir now open
बिना इलस्ट्रेशन के क्या बोलते सीक्वेंस है वो पता नहीं यू टेल मी अब पूछिए अत्री साहब को पूछिए आवाज आ रही है
Yes, sir. Yes, sir.
मुझे देंगे कब कैसे कहाँ पे बताएंगे
हाँ तो ये इसकी गलती है ना तो इसको कई बार कहता रहा है हो जाए कि ये प्रॉब्लम ऐसी आ जाए सारा मोमेंटम खत्म हो जाता है
ये वाला है ये नहीं है क्या चाहिए वाईफाई कनेक्ट किया हुआ वाईफाई कनेक्ट हो गया है ये रहा बच्चे अब सब लोग कनेक्ट ये
मैं ही कह रहा yeah that is what I told start with the next hello sir Raman sir
Professor Madhivanan.
hello sir
Yes.
there is some technical issue please
Okay.
there is some technical issue now
Yeah.
shall we start Professor Bhagyaraj lecture now by the
Yeah.
time you will rectify it and then we'll ask Professor Lakhanpal to speak.
Yes. Yes.
Is
Sir.
it okay?
Professor Bhagiraj, sir, we can request Professor Bhagiraj.
Yes, sir.
Professor Bhagiraj, sir.
भागीरथ I am ready
You can share
ऑनलाइन
your
है
slides, sir.
उनके बारे में नहीं वो तो उनके थोड़े उनके बारे में दो ना
You can share your
I can
slides, sir.
enjoy पर उनके बारे में थोड़ा इंट्रोडक्शन या इंट्रोडक्शन थोड़ा सा इंट्रोडक्शन तो होना चाहिए ना
Bhagaraj,
Sir, you shared your slides, sir.
नमस्कार
Namaskar, sir. Namaskar, sir.
Good morning, sir.
Sir, you shared your slides, sir.
कहाँ है उनका ये
Bhagiraj, sir.
हेलो
Bhagiraj, sir, you share your slides, sir.
ओके श्योर
And I request you to restrict for 20 minutes, sir.
श्योर या
And some interaction About Professor.
There is a technical problem there, and
Another lecture. Another lecture is by another well-known authority in the area of mycorrhiza. That he was Professor Lakhanpal was in mushroom, and here is mycorrhiza. I think again when we talk of the Professor Bhagyarat, he is well known internationally, not nationally, internationally known mycologist, especially in the area of mycorrhiza. And again, when we talk of the mycorrhiza, how he has gone from soil to that, what is from basic training to the applied training, and no other worker, working has done as Professor Bhagya has done in the area of mycorrhiza. And every one of us is known to Professor Bhagyaraj. He is the editorial board member, Kavka, MSI, NASI, senior scientist, chairman, Center for Nature, Biological Resources and Community Development, and residing in what we call as the Karnataka. And I don't want to come in between you and Professor Bhagyaraj, and I invite Professor Bhagyaraj to start his lecture, please.
Yeah,
Yes, sir.
the thing is I cannot share. It says you can't share the content unless the host allows you to present.
yeah, the Professor Madhivanan ask them to permit Professor Bhagira's slide.
We are allowed. Please share your slide, sir.
Sir, allowed, sir.
Start your presentation, please. Start your presentation, please.
Yeah, it's coming now.
Come, sir.
Come. You start.
Yeah, you can see the slide.
Yes, sir.
Yeah, okay. Is it moving also? Is it moving?
Yeah, it is moving. It is moving, sir.
Yes,
Thank
sir.
you. Thank you, the chairman, co-chairman, and very distinguished mycologist, student friends. And so the topic I wanted to share is mycorrhizal technology for enhancing crop productivity and farmers' income. The Government of India, during its annual budget 2016-17, set a policy target of doubling farmers' income by 2022. The same year, the ICR came up with and projected that it is not possible to double farmers' income in 6 years, but it is possible to increase the farmers' income. So that is what. And what is the Committee of Doubling Farmers' Income recognized 6 major agricultural strategies for increasing income. As mycologists, we can follow 2 strategies: improve crop productivity and save the cost of production. Okay. So let us see whether microorganisms, especially the arbuscular mycorrhizal fungi, can help in improving crop productivity and save cost of cultivation. And we know that at the time of agriculture, independence, there was shortage of food. But because of green revolution, high yielding varieties were there. There was no shortage of food. We have plenty of food now. But what went along with the green revolution is the use of heavy doses of fertilizer and pesticides. Now we know, after several decades, we have understood the ill effects of using heavy doses of chemicals. So what is now recommended is use of less and less of chemical inputs and more of biologicals. And people have reported nitrate in the tender coconut water, pesticides in the tender coconut water, nitrate much high beyond the recommended level in the groundwater, and so on. So here the microorganisms, especially fungi, as far as what we are talking today, comes into the picture. And we know that nitrogen, phosphorus, and potassium are the 3 major plant nutrients. So mycorrhizal fungi helps mainly in the phosphorus nutrition. And we know that phosphorus is deficient in tropical soils. And any soil having less than 10 ppm of available P is said to be deficient. And we know that many of the soils in South have only 2.4 or 3.5 available P. That shows our soils are so low in available P. And there are certain, but total phosphorus is available in plenty in the soil, but it is not available for plant growth. It has to be solubilized and made it into available form. There are several bacteria and fungi which can solubilize these unavailable of form of phosphorus to available form. But mycorrhizal fungi help in the uptake of this phosphorus by the plant roots. And we know that these mycorrhizal fungi all belong to Glomera mycota, which has 3 classes, 5 orders, 14 families, 26 genera. I am following the O'Haley et al. 2011, and then 230 described fungal species. Though there are 26 genera, there are 4 of them are more common globally as well as Indian soils. Of this is Glomus, Gygospora, Aculospora, and and Aculospora and Entrophospora. And if you ask the next question, of these 4, which is the most common globally as well as in India, it is the Glomus, which is most commonly present. And here I just given a transection of a root, a non-mycorrhizal root. And here we have the epidermis and cortex and the endodermis. And this is the transection of a mycorrhizal root. And the mycorrhizal fungi lives in soil and it produces very large clematоспores. And it penetrates the epidermis, ramifies in the cortex region, and produces vesicles and orgascles. And so these fungi are all obligate symbionts. So we maintain them in the glass house with a suitable grass as the host. And studies have shown, plenty of studies have been done, and they have shown that inoculation with an efficient mycorrhizal fungus improves plant growth. We can see these various different plants. And what is the mechanism by which they improve plant growth? For example, let us take cotton as an example, uninoculated versus inoculated. You can see the significant increase in dry weight, and you can see significant increase in phosphorus, zinc, manganese, which shows they help in uptake of nutrients. And here again, some nutrients do not move freely in soil solution. Some of them move very freely in soil solution. Now, phosphorus is an immobile nutrient. It doesn't move freely in soil solution. So we have the plant root and root hairs. We know the plants take the nutrients from soil through root hairs. So all the phosphorus that is present in the root hair region, they have taken. But there is no more phosphorus left. But there is phosphorus here, away from the root hair region. So the hyphae travel longer distance and then take the phosphorus and pump it into the host. This is the main mechanism by which they help plant growth. Another mechanism, they have synergistic interaction with beneficial soil organisms like rhizobium, phosphate solubilizers, and so on. And so dual inoculation always helps in better plant growth. Another mechanism by which mycorrhizal fungi help plant growth is suppressing plant pathogens, and mainly soil-borne plant pathogens. You can see the groundnut plant infected with Sclerotium rhodesi, and the same amount of pathogen plus mycorrhizal fungi, which very clearly shows they help in the alleviating the plant biotic stress. And here another experiment which we completed recently, how they help the plant in water uptake. This is under water stressed conditions. You can see the soybean, how it looks. And the same thing, this is uninoculated. This is inoculated. You can see the difference in plant growth. And summing up the whole thing, the mycorrhizal fungi help in the uptake of nutrients. They have synergistic interaction with beneficial soil organisms, protect plants against soil-borne pathogens, protect the plants against drought. They also produce plant growth promoting substances like indolystic acid, cytokinins, etc. And we know that mycorrhizal fungi are all not host specific, but it has been proved that they have host preference. That means for a particular host, there is a best fungal partner. We can screen and select the best fungal partner for a particular host. So for this particular plant, this is the best fungal partner. So we have screened and selected the best fungal partner for various agricultural.
God bless you guys.
Hello? 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 Any problem? Yeah.
Others may mute and listen to the lecture.
Yeah. So we have selected the best mycorrhizal fungus for various crop plants important in agriculture, or forestry, horticulture, and so on. And again, inoculating the mycorrhizal fungus, we can save the 50% of phosphatic fertilizer. This is the recommended level of phosphatic fertilizer for chili, and this is in the uninoculated plant given 75 kgP. You can see this is the yield. With 50% reduction in the phosphatic fertilizer, with the inoculation with this particular mycorrhizal fungi, this is the yield, which very clearly showed 50% of P can be reduced. And in and around Bangalore, the farmers go on to their nursery and then buy the seedlings and plant it in the field. And here it makes it very easy to inoculate the nursery bed or the substrate with the mycorrhizal fungus. And so it gets colonized and the farmer takes it and then plants it in the field. And at this time, the floriculture industry people came to us and they said how mycorrhizal fungi will help us. And this is how it has helped. In increasing the flower number, not only increasing number, it increases the vase life of the flowers kept in the flower vase. And my biotechnologists came and then said how mycorrhizal fungi can help at the tissue culture plants. So at the time of hardening, we have introduced, this is the difference in growth. And now we have a new concept of inoculating a microbial consortium, that is mycorrhiza plus PGPR. You can see acacia inoculated with only mycorrhizal consortium. And so another DST project was given to us, and they said, you have shown all these with your PhD students work with 100 uninoculated, 100 inoculated plants. How will it work when a forest nurseryman takes it with thousands of seedlings? So this project was given to us, and we were asked to take 9,000 seedlings. So anyway, it worked even with the 9,000 seedlings. You can see the difference between uninoculated and inoculated plants. And when these seedling inoculated plants and uninoculated tree seedlings were planted in a degraded forest, and after 52 months, this photograph was taken. Again, this is the forest person standing before the uninoculated plant, and he is standing here between the inoculated plant. He can be, when he is standing before the inoculated plant, he becomes small because the tree is very, very, very big. So this is a very simple technology which the DST wants us to take to all the states in the country. And again, the Coleus forscoli is a medicinal plant which was introduced into the southern states with a buyback arrangement with a pharmaceutical company. And there is a disease problem here. You can see this is the disease index was 85. When a consortium with trichoderma was developed, the disease index came down to 33. And same thing applied to chili also. We have developed a consortium. You can just see with 100% of NPK, the fruit yield is this. With 50% reduction in the NPK with the mycorrhizal consortium, you can see this is the yield, which very clearly shows 50% of N, P, and K fertilizer can be reduced through the developed consortium. And again, somehow the Planning Commission wanted me, and then we explained the economics of the mycorrhizal technology. And I did this taking the example of tomato. You can see the yield of uninoculated tomato given 100% of phosphatic fertilizer, it is 9,000 kg. With 50% reduction with the mycorrhizal inoculation, it was 10,000. So the farmer gets 1,400 kg more. So the cost of tomato varies very much in Bangalore. I have taken 25 rupees. So the farmer gets 35,000 rupees more, and he has reduced 50% of phosphatic fertilizer. So he gets 36,875, which was very much convinced. They were all convinced. And again, I have just included one table on about the cost benefit ratio. And with uninoculated plant in one of the reports, which says the farmer will get 80,000 rupees. With inoculation, he gets 1,00,000 rupees. And this is a technology which we developed with the DBT funded project, the nursery technology. You can see inoculated, uninoculated brinjal, which was a real surprise for us. And this is how the difference is, and which has been given to the farming community. And they say they were selling brinjal seedlings at the rate of 1 rupee earlier. Now they are getting 2 rupees per seedling now. So, and this is again the report of a DBT project. And here In case of tomato, there is a very serious disease, wilt disease complex caused by a fungal pathogen, a bacterial pathogen, and a nematode pathogen. So we have developed a consortium consisting of a mycorrhizal fungus, a nematode trapping fungus, and a bacterium, which can take care of all the three pathogens. Thereby, here you can see the difference in the nursery. It has been planted in the wilt sick soil in the field. Again, the technology worked extremely well. And again, the ICR came to us and said there is a problem in case of cashew when the farmer raises the seedlings and then plants it in the field. There is 40% mortality. So through mycorrhizal inoculation, we saw that there is 100% survival and there is no transplant shock. And this is my lot side. And what I want to tell here is, you know that in case pepper is a very important commercial crop, fetching 226 crores per annum as export earning to our country. And there is a serious disease in case of pepper, which is Phytophthora foot rot. And with the collaboration with the Spice Research Institute in Kozhikode, we have developed a consortium of a mycorrhizal fungus plus trichoderma plus glyogladium. This is applied to the nursery bed and then it protects the plants against the disease. And when it is planted in the field, again, it protects the plant in the field. This is a technology which has been followed now. Technology has been transferred to the pepper growers. They are not only the Indian farmers follow this technology, but the FAO wanted me to take this technology to all the farmers growing throughout the world, the farmers growing pepper throughout the world. So a training program was organized in Indonesia and all the farmers, pepper growers came, and now this technology has gone. So I am sure that definitely by this technology, it is doubling farmers' income. So with this, I would like to conclude saying lamb fungi alone or together with other beneficial organisms play a very important role in increasing crop productivity. They are cost-effective, environment-friendly, improve soil health. They increase farmers' health, and they are a boon to farmers, especially in a developing country like ours. Thank you very much.
First of all, I congratulate Professor Bhagiraj for delivering an excellent talk, and which is especially important for the budding scientists. As you have seen, Professor Bhagiraj has not restricted mycorrhizal fungi for increasing the fertility of the plants. Normally, we know these mycorrhizal fungi are being used as biofertilizers, and if you go to the market, you will find maximum of the biofertilizers are based on the mycorrhizal fungi. In addition to that, what Bhagiraj has suggested here that use one thing not individually, consortia, mycorrhiza in consortia for increasing the fertility. In addition to that, what he has taken into consideration, you can use not mycorrhiza only, you can use the what we call as the biofertilizers, bacteria as biofertilizers which are being used, which are available in the market. You make the consortia of the mycorrhiza plus the bacteria for increasing the fertility of the plant. In addition to that, the third parameter which he has taken into consideration mycorrhiza plus the fungal biocontrol agent. As you have seen here, two important organisms, trichoderma and glycadium, he has suggested and he has used in combination. And what trichoderma and glycadium do, they are the antibiotic producers and they have the ability to control the soil-borne pathogens, and these formulations are already available in the market. So in that, if you use the consortia of the mycorrhizal fungi along with the fungal biocontrol agents, you can do two purposes. You can increase the productivity of the plant. In addition to that, you can control the soil-borne pathogens. So this is very excellent as the budding scientists are concerned, and we should take some of these things for carrying our research in the future. Thank you.
Thank you, sir.
Next, we are having a talk lead lecture by Professor K. Acharya. And Professor Acharya is a sectional editor, Kavka. He is molecular and applied mycology and plant pathology laboratory especially in this area belonging to the Department of Botany, University of Calcutta. And I request Professor K. Acharya to deliver his lecture, please. Online है, अच्छा. So the title of the lecture is Mushrooms from Forestry to Food Factory. Food factory, it will again be a very interesting topic again for the youngest scientists, how we can start our work and then we can go to the industrialization of that very product. And I request Professor Acharya to start his lecture, please.
Acharya is offline or online, sir? He is not in the online.
Online.
He is not.
It's offline, sir.
Professor Acharya?
It is offline. But he is not. Sorry, not yet, sir.
Professor Madhivanan offline or online?
No, he is not in the offline. He is. We are not seeing in the online list also. Probably we can.
Online also. You go move to the next lecture, please.
प्रोफेसर लखनपाल जी का आप पहले करवा दें, उसके बाद आप पहले करवा दें. प्रोफेसर लखनपाल जी का पहले करवा देते, उनका सेट हो गया, रेडी है. उसके अभी नहीं हुआ तो ओके. We go with the another talk in the morning lead lecture by Professor Janardana. Professor Janardana is again known to all of us. He is the member of the editorial board Kavka.
Yes, sir. Good morning.
He is.
Good morning, sir.
Good morning. Good morning. Good morning, Professor. Good morning. Good morning.
Good morning.
He belongs to University of Mysore, Department of Botany, Karnataka. And this is again lecture online mode. And I request Professor Janardana to start his lecture, please.
Okay, sir.
Professor Janardana?
Yes, sir.
Sir, kindly restrict for 20 minutes, sir.
Okay, sir.
उनका हुआ सेट.
Sir, will you be able to see my presentation?
You share the slides, sir.
Starting to share the content.
Share the content, sir.
It's starting.
Hello. Sir, will you be able to see the presentation?
Not yet, sir.
Not yet, sir. You share your slides, sir.
Not yet come up.
It's coming.
नेटवर्क का प्रॉब्लम होगा, कुछ स्लो होगा.
Or any permission problem?
हाइब्रिड के ज्यादा नुकसान होते हैं, फायदे कम होते हैं. लॉन्ग रन में उसके नुकसान ही नुकसान है. हाँ, अभी देखा ना, कोविड में हाइब्रिड आ रहा है. हाइब्रिड दे आर टू डेंजरस. अभी नया स्ट्रेन आ रहा है, एक हाइब्रिड.
Professor Janardana, sir, your slide is not coming. You switch off and you try to share again.
I am trying, sir. It is coming here. My students are.
Okay, maybe in the organizing room, whether permission has been given or not.
Professor Madhivanan, kindly check the permission has been given to Professor Janardana.
Sir, we have already given. That network may be slow. I think ask him to again reshare it.
Yeah, I can see it in my screen.
Your screen. Oh, fortunate, madam.
Yeah, yeah.
But,
It's on my laptop. Yeah.
oh, is this Sasireka?
Yes, sir.
Yeah, you are so fortunate.
Thank
Sir,
you.
we can see. We can see his slide, sir. It is visible.
Oh, it is visible. Okay.
Yeah, sir.
In the auditorium, it is. I mean, conference hall, it is visible.
हाँ, ये तो बात ठीक है।
सोलन में नहीं आ
और
रहा है।
एक्सटेम्पोर ज्यादा ठीक है।
Coming here.
लो सर, इनको को रिक्वेस्ट करो, एक्सटेम्पोर कर दो। लीव फॉरगेट अबाउट द स्लाइड। हेलो प्रोफेसर साहब, जरा प्रोफेसर जलार्जा,
yes,
यू
sir,
स्टार्ट
I'm
योर
here.
लेक्चर एक्सटेम्पोर, फॉरगेट अबाउट द स्लाइड,
Yes, sir.
फॉरगेट अबाउट द गो एक्सटेम्पोर, डिलीवर योर टॉक।
Okay, thank you very much, sir.
Yeah, you are having the slide.
Very
You
good
can
morning.
deliver.
At the outset, I immensely thank the organizer, Professor Sushil Kumar Sahil, and the president of MSI, Mr. Abzar Ahmad, and MSI secretary N. Raman, and all the executive team of MSI for giving this opportunity to share my presentation on this important annual meet of MSI at Bilaspur. At the outset, I thank once again, and the topic that I have chosen for today's presentation is on emerging fungal diseases that are affecting the very important, medicinally important and economically important tree species, Azadirachta and Melia. As you know, the trees are an important ecological components of the nature and very essential for other communities for their sustenance. They
Azadirachta and Melia के fungal control.
providing different food systems and generating economies, and also for the betterment of the healthier planet. The tree lovers have listed more than 22 benefits of trees, of which the most important one, the trees provide the most important oxygen, food, save water, clean air, cool the streets of the cities, prevent water pollution, prevent soil erosion, mark the season, and most important, they do create an economic opportunities, provide canopies and habitat for wildlife, and beautify the space. And the most important in the 21st century is the cost-effective solution for all climate change. Therefore, it is very important that we need to conserve the tree species is one of our important sustainable goals of the present generation. Among the tree species, I have chosen two important tree species today, the neem Azadirachta indica, and neem has been the friend and protector of Indian villages and very long time. For ages, Indians have trusted this particular tree to strengthen their health, and also the neem provides remedy for many diseases. It has been used for protecting food and stored grains, and also as a fertilizer, a natural pesticide for fields. In our Vedas, it has been mentioned that neem is a Sarva Roga Nivarini. So therefore, it is in that context, neem is very, very important. And though it is a native of subcontinent, Indian subcontinent, it is also distributed in other Asian countries. And it is in India, it is widely distributed. Look at this particular table, which speaks about the neem product and the biological activities. The neem products of millions of rupees worth of products are being sold in the field of agriculture and other fields. Every part of the neem plant, whether it is a bark or a twig, leaf, flower, fruit, seed, oil, gum, have medicinal values. So therefore, it is very important that we need to protect this particular tree. Another important close relative of neem is Melia. Both Azadirachta and Melia belongs to the same family, Meliaceae, and Melia is another important relative of Azadirachta, equally important, as good as neem. The genus Melia comprises about more than 40 species. Among them, the two species, Melia azadirachta and Melia dubia, are well known all over the world. Melia azadirachta is a small, medium deciduous plant. It is an ornamental, and also it is being used as a shade tree in most of gardens. However, Melia dubia is now cultivated in commercially for wood purpose and has many other applications also. So this is a Melia dubia plantation in Mysore and other surrounding areas. Just like neem, Azadirachta, Melia is also distributed all over the world, and many species have been introduced and acclimatized. If you look at the medicinal values of Melia Azadirach and Melia dubia, both are have great ecological, economic, and medicinal significance. Many natural health products are derived from Melia species are reported to be useful in treating the human diseases. Many are also used as rodenticidal, insecticidal, antioxidative, anti-diabetic, and many other properties have also been useful as a. That is why both in Melia and also species of Azadirachta are treated as village pharmacy in India. Look at the table, which speaks about the neem products from Azadirachta and Melia Azadirachta and Melia dubia used for treatment of many different types of diseases. Both contain antiviral, antibacterial, antifungal, anti-diabetic, and many other properties. Though both Azadirachta and Melia both are contains anti-diabetic, anti-fungal, antimicrobial agent, these three species are not free from fungal pathogens. Fungal pathogens are always dangerous and cause crop losses, affect food quality in many agricultural products. Sometimes accumulation of secondary metabolites by these fungi in the form of mycotoxin, which render the food unsuitable for consumption. There are many fungal diseases which are being aggressive on many valuable trees all over the world. Similarly, on neem also. Very recently, the leaf web blight of seedlings of Azadirachta indica caused by Rhizoctonia solani, seedling root rot by Phaseria solani, leaf blight caused by Coletoricta dematium and Phaseria solani. All these are nursery diseases. But in 1997, for the first time, the dieback disease caused by Phomopsis Azadirachta has been reported from University of Mysore. The button is students reported the dieback disease of neem.
बिना शुगर का आएगा कि नहीं?
Dieback of neem is a destructive disease found affecting the tree almost everywhere.
कम शुगर का?
Disease symptoms include drying of young inflorescence from top to bottom, resulting complete 100% destruction of a tree. The disease can be seen in all the tree, irrespective of the size, age, with almost 100% disease severity. The pathogen was a seed-borne and seed-transmitted. It was in the year 1998, for the first time, the Phomopsis Azadirachta species nova was described by Satish and et al. And it was an important paper which was published in Mycotoxin that speaks about the deuteromitis of fungus, Phomopsis Azadirachta, causing the dieback disease. The pathogen Phomopsis Azadirachta was known to, it is a pycnidial form, belongs to the class Coelomycetes and produces two types of conidia, both alpha conidia and beta conidia. Studies have been conducted to note that only alpha conidia were infective and able to spread through wind current and able to cause diseases on healthy plants. And the exact role of beta conidia was not known, and even now the people are searching for the clues why the fungus produces beta conidia. The exact role of beta conidia yet to be ascertained. The pathogenicity test conducted on neem, Azadirachta, and also related species revealed that the Phomopsis Azadirachta only infect the taxon Melia, the Azadirachta indica, not its relative Melia Azadirachta. And studies have also been conducted both in vitro and in vivo that Phomopsis Azadirachta could be effectively reduced by 1 to 1.5% systemic fungicide, that is carbendazim. Carbendazim was very effective both in vivo and in vitro, and it was suggested. As the disease is very common in large scale, the large-scale spraying of systemic fungicide on aerial parts of the affected neem trees was opposed by environmentalists. So therefore, eco-friendly approaches to control the disease have been suggested, and especially the eco-friendly approaches were not effective, and hence the disease is spreading as before. Just like Azadirachta indica, Melia species, which are known to be source of bioactive compound, are also affected by many fungal pathogens. And among these, in 2017, we have discovered the presence of a destructive dieback disease in different parts of Karnataka. These are the pictures of Melia azadirach trees suffering from dieback disease, the terminal branch drying, wilting, and complete defoliation, resulting in reduced fruit size. These are the other trees, the same Melia azadirach from different farm showing the same dieback symptoms. We have surveyed in 4 districts of Karnataka. We found that the disease incidence was very severe, and 59.86% of disease incidence was reported. The disease symptoms include gradual wilting, finally drying, young shoots from top to downward. The affected tree branches were visible as dry, defoliated, and remain attached to the main trunk. The onset of disease starts well in December, late December, but complete appearance the disease was visible from March to April. The infected plant parts were affected plant parts were brought to the laboratory, and on PDA medium, it was shown that the fungus able to produce white copious growth of the mycelium and produces black globose pycnidia, and these pycnidia are known to produce conidia, which are hyaline, unicellular, thin-walled, smooth, and aseptate. The conidia are of only one type. We expected the same fungus, Pomopsis azadirachtae, may be responsible for causing the dieback disease in Melia azadirach. But to our surprise, we found that a new fungus, Neophysicoccum parvum, known to cause the dieback disease in Melia azadirach. The systematic studies further, but using molecular methods, also revealed that the fungus was a Neophysicoccum parvum responsible for causing this disease. The Melia dubia plantations are being extensively cultivated in different parts of Mysore nowadays. When we have visited the different farms of Melia dubia, we found that the similar disease was also common. Dieback disease was also common in different parts of Karnataka. And the studies revealed that the disease incidence was about more than 20%. Out of 3,378 trees observed, 501 trees were affected by dieback disease. And disease has been observed in the last 5 to 7 years, and the disease is likely to initiate during winter season from December to March, but in April, during onset of summer, the disease will be very severe. The affected branches were brought to the laboratory, and the causative agent of the fungus was isolated. The fungus was known to produce Conidiomatal pycnidia, which are solitary and aggregate and raised black in color. And the fungus was known to produce 2 types of conidia, both alpha conidia and beta conidia. Studies conducted on these conidia revealed that only alpha conidia were able to germinate and produce the germ tube, and the exact nature of the beta conidia is still known. The pathogen, by using cultural, microscopic, and molecular methods, we could able to identify the fungus as Diaporthe fasciolarum, a perfect stage of Pomopsis azadirachtae. And these are the pictures of Conidiomata, pycnidia, the isolated fungus, and the ooze that is coming out from pycnidia. Both alpha conidia and beta conidia are produced. Alpha conidia are produced well in advance, but in the later stages of the culture, the beta conidia are also produced. The molecular studies revealed that the fungus belongs to Diaporthe fasciolarum. The systematic studies were also conducted on whether the Phomopsis adarete or Diaporthe fasciolorum one and the same. The studies revealed that culturally, morphologically, and genetically both were different. So therefore, further studies are going on about their anamorphic and teleomorphic stages, whether they are the same or different from each other. Further pathogenicity tests were also conducted on Melia azadirect, Melia dubia plant by using both Neophysicoccum and Diaporthe fasciolorum alpha conidia. The studies revealed that the both Neophysicoccum parvum able to cause the disease within 15 days on the detached leaf facet. Similarly, on Melia dubia, Diaporthe fasciolorum also able to cause a similar disease. The systematic studies were also conducted in the screenhouse condition, revealed that both pathogen able to cause a disease within 30 days of post-inoculation. 6 systemic fungicides have been selected for screening their fungicide activity against Neophysicoccum parvum and Diaporthe fasciolorum in vitro. Among these 6 fungicides by food poisoning technique, the carbendazim and also cymoxanil at 0.5% were effective. I'm not sharing the details of the studies, but carbendazim was very effective at 0.5%, and also cymoxanil was also effective in reducing the fungal growth of Diaporthe fasciolorum in the laboratory. Many ascomycetes fungi causing diseases of economically important tree species around the world has been reported, and we have powdery mildew of grapes caused by Anxinilla nicator, Dutch worm disease caused by Ophistoma ulmi, chestnut blight, Cryptonectaria parasitica, apple scab, Venturi inequalis is very common and well documented. However, a Deuteromycetes fungi belongs to the Coelomycete, both Azadirachta indica or Diaporthe fasciolorum, Neophysicoccum parvum, causing diseases in tree species is something very interesting. Diaporthe fasciolorum is the perfect state of the Phomopsis, is now causing dieback disease in Melia dubia, and hitherto the fungus Neophysicoccum parvum is also causing dieback in Azadirach. So therefore, it is interesting to study. It is important to note that medicinally and commercially important species such as Azadirachta and Melia affected by fungal disease, it was not very common 10 years before. In the present study, Neophysicoccum parvum associated with Melia and Diaporthe fasciolorum associated with Melia dubia have been characterized. Systemic fungicide carbendazim and cymoxanil at 0.5% concentration were effective in vitro. Field trials are underway to manage the disease. The dieback disease on these 2 species is affecting the aesthetic beauty of the urban environment wherever these tree species are grown. Neem trees are at the verge of losing their life in around South India, and this condition is not far for the species of Melia also. Hence, management of disease is very important, as dieback disease is likely to affect the village pharmacy and may have huge impact on the lifestyles of rural India. So in this context, it is important that we need to control the dieback disease caused by these emerging diseases. Thank you very much. I finally thank the organizer for giving this opportunity to speak on this MSI annual meet.
Thank you very much, Professor Janardana, for
Thank you,
an
sir.
interesting talk.
Thank you very much, sir.
What the lecture says of Professor Janardana,
Yes, sir.
he has emphasized on 3 things in his lecture. First, what is the importance of Melia species? We are all familiar with the 2 species of Melia, Azadirachta indica, that is neem, and the Melia azadirach, that is the darek, which is very common in the plains of our country. Now he has gone for the diseases, and especially what he is emphasizing one important fungus, that is the Phomopsis, and the species is Azadirachtus. What is the importance about that fungus? All of you are familiar with the Phoma fungus, Phoma, which normally comes on the leaves of the plants called the leaf spot diseases. And we can identify this Phoma by the formation of the Pecnidia, which produces only one type of unicellular spores, which are produced on the conidiophores in the fruiting bodies. And what he is emphasizing, the another fungus, that is the Phomopsis, which is responsible for causing the dieback disease. And what the dieback disease is? Well, the symptoms start from the tip and goes back on the leaf. That is the dieback disease. Leaves are affected, then it will go to the branch of that, and the infection starts. Now, what is interesting about this fungus, Phomopsis, is instead of one type of conidia, it produces two types of conidia. One alpha conidia, another is the beta conidia. And what is the interesting about thing, what he is emphasizing, that only alpha conidia has the ability to germinate and infect the plants. And it is still not known the beta conidia which are produced in this fungus, what is the function of those beta conidia which are produced in this fungus. Naturally, they will be having some fungus, otherwise, why the fungus is going to produce two types of conidia? He is very intelligent than us. He is having something which we are not able to understand, We have not developed technology so that we are able to germinate the beta conidia and find what role, what function they are playing when they are causing the dieback disease. This is important. Then he has gone called a fact that how we can control these fungal diseases. But it is interesting that we know, as the melia species are concerned, they are having insecticidal properties. We are using for controlling the insect. Yeah, it is well known fact. Even we are using your neem datun use karte hain. It has got those antimicrobial properties. That's why we are using that neem datun. But what is interesting about this, in spite of having all those antimicrobial, as he's saying, he's having antimicrobial properties, these plants are, why the fungus are able to attack? Means fungus are more super powerful than the properties, antimicrobial properties which are produced by the trees, these milia. So that is the interesting to note, what is that mechanism which the God has given to them, that they have the ability to attack these plants which have got the antimicrobial properties. This is for you to learn, for you to find out the answers, what we are not able to find out till today. Thank you. That's now we request Professor Lakhanpal. Thank you very much, sir.
Thank you very much,
for
sir.
the
Thank you,
excellent
sir.
talk.
Thank you very much.
हार कर रहा है ये हार है इसमें दी हार very sorry, nothing to be sorry. Yes. There are sometimes some tense moments and let them be go to the past tense. और ये लेक्चर जो है वो उस तरह से हो रहा है, एक एनेक्डोट है, एक आदमी यूएस जा रहे थे और वो ब्रेकफास्ट सर्व करने जब एयर होस्टेस आए तो उन्होंने कहा कि नहीं मेरा अपना है खाना। लंच में भी ऐसे ही हुआ तो कट इट शॉर्ट, और डिनर में भी तो उन्हें हुआ कि शायद इस आदमी को पता नहीं है कि ये सारा कुछ उनके फेयर में इंक्लूडेड है। तो उन्होंने कहा कि सर एक्सक्यूज मी, एवरीथिंग इज इंक्लूडेड इन योर फेयर, हम आपको कोई कहते इज इट सो, देन लेट अस स्टार्ट फ्रॉम द ब्रेकफास्ट। तो हम ब्रेकफास्ट से शुरू कर रहे हैं। वो जो पहले मॉर्निंग का ब्रेकफास्ट था पूरा डाइजेस्ट हो गया, फिर ब्रेकफास्ट पावर पॉइंट किधर खुल जाए, ये नहीं। हाँ, this is. I wanted to start with this quote. And this quote is from Robert Cook. Average person associates fungi with molds and mildews, mushrooms, bracket fungi, puffballs, rust, and smuts, etc. only. When Robert Hooke discovered the microscope and he examined different materials, now see how ingeniously he writes. I think there cannot be any better description than what he observed in 1665 and wrote in Micrographia. The blue and white and several kinds of hairy, moldy spots will not be unworthy of our more serious speculation and examination. And again, they consist of an infinite company of small filaments every way context and woven together to make a kind of cloth. इस अच्छी definition fungi की for a layman and for a beginner. And in 1665, if you go back 400 years back, cannot be there except this. Next slide. Now, I always dedicate it, your generation perhaps follows or doesn't follow, to the Almighty, to my parents, and two of my teachers. You have heard great about the last person who is Professor K.G. Mukherjee. I was his first student in Delhi University. And I did my master's with Professor K.S. Thind at Punjab University, Chandigarh. Very illustrious teachers. And we don't have teachers like them now.
He was the person who was in my selection committee when I was appointed as lecturer in the Mr. K. Singh was one of the experts.
And to all my, and how I could do something, a little bit whatever I could do, is because of these students. And there have been, because 30, 40 years is a long period, so many students come in MSc and MSc honors, join you and go. These are the 30 PhD students who have worked specifically for their doctorate degrees with me. Next, please. Next. So that is the title that I chose. Fungal biodiversity is an untapped source. And why it is an untapped source? Next, please. This is the outline. What is biodiversity? What is the significance? What is the source? What is the relationship between biodiversity and biotechnology? And what are different aspects of biodiversity? And because we are talking of fungi, how diverse are the fungi? This is the classical definition which has been given for biodiversity, and each one of us knows this. Next, please. And why study biodiversity initially? If you read the first preface or the introductory part of the book by C.G. Alexopoulos, it is from there. And incidentally, Professor C.J. Alexopoulos was one of the founder members of MSI in 1973. I was working on myxomycetes in Delhi University with Mukherjee sir. He was my PhD examiner, and he identified most of my specimens in Delhi University. And see how he says, how to justify when somebody asks you, what is the role of biodiversity? We always shun it away, that there is no need for biodiversity, there is no need for taxonomy, there is no need for all these systematics. But how simply and how powerfully he explains it. In the beginning, one understands function only through the structural features
You don't cut them or cut sections and then examine them or elaborate them. Structural features associated with function. One does not see photosynthesis. One sees the green leaves, oxygen bubbles, and eventually chloroplast. One does not see parasitism, but sees hyphae among host cells, haustoria, which had invaded the cells, and symptoms in the form of leaf spots, quick cankers or skin lesions, and sores. One does not see percentage of oxygen. So he covers pathology, he covers physiology, he covers biochemistry, which are incidental from the basic studies on biodiversity. Remember it. So percentage of oxygen, nitrogen, phosphorus, and carbon, but what one observes is fascinating streaming of plasmodium, but does not see the genes. So genetics, you cannot see the genes. So one does not see the genes A and A, but rather the two colonies which grow together to form zygospores, in which homothelism and heterothelism, how that was discovered. Next, please. And how is this biodiversity that we talk of, how it has all come about? From where have we received it? Biodiversity is inherent in every propagule. We always say we study in angiosperms. When we study embryology, we say 3 generations are locked up in a seed: the past, the present, and the future. And it is the seed which on germination gives rise to the huge tree, to the varied colors, to the branches, to the root system, to the shoot system. So everything is inherent in the seed. So biodiversity is inherent in the seed. It is not what we see in nature. It has some beginning. And, and this is not because that we are germinating the seed. The nature has planned it the same way. And what has happened is, what is the motivation? Motivation is for proliferation or multiplication. So every living organism strives to manifest life and enjoy nature by multiplication. We describe the characteristics of the living beings. And then multiplication and variation, that creates diversity that we observe. And how does it come? What is the motivation? A seed in the soil pushes itself upwards towards light. Don't see these wrong spellings. A chick, you have seen the hen laying egg and sitting on egg to provide the requisite warmth which cracks, and the chicken comes out. Chick cracks its enclosing shell to enjoy the outer life. Every crawling baby, if we talk of human beings, every crawling baby strives to stand despite repeated falls to attain its full potential. So all these characteristics that we describe and take credit that we have bestowed all these on organisms, they are what nature has inherently placed in every propagule of every organism, and same applies to the fungus. If a tiny fungal spore also cracks, crawls, and pushes up through the substrate to express its inherent latent potential. That is why we see the wide variety of fungi. That is how this variety has come up. So, why do we talk about biodiversity? If there was no biodiversity, it would have been a monotonous life. Why do we have changed clothes? Why do we have so many ornaments? different types, because the white light does not motivate us. We take it for granted. It is in the rainbow, the 7 colors when we see, that we become expressive and start appreciating the nature and the colors. So that is why biodiversity is more pleasing and more appeasing to the eye. Because whatever we perceive through the senses, that is reflected on the screen of our mind. That is how the life goes on. So we experience more joy in rainbow than in white light. When you have harmonium just placed like that, and if you play on that, the air that is pumped into it, that produces the different musical notes. We like them. Different fragrances in the flower. Why do we like them? Because of diversity. And the variety of food items than the single item. So biodiversity is pleasing. It is refreshing. It motivates our mind. Next, please. And all that we are using from biodiversity, that is not the source. That is the resource. Source is the mother nature. How mother nature is the source? Because nature has the potential, only nature has the potential of generation or creation, of organization or sustenance, of dissolving or merger. If you take the 3 words of generation, organization, and dissolution, it becomes God. Whether you believe in it or not, it is not an entity. It is a phenomena. And it is a natural phenomena. Nature has inherent this type of phenomena which creates life, sustains life, and merges the life into it. That is what we call as God with 3 potencies. Brahma, Vishnu, and Mahesh, we distinguish. And then Dr. Radhakrishnan, see how philosophically he explained it. God sleeps in matter because it is stone is unexpressive, breathes in plants, moves in animals, and speaks in man. Power of speech is only given to man. So that is why the man is at the pinnacle of evolution. Man is the highest or the highly evolved individuals. So therefore, how could nature give all these things to us and create biodiversity and still unite it? It is just like a shoot system and a root system. Whatever we see outside, this building has a foundation. We can't see the foundation. But this is unified and strengthened by the foundation on which it has been raised. So, nature observes no holidays. If sun does not come or rise for 1 second, the entire Srishti would be destroyed. So nature observes no holidays. Nature has created cycles. Nature has created seasons. We enjoy all those seasons. And it is all days for nature are not holidays. They are holy days. Because it observes the same discipline which is required of the human beings. Human beings are born. We are all born, not to born again. So a human being has to rise from humanity to divinity. He has not to fall from humanity to animality, just as is happening in different countries. And nature has a simple principle. on which it operates. Nature loves all and serves all without any distinction. That is the beauty of nature. That is the beauty of natural phenomena. And see, plants are the true role models of this love all and serve all phenomena, because in nature, tree gets sustenance from the earth, water, air, sunlight. In turn, it provides sustenance to millions of microbes, birds, animals, and man. The mountains and rivers, animals, and plant kingdom sustain one another along with different cycles. We have come from mega characteristics to molecular basis, and we find that all organisms are joined together. Those mega structures that we visualize at the molecular level, they are all the same. So there is unity in diversity. And that is a characteristic of nature. Next, next. This is how we, for our convenience, nature has no watertight compartments, and nature doesn't need to pigeonhole them into different groups. We want to study them, so we classify them. And that is why, and the viewpoints differ. Different viewpoints, different classifications, but broadly, we have the ecological diversity, taxonomic biodiversity, genetic biodiversity, and molecular biodiversity, which are the main themes of biodiversity studies. Next, please. But Professor Aneja yesterday said it is the same, but one missing slide is that he quoted that there are 50 lakh species. Oxford has put up, he has written a sentence that the little that we know, we require 4,000 years to describe the total estimated fungal diversity. And he has given that on an average, if we discover 12 species or describe 12 species per day, then these lifetimes are not enough. You require 4,000 years to describe all those species on that average. Next, please. So, how diverse is the fungal world? How much is the resource that we have tapped, even from 150,000 species that have been described? Not even a fraction of that. Fungal world is, and if you see fungi are more diverse than any other group. Be it plant kingdom, bryophytes, pteridophytes, gymnosperms, angiosperms, or the animal kingdom. The variety that you get in fungal world is unmatchable. You don't find that variety anywhere in any other group of plants. And how that is reflected even in their structural organization. What do we study in fungi? We study thallus, we study hyphae, we study reproductive cycles, and they grow. How do they grow? They grow in different environments. They grow in water, in air, on land, and like reptiles in between water and air, all habitats. They occupy all niches. Whatever niches are possible or impossible, you will not find any other organism, but you will certainly be able to locate some fungi there. So they grow in very diverse environments. Diverse environments match their nutritional requirement. Why do they occupy different environments? This is simply to get nutrition because they are heterotrophic. They lack chlorophyll. And it is the lack of chlorophyll which forces on them is lifestyle, But we classify them as saprotrophs or saprophytes, parasites, or symbionts. They don't do it for us. We utilize them as saprophytes. They act as scavengers of nature. If they were not there, then we would have been living in garbage huts, not in palatial buildings. It is because of that they utilize them, recycle them, produce enzymes, and then we use those enzymes for industrial purposes. So they are not secreting them for our purpose. We are creating hurdles in their lifestyle and utilizing at different steps how we can make full use of them for our benefit. So it is the nutritional requirement of these fungi which enforces on them a typical type of lifestyle of saprophytes, parasites, or symbionts. Maybe for thousands of years, they must have tried. In evolution, we always say this, that nature must have tried different paths and then adopted a path which suits and benefits a specific organism so that it can grow, regrow, multiply. We derive benefits from them. And then they show diverse morphology. You have unicellular fungi, we have multicellular fungi, we have septate fungi, we have aseptate fungi, we have xenocytic fungi, and in septate fungi, we have fungi which have a simple septum, we have fungi which have a dollipore septum, like bordered pits of angiosperms. So see the wide variety that is exhibited, and these are the criteria. These are the criteria that we have utilized to classify them. Until and unless we know the basics, how do we classify them? So we have to know them. We have to study biodiversity. If we do not study biodiversity, then we are living, I would not call it, but it is a fool's paradise. We have to study it. And why are we failing in our research? Because we are not attending to the basic problems of science and jumping over to molecular aspects. Until and unless we know the basics, you cannot jump over. Mind you, if you just work on one plant, but have a holistic and a logical conclusion approach, you do everything for it. Start with morphology. Go to then molecular level. No one stops you. But don't ignore the basic part and end up in mess. We have very many instances, not one or two, where one lab has given different molecular analogy, the second lab for second, the same organism, different gene mapping, and there is all sorts of confusion, even in classification. Because whatever we publish, they quote. Because we quote whatever they publish. But they have the basic integrity. And they publish whatever is correct. We just mix up things and then mess. So we have to have a sound basic system on which we should raise our aspirations and our molecular characterizations. They show diverse morphology. Then they possess equally specialized somatic structures. They produce rhizomorphs, they produce sclerotia, they produce pycnidia, they grow in acerbuli, They grow individually. So many diverse types are present in fungi, unmatched in any other group of organisms. Next, please. So they show diverse methods of reproduction. Asexual reproduction by fragmentation, fission, spongiospores, conidia. Conidia are produced in fruiting bodies, cinemata, sporodochium, acerbi, pycnidia. Where do you have these in asexual reproduction? Nowhere else. Sexual reproduction, they have planogametic contact, copulation, gametangial copulation, gametangial contact, spermatization, somatogamy, and then homothelism and heterothelism. If you closely look at this scheme of nature, planogametic copulation, where we have isogametes, anisogametes, and then oogametes. Call it isogamy, anisogamy, and then oogamy. So there is increased specialization and complication of the somatic structures. Holocarpic, the entire thallus participates in reproduction. Eucarpic, there is division of labor. A part of the thallus is set up for reproduction. So we have the simplest type of reproduction, the most complex oogamous type of reproduction in oomycetes traditional and in ascomycetes, and even in ascomycetes, the group of discomycetes that we used to have. There, they lose. Even in Aspergillus and Penicillium, where we have Talaromyces, Carpentalis, such type of sexual structures, what do we have there? In Penicillium notatum, in another species of Penicillium, Anthridium is there, but it is non-functional. Oogonium is there. There is self-arrangement of the nuclei. So they drive all the advantages what Anthridium would give them. So there is gradual evolution of sexual structures from simplest to more complex, and then spermatization, simplest in rusts, and then somatogamy. In higher basidiomycetes, what do we have? Simple two thalli. And here you can see the wisdom that nature reflects. You increase in complexity. When you produce sex organs, delimit them, you spend energy. Then nature must have realized that this is a wasteful expenditure. So they resorted to somatogamy. So somatogamy structures, there are no sex organs produced, simple hyphae. They come together and produce the effect of this sexual reproduction. Then homothelism, heterothelism, lot of has been talked about this. They have a unique parasexual cycle by which those fungi which we used to consider lack sexual reproduction, they derive all the benefits of sexual cycle through a parasexual cycle without expending any energy. Simple thalli, heterothelism, heterokaryosis, and then producing of haploid, meeting of haploid strains, formation of in 1 lakh multiplications, one fungus gives rise to a sexually reproducing structure. So that is how it is affected. So no, all these have a great bearing on biodiversity group and delimitation and evolution. Next, please. And this, what I said, this all is reflected in the classification systems, where we have tried to group them together, and sometimes we have simplified them. Now we have complicated them to the extent that a normal student who doesn't have molecular lab to work with will be confused. And that is why the term came that taxonomy is a tax on me. Why do they lose interest? Why do you lose interest? You lose interest because we do not know the basics. We do not know the basis of classification. And simply for our ego, self-esteem, I work on a group and then delimit all the groups in the same manner and propose a new classification. So this is all man-made classification. Nature has not given us these. Next, please. Now, टाइम कितना है? हो गया? हो गया. बस 5 मिनट लूंगा because I have just to rush through these. These were the basics I wanted to know. We have fungi used in baking and brewing industry. You are all familiar. Next, please. There are fungi which are used in fermentation of foods. This number, 9, 10 fungi. Next, please. Next. We have fungi which are used as food, the cultivated mushrooms and the others. Next. We have fungi used in medicine and antibiotics. Specific mention has to be made to Claviceps purpurea, which produces LSD and many alkaloids, which are toxic, cause St. Anthony's fire, which is known as a disease. Dieback, what Professor Rameja explained, that it is dieback, the drying starts from the tip, comes down, and the whole tree gets dried. Next, please. But this has very powerful alkaloids. They cause gangrene. But they have been used to produce medicine which is injected at the time of childbirth to check the excessive blood loss. That is why they have found use. Then fungi use, this is a new field. This field, in fact, came into existence only in 1995, especially with regard to the mushrooms. And this is the summary how all these have been used: dietary food, dietary supplements, and all these products. Next, please. And what do they have? Collectively, what do they have? Next, please. They are antioxidants, polysaccharides, carbohydrates, proteins, lovastatin, eritatimine, gamma aminobutyric acid, ergothioneine, and so many other compounds which we get from mushrooms. Next, please. Ganoderma lucidum. This is called linked Z in Chinese, and this is called Panacea. And wherever you see Ganoderma, a Chinese person, just as you have heard Pied Piper of the Hamlet City, a poem we used to have when we were in 10th or 11th class. So they will hang one Ganoderma and go to different areas because any disease, they will just crush it and eat it. It is not an edible mushroom otherwise. But it has so many compounds. This was the first medicinal mushroom in Chinese literature. Next, please. This is shiitake, Lentinula edodes. Next, please. No, this is shiitake, that was pleurotus. Next, please. Coriolus. Next. ये coriolus है कि पीछे वाला है? ये coriolus है. That is, I will get the name, forget. That is a medicinal mushroom which has been used. Next, please. Schizophyllum, small fungus which grows on all twigs everywhere, universal. But this is the only fungus on which the entire fungal genetics is based. Because the fungal nuclei, they are below the resolution power of a microscope. This has a slightly larger nuclei, and this was the first one which has been. Later on, and morphogenesis, you dry it, it will get dried. You wet it, it will expand, start multiplying. So used as a role model. This is Flammulina voluptuous, another edible cultivated mushroom. Next, please. Marshala Gucci. This sells for around 20,000 rupees a kilogram. Next, please. This is one of the most important fungi, cordyceps, Ghasidas in Pithoragarh and all these areas. This occurs. China abundant, Nepal it is abundant, and it costs 1.5 lakhs per kilogram now. And it has all the immunopotent potentialities. Next, please. It has a long history. Next, please. Then there are fungi which provide vitamins. Next, please. There are fungi which are used as beverages, organic acids, and fats. Next. Fungi producing enzymes and growth regulators. See the products. Next, please. Forming mycorrhiza and living as endophytes. You have heard of these. AM fungi. This is what Professor Mukherjee had drawn. This diagram is from Professor Mukherjee. Had a very good hand at drawing. So all the 7 types of mycorrhizae and are depicted in this with the characteristics. Next, please. Universal and most versatile mycorrhizal fungus first discovered Physolithus tinctorius. You have eucalyptus growing. Away from the eucalyptus roots, if you find something dung-like or white fruitings coming up, that is Physolithus. It forms ectomycorrhiza with not 1 or 2, 172 plant species. It is such a versatile fungus. Next, please. Lacaria lacata, another versatile ectomycorrhizal fungus. It forms mycorrhizal association with 52 plant species. Next, please. Theliphora terrestris. It is a nursery mycorrhizal fungus, important at the nursery stage. This also forms mycorrhiza with many tree species at the nursery stage. Next, please. Then there are fungi which are used in fundamental research which have been used. The biochemical genetics or the haploid genetics, which got the Nobel Prize is on Neurospora crassa. We have Physarum polycephalum, which shows whatever we know about physiology or biochemical nature of cytoplasm or protoplasm, all those studies are based on Physarum polycephalum. And Physarum polycephalum has been used because it can be multiplied and mass multiplied and protoplasm can be isolated. And there are many labs since 1962 at Wisconsin. There is a Cancer Research Institute in Germany. They are all working for anti-cancerous properties with this fungus. And Dictyostelium discoideum, they are not considered now fungi, but only mycologists study them. So we have a claim to call them fungi. So all morphogenesis, Stabularia and Dictyostelium, they are the organisms which have been extensively used. Next one. And these are the fungi which have been used as biocontrol agents. First natural biocontrol of butt rot in pines by Rishwath et al. In 1962 was devised on these two fungi. Next, please. Only practical. So there is a large list of fungi used. You have heard a beautiful lecture and slides on entomogenous mycoinsecticides. Next, please. And then mycoherbicides. Next. Then fungi used as biocontrol agents of fungi. There are so many. Next, please. So, what is the ultimate message that we derive from biodiversity? The ultimate message is all visible diversity is united through such basic characteristics as order, growth, development, adaptation, regulation or homeostasis, and energy utilization. The diverse forms are like the shoot system, which is unified through an unseen root system. And that is true of the entire nature, because we know in nature there are many, galaxies are many, but sky is one. Nations are many, but earth is one. Jewels are many, but gold is one. Beings are many, but breath is one. Castes are many, but humanity is one. Religions are many, but God is one, Ek Onkar. There is only one God. There are different aspects which have been depicted as different gods. So this is from where we have to take lesson and understand, internalize it, so that whatever we do, we follow nature, love all and serve all. So this emphasizes the mutual role of all, need for symbiotic relationship. And there can be no better example for symbiotic relationship than lichens and fungi. Next, please. So this is for you, how you have to take life. Life is a challenge, so meet it. Life is a game, but play it. Life is a dream, realize it. And life is love, to share it. And we may not be able to foresee a bad end as we had with when the compatibility was not there. So we may not be able to foresee a bad end, but we can always make a good beginning, because they could find compatibility and I could make the presentation. One should not only do the work that one likes. Take this as a sermon or a motive for life. One should not only do the work. Grumbling रहती है, मैं ऐसा करता तो ऐसा हो जाता, मैंने ऐसा किया होता तो मैं ये हो जाता. No, that is a negative, that gives you negative thoughts. Let those thoughts not come to you. Be an individual. Don't see at others. अगर दूसरों को देख के भी वो करना होता तो नेचर आपको अलग नहीं बनाती. We all would have been clustered into one, but we are different. So we should not only do the work that one likes, but also like the work that one has to do. हम अपनी यूनिवर्सिटी में सुनते थे, क्लर्क लगे कि मैं तो रजिस्ट्रार से ज्यादा क्वालिफाइड हूँ. न खुदा ही मिला, न बिसाले सरम, न इधर के रहे न उधर के. उसमें ग्रज में ही पूरी जिंदगी निकाल दी और न वो रजिस्ट्रार बन नहीं पाए और क्लरिकल में काम नहीं किया. So woods are lovely, dark and deep. Jawaharlal Nehru used to say this poem, but I have promises to keep and miles to walk before I sleep. So coming to this, what is the relationship between biotechnology and biodiversity? Biotechnology is blind without biodiversity. And biodiversity is lame without biotechnology. So there has to be developed a symbiotic relationship that you can develop, that one can understand, that we can evaluate only when you come up to that level and start working, thinking in a positive manner. You have long way to go. तो डोंट डू लाइक कॉकरोचेस. ये प्रोफेसर्स के लिए, सॉरी एक मिनट ले लूंगा, और प्रोफेसर्स के लिए कहा गया है कि एक प्रोफेसर हवाई जहाज में जा रहा था और एक बॉक्स में कॉकरोच ले जा रहा था, तो उन्होंने कहा सर यू कैन नॉट गेट डाउन. उन्होंने कहा नहीं, ये सारे फैल जाएंगे ये. तो उन्होंने कहा नहीं, बिल्कुल नहीं फैलेंगे. कहते नहीं, ऐसा कैसे हो सकता है, जानवर है तो कहीं भी फैल जाएंगे. उन्होंने कहा कि बिल्कुल नहीं. उन्होंने कहा कि आप देखिए एक भी बाहर आ रहा है कि नहीं. क्यों? कहते देखो ये यूनिवर्सिटी प्रोफेसर की तरह हैं, एक ऊपर चढ़ता है तो दूसरा टांग खींच लेता है. So don't be like cockroaches. See nature, help nature, and change your nature. And thank you very much.
I don't have words to express my thanks and to summarize the lecture of Professor T.N. Lakhanpal. Such an excellent speaker I have seen since I was a student. I am learning from him itself. Professor Lakhanpal is so talented, so and he is very much master in speaking. And there is no match as he a good speaker as you have seen yourself. So what is interesting about this lecture, that this lecture was not devoted only to biodiversity of Punjab. This lecture was devoted to what we call as जो human beings से हम बात करते हैं, उसके साथ ज्यादा relation था. He started with that and he ended with that. That is the masterpiece of this lecture. Another important, when I start with his lecture itself, of course I cannot be a good orator like him. So what is saying? A seed, we put a seed in the soil. The seed is having all the three generations, past, present, and future. You know, when we germinate a seed, that is all the three generations. And similar is the case with our soul. We talk, we is talking that God is one. We say soul is one. God is, all of us having the same. We are having the fragments of that very soul. All are having the soul. Whatever we are doing, that is because of our soul, means because of that God which is present in every one of us. And that soul or God is having all the three generations. We are having the past. You know, when we are born, the Pandit make our kundli. How that kundli is made? If we are do not having that past in our, then how the kundli is made immediately as soon as we are born? We are having the past. Then present, whatever we are doing, we are getting up that present. Then we are going to the future. The three generations of this. Whatever we are doing now, that will be carried in the future. Same is the case with the seed. If that God is present with us, that is the soul. Now, I was saying, what is the interesting thing? What is the interesting about that lecture? The biodiversity. We are talking of the biodiversity. Is Punjabis are such much diverse than any.
Then any other kingdom in this world, as the fungi are concerned, is talking of 12 million species. That will take not only, as we told you, 4,000 years if we have to identify as the fungal diversity is present today. Of course, we are not able to explore, and that will take generations. And if we talk of the fungi, as the origin of fungi is concerned, they are much millions of years. They were born millions of years earlier. He is talking of 1665. Robert Hooke. He was the first man. When we talk of the microorganism, it was not. These were not the bacteria which were seen by the Leeuwenhoek. These were the fungi which were first seen by Robert Hooke. Robert Hooke saw fungi much earlier than that discovery of the bacteria, which we are called as the animalcule. He gave the term animal tools. He saw the vibrating in that your tooth water. He saw that. The Robert Hooke was the first man who made the microscope. He examined the microscope, that molds. He saw that. Of course, later on it was found that these molds, the filaments or hyphae were not up there. They were up the Aspergilla and Penicillia. Later on it was identified they belong to the Penicillia. It was discovery of the molds is much earlier than the bacteria, whatever we are talking of the other living organism on this planet. That is, but what is the drawback in our system? Microbiology is dominating. Microbiologists are dominating. Microbiologists are not giving importance to the fungi that they were discovered earlier than these bacteria or viruses, whatever we are calling. They are and you are seeing the beautiful diversity. No other beautiful on this planet is as the fungi are. And the excellent of this paper talk is that Professor Lakhanpal has shown so nice, beautiful, and the pictures are excellent. You will not get such pictures on the internet too. And these are the most wonderful pictures he has shown in the biodiversity, and we can say that. Thank you. Thank you, sir. Thank you. Thank you. Professor Ati, sir.
Yes, sir. Yes, sir. Good afternoon.
Sir, we will continue the session without tea break. Please, you can take care of the next session.
What about Professor Krishnendu Acharya?
Sir?
Professor Krishnendu Acharya.
No, actually, we have. His health is not. No, his health is not good, it seems. So he is not available, and he is not here in the offline. And both online also he is not there. He is also not. So therefore, this presentation is not there, sir.
Okay.
Please. Yeah, we can continue, sir, the next session. As Mr. Acharya is not present here, so we can continue the next session, sir.
Okay, sir. Dr. Madhivanan, I am taking over.
Yeah, please.
Now, should we start, please?
Just a minute, sir.
Okay.
Coordinator is there. Surely. Okay. Okay. So, very good afternoon to all of you. Now we are getting late, 1 hour late, because session 5th schedule is 11:30 to 1 p.m. So we are 1 hour late, but there is nothing. In this session, we have a chairperson, Professor N.S. Atri. He is from Punjab, and now co-chairperson, Dr. C.K. Pradeep. He is from Kerala, and I think both are on online mode. In this session, we have 3 lead lecture by 3 eminent persons from different places of the country. So I request to our chairperson, Professor N.S. Atri, and co-chairperson, Dr. C.K. Pradeep. I welcome you, sir, for chairing this session here in Guru Ghasidas Vishwavidyalaya in the MSI meeting, 49th MSI meeting. So I want to request you to start by first speaker, Professor N. Matthew Venon. He is here. Okay. Okay. So I request Chairperson Professor N.S. Atri, please, something you would like to say?
I am taking over now, please. Yes. Hope Professor S.S. Duria and Dr. Santosh Singh are in place, because Professor S.S. Duria is coordinator and Dr. Santosh Singh is rapporteur. I welcome Dr. C.K. Pradeep, my honored colleague here from PBGRI, Lord Kerala, for this session 5. So I'll request all the speakers, because all the speakers seems to be offline and they are there at the venue of conference. So I'll request them to begin with to kindly restrict to 20 minutes time, as has been instructed by the Secretary MSI, Professor Raman. So to begin with, we have one lead lecture by Professor N. Mathivanan, who is Director of CS in Botany, Gundi Campus, Chennai. He'll be speaking on discovery of natural compounds as promising drug candidates against multi-drug resistant Candida species. So I'll straightway, instead of wasting time, I'll straightway request him to kindly please begin his talk. And I will request Dr. C.K. Pradeep to kindly summarize at the end of the talk. Just few lines.
Yes, sir. Yes, sir.
Yeah, please.
Yes.
Dr. Mathivanan, please.
Yes, sir. Thank you, sir. Thank you, Chairman. Thank you, Co-Chairman. And participants and members of MSI, I'm extremely happy to stand before you to make my presentation on Candida species, which are really becoming a major health threat in worldwide. And they are initially opportunistic pathogen, but now become a major pathogen, especially the immune compromised patients, especially in the clinic, and it is a life-threatening issues. And after AIDS and as well as the diabetic, and most of these patients are bound to get this Candida infection. Yeah, please, you can go ahead with my slides. Yeah. And actually, I'm a plant pathologist. I'm a botanist by education, plant pathologist by research. But later, because of some the pressure on us to work on medicinal plants, because the University of Madras has awarded the flagship program of UGC, that is UPE, University with Potential for Excellence, that is on herbal plants. So therefore, we have asked to work in the botany department, not only in the botany department, almost all the science departments were working on initially. And now we are continuing in the RUSA project also. Therefore, we have started working on the human pathogens. So my topic today goes on discovery of natural compounds as promising drug candidates against multi-drug resistant Candida species. So these species are developed resistant, even very recent, the antifungal antibiotics. That is a major bottleneck and issue for the treatment of candidiasis. Next slide, please. Madam, next slide, please. Okay, I already mentioned this is the second most important nosocomial infection worldwide, and they are causing superficial as well as the bloodstream infection, and rapidly develop the resistant in almost all the antibiotics whatever we are using, and the major drugs whatever we discovered already using extensively, they are not able to control these Candida species. So the important the resistant mechanisms is biofilm formation. Not only that, there are other mechanisms like the yeast form to mold form. Yeast form is a pathogen. The mold form is non-pathogen. Sorry, mold form is a pathogen. Yeast form is non-pathogen. So this is the issue, and not only that, there are resistant genes which are responsible for developing resistance for multi-drugs quickly in the pathogen. So therefore, these are type of candida. You can see this how bad it is, the infection. So it causes infection from top to bottom. Next slide, please. And for your information, this is a slide I have taken from the Gulati in the year 2016. They describe it affects top to bottom. Not only that, it's also infect the implanted material like catheter or heart valve and other implants. Such a bad level, so therefore it is difficult to treat them, and it also goes to bloodstream causing the candida disease. So therefore, ultimately leads to death. So then next. So therefore, it is our prime responsibility to find a way to control this. And you can see this pie chart. It shows that next to Acinetobacter, the candida is a worst pathogen, almost 20% in the total infectious disease. It is responsible for it. Next. So with this background, next. So we have set our objective to screen marine bacteria and medicinal plants for anti-candidal activity, and also to isolate, purify the bioactive compound, and also test the mechanism of action. So these are the three objectives in which we have started working with bioresources. Next. These are the bioresources. Actually, to begin with, we also procured the multi-drug resistant because we need to have a pathogen. These are the multi-drug resistant pathogen ATCC cultures. Candida albicans, Candida tropicalis we have used for screening. Next. And we also got the 7 clinical culture. So they isolated clinical, the hospital, Kilpauk Medical College Hospital is a government hospital. So they donated 7 cultures, and they didn't identify up to species level. They believe that they are Candida species. And for our your information, my information, they are multi-drug resistant pathogens. So therefore, then we started identifying them. Next, we did the antibiotic resistance susceptibility test. We established they are multi-drug resistant. Next slide, please. We go through. See, these are the drugs we have tested, and they are showing the multi-drug resistance. Next, after confirming the multi-drug resistant, we also did the sequencing analysis. Finally, we deposited the information in the NCBI, and we confirmed all our candida. 6 species we identified up to species level. 1 species we could not identify even through this sequencing. Next. Then we started our screening on bioresources. Next. And for your information, all are multi-drug resistant, whatever work we have carried out. And we used initially terrestrial actinomyces and marine actinomyces. Then we move on to calo-tolerant actinomyces and halophilic bacteria medicinal plants. So these are the biological resources we have used for our screening purpose. Next. And why actinomyces? Pretty well actinomyces are king of antibiotic producer. I do not want to highlight their importance. So produce various antibiotics. More than 50% of the known antibiotics came from actinomyces. And they are also known as actinobacteria. And my research started with this particular molecule, abamectin. Abamectin has been produced by Streptomyces avermetilis, which has been discovered by 2 scientists by name Campbell, William Campbell from John Innes Laboratory, and also Satoshi Omura from Kitasoto Institute, Japan. These 2 responsible for discovery of abamectin, for which they have been awarded Nobel Prize in the year 2015 for medicine, along with 2 UU. She is also been awarded Nobel Prize for the discovery of artemisinin. This is anti-malarial compound from Artemisia annua. So I was started working on abamectin initially. Then I had a contact and opportunity to interact with Professor Satoshi Omura in the year 1999, 2000. But later 2015, they have been awarded Nobel Prize. I was so happy. I had an interaction with him. Next. That was the starting point for my work. Then we initially working with the soil actinomyces, collected soil sample, isolated the actinomyces. It's a big story, but then cut short because of want of time. I'm not going to detail everything. This is the work we have carried out initially. We isolated soil actinomyces, more than 200 different actinomyces, not this Malarvidi. It's my first student, and now she is in USA working in Reading University. Next. Next. And then We also moved on to marine because of certain the important features of the marine microorganisms are having. So we have used these locations in Tamil Nadu, especially the Bay of Bengal, Pichavaram mangrove. You would have known this, Adyar, Echuary, and Kelambaka salt pan, where salt is being produced. These are the locations we collected soil samples. So there is an institute called National Institute of Ocean Technology at Chennai. So they helped us. This helped us to venture into sea to collect the sample from different locations. It's really a very good experience, and they have onboard a lab also to analyze the physical-chemical parameters. And we collected sample from entire Tamil Nadu coast, and these are the marine actinomyces. We initially collected, one of my students has collected, and we also collected the allotolerant actinomyces from the salt pen sample what we have collected. And for your information, and I'm not presented everything because the soil actinomyces didn't show much activity. And also marine actinomyces, they show only 17% of the soil actinomyces, and also 30% of the marine actinomyces were showing activity against various human pathogens, including Candida. But when we come to the haloton actinomyces, many of the haloton actinomyces were showing activity. So all these 30 plus cultures, we have did the identification. All this information we have deposited in the NCBA website, and you can see here many of the haloton actinomyces crude extract, just the crude extract showing very good activity. You can see the incubation zones, very good activity. Then cut short the story, and one of my students now, Dr. Abrami, she has worked on this particular topic, and finally she has isolated a purified compound. Next, purified compound. It's not coming up. Okay. This is a culture, Streptomyces celestris. So each culture we have accession number, MML. This is a microbial metabolite of my lab. We have accession number. So this particular culture was showing very good activity against the multi-drug resistant Candida species. And this is a compound we have isolated and purified. But this compound is showing only moderate activity, not the very high activity. To develop further, but anyway, the bottom line is the drugs whatever available they are unable to control. But this gave a moderate.
There is an audio problem, sir.
They are also showing antioxidant and anti-prolifertic activity. Next, quickly go. Next. Next slide, please. And you can see here, I told so how they are very effective. Almost all the cultures are showing very good activity against Candida albicans. Every culture, each and every culture. Therefore, you can count these halophilic bacteria for any kind of biological activity. I'm guarantee for it. Next, so very good activity. And this is a particular culture, Halomana salinifolioideae. One of my students, Dr. Ramprasad, he has done the work on this particular organism, and he has purified one compound. Next. This is a particular compound he has purified, and it was showing very good activity. This is another result. And then because of we received another project by UGC, that is Center with Potential for Excellence in the particular area, to work on the human pathogen. Next slide, please. And we started working with the multi-drug resistant Candida, especially on medicinal plants. Next, move on to next. So these are the plant extracts. Actually, we have collected 121 plants. They are rare medicinal plants we collected from the Western Ghats with the help of the local tribal people. And we made 180 extracts out of these medicinal plants. Actually, we collected for these medicinal plants for anti-seneca activity. That is a different story. We established the anti-seneca activity of the medicinal plants. and make use of those extract, and we screen for anti-candidal activity. Next slide, please. And initially we screened against two pathogen, the Candida albicans and also Candida tropicalis. Next, further, move on further. You can see these are the results we obtained after the initial screening. Next. And out of 180 that we screen for anti-candidal activity, whatever the plant extract showing high activity, 26 we have taken into consideration, and we bioassayed against all 9, 2 ADCC as well as 7 clinical isolates we have screened further. Then we shortlisted 12. Next. These are the 26 results I'm presenting before you. Next. And then we moved further. Then we selected out of 26, 12 plant extract which are showing much activity. Then again we did the screening by using 100 microgram per ml concentration using the MTC assay. Then we shortlisted the 404. Next slide, please. That will show you. These are the 404 showing very good activity against all the 9 culture. You can see here minimum of 51 and maximum of 91. And other lowest activity is 30 of 78 plant extract. So therefore, we have chosen 404 is showing very consistent activity. Next. So 401 plant extract. And again, we did the MIC. You can see here it is showing very good activity at lower concentration. Next. It shows about the MIC of minimum of 10 microgram or 20 at the maximum 80 in particular pathogen. That shows that this particular 404 plant is having very good activity against the Candida, multi-drug resistant Candida pathogen. Next. Then we moved further to next. So this is a summary. So we finally shortlisted plant 404. Next. Then mode of action. What is the mode of action? So then we did the crude extract alone for study the mode of action. As I mentioned, it is different way it is actually developing resistance. One of the mechanism is next slide, please. The ergosterol biosynthesis. This is ergosterol is responsible for the yeast to become a mold for mycel formation. So we wanted to test whether these compounds are inhibiting the ergosterol biosynthesis. Next. Then we have isolated the ergosterol by using this methodology. There is another calculation also there because of time pressure. I'm not presenting how we have done, but this is the procedure we have followed. Next. Then we found out, you can see this graph shows how it is really inhibiting the ergosterol. You can see here very significantly our crude extract, the minimum inhibition concentration we used, it is significantly suppressing the ergosterol biosynthesis. That shows that it's having the mechanism. Next. And not only that, there is another important the characteristics is to hyphal form. When if it is not pathogen. If it is forming hyphal formation, the mold form, it is causing the disease. And we have tested and we found no or very less hyphal formation after treating with our the medicinal plant extract. Next, 404. You can see here, there's a left panel showing control and right panel is treated. All are as it is in the yeast form, mold form is not allowed. So that shows it is also the inhibiting the yeast to become a mold. The next, you can see all 9 pathogen. Next. These are the 9 pathogen. Next. And coming to the biofilm, biofilm is also another important resistant characteristic. So if form biofilm, then antibiotic cannot penetrate and cannot cause the death. So therefore, we wanted to check whether it is having biofilm formation. It's one of the very virulent factor. Next slide. You can see here, this is the microtator plate assay we have done, and we also quantified. Next slide, please. You can see we have quantified. This is a formula is already there, and based on that we have done. You can see significantly it is actually inhibiting the biofilm formation. So therefore, these medicinal plants, we are thinking they are all for other activities, especially people are working on anti-cancer activity or other activity, but you can also think of antimicrobial activity by selecting the medicinal plant and medicinal plants. And we also wanted to see whether it is having any cytotoxicity because it's affecting the pathogens very effectively. So therefore, we wanted to test again the vero cell line. We found they are not showing toxicity in the vero cell line, normal cell line. So therefore, it is safe to use it. Then we moved further to purify the compound. Next slide, please. And the IC50 value is 1580 microgram, 1.5 mg. That is fair enough to show that is non-toxic. Next slide, please. And then we moved further to purify and characterize the bioactive compound. And next, you can see this is the plant. I'm not presenting the name of the plant because now we are in the process of patenting our data we have given to the attorney, now writing the patent. So therefore, I'm not disclosing. Maybe after submission, I will disclose in the next time. This is a plant. At least I'm showing the root. This is the root, and we made a powder and made a metallurgic extract. Next slide, please. And you can see these are the different planting pattern in the TLC we have seen. Next. And then you can see we did the column chromatography to purify the compound and see the fraction, 20 fraction we obtained all the fractions. Most of the fractions are showing activity. Out of these, fraction number 9 was very effective next against all the pathogen. Next panel, next. Yeah, this is the data. See, 9 showing good activity. The minimum is 16 and the maximum is about 23. So this showing very good consistent activity against all the pathogen. This particular fraction number 9. Next. Then we purified further and we obtained 2 pure compound. You can see here 2 pure compound we obtained from this fraction number 9. Next. And you can see here very clearly the crude extract, that is 1 mg we have used, and fraction number 9, 100 microgram. The compound on 25 microgram, compound to 25 microgram. You can see all these are showing good activity against almost all the 9 pathogen whatever we have tested. Next. All the pathogen. And see, this is a table shows the minimum the inhibition in mm. Minimum is about maximum is 24 and minimum is 22. So that shows more than 20. It's a very good activity in next slide. So then to sum up, we have purified 2 compound. Out of 2 compound, 1 compound we have fully characterized. The physical price is a pale yellow oily substance and is soluble in chloroform, ethanol, DMSO, and ethyl like that. Structure has been elucidated. We have structure right now. And the molecular formula weight are also been determined. So this is a brand new compound. It's not been reported so far. It's a novel compound. Therefore, it is qualified for patent. So therefore, we already submitted for patenting. Next. This is a summary of our research. 180 plants, and we started 26 based on the activity, then 12. And out of 12, 404 was showing very good activity. And it was inhibiting ergosterol synthesis as well as inhibition of yeast to hyphal formation, and biophil inflammation also was there. And we see there is no toxicity. And the TLC we purified, they call 20 fraction we obtained. The fraction number 9 was very effective. Then out of fraction number 9, we purified 2 compounds and characterized. Since it is a novel compound, patenting is in progress. So therefore, I'm not presenting. Sorry for it. Maybe next time I will be presenting the data. Next. Thank you. Actually, I'm a pathologist now. I'm working on. So the many more interesting observation we made. Actually, we did anti-snake venom activity. We already got the patent. It's a global patent. We already obtained it for the first time. We patented it. It is funded by a company fully, and if it is going for commercialization, 30% royalty they are giving to the University of Madras, the profit. That's the one achievement. Another one is very recently we have completed along with the anti-candidal activity, anti-mycobacterium. That is a TB activity with the support of National Institute of Research in Tuberculosis. Previously it was TRC, Tuberculosis Institute. Now it has been elevated as a national status. With their collaboration, we have established the extract of the plant, edible plant, which is showing very excellent antimicrobial activity for their surprise. It's a multi-drug resistant, extremely drug resistant, as well as the latent form of mycobacteria. So therefore, with this information, I wanted to inform you, the plants are source of novel compounds. So please do in this area research. Definitely, it will give you dividend. With this, I take this opportunity to thank profusely the organizers and the chairman and co-chairman, all the participants for your support in listening. Thank you. Thank you, Anand.
Thank you, Dr. Mathivanan, for excellent presentation and for keeping time. You took exactly 20 minutes. I request Dr. Pradeep to please say a few words just to summarize.
Sir, it's an excellent work, and we have all know that candidates issues, and sir has been extensively worked from the soil actinomycetes. Another important group is marine actinomycetes and also medicinal plants. So we can hope that shortly we will get a new compound to tackle this issue. All the best, sir, for your wonderful work. Thank you.
Thank you, sir. Thank you. Thank you. Thank you, sir. Thank you. Thank you.
Great lesson from your talk
Thank you,
to
sir.
evaluate the medicinal compounds for components. Thank you very much.
Thank you, sir.
So
Thank
we
you.
move on to the second talk by Dr. Anupam Dixit, who is from Allahabad. He is one of the pioneers, great scientist, and he'll be speaking on management of human pathogenic fungi using botanicals, almost a topic with the similar connotations. And I will request Dr. Pradeep to kindly help in summarizing afterwards.
Yes, sir.
Professor Dixit, sir.
Yeah,
Please.
please. New BSP करके होगा. Yeah.
As the name indicates, his lab itself speaks volumes about the type of work he's doing, biological product lab.
Yes. Anyway. Mr. Chairman, Professor Atri, sir, am I audible?
Yes, sir, you are audible. Please kindly manage within 20 minutes. This is a request from MSI. Thank you. Please, you are audible. Your slides are visible.
Slide यहाँ पे दिख रही है.
ये तो मेरे ख्याल पिछली स्लाइड से
I'm grateful to the authorities of the Mycological Society of India for providing me this opportunity to interact with the delegates who are online as well as the delegates who are present in a physical mode here. So,
sir, slides are not visible. I think Dr. Mathivanan has
they
not
are
shared
in
his
the
slides.
process of projecting.
Okay, Dr. Mathivanan should unshare his slides to begin with, and subsequently your slides will be uploaded. But still, Dr. Mathivanan's slides are being projected. Stop sharing the slides of earlier presentation.
मेरे लैपटॉप में नहीं इसमें चलेगा.
पिछली प्रेजेंटेशन की स्लाइड्स अनशेयर करनी पड़ेंगी पहले.
पहले वाला ना.
क्या हुआ?
Now everything is okay now. You can see.
Yes, sir.
So
make this full screen.
Full screen, sir.
This is the University of Allahabad. This is only 135-year-old university. I don't know how many of you are knowing about Allahabad, or it is a new name. Its new name is Prayagraj now. So it is situated at the confluence of Ganga, Yamuna, and Saraswati river, and there this Saraswati it is in the form of the Oxford of East. And as you know, the India is known with the Qutub Minar and Taj Mahal. Like that, Allahabad University is known with this Vijayanagram Hall and Tower. So I'll be talking on management of human pathogenic fungi using botanicals. Next, please. Next.
Kindly make these slides full screen. Yeah.
What happened? उसको नीचे पुश करिए ना.
चलो बोलिए सर, आप कंटिन्यू करिए, दिख रहा है वैसे.
Just a minute. Then this problem will be there, na?
Sir, you can continue.
Yes, yes. Whatever time you will be giving, sir, Professor Atri, sir, I'll be completing within that time. Huh?
Please, sir, please,
So,
please.
I'll not be snatching time from anybody. Huh? And if some problem is there, I have to sacrifice something.
आ रहा है सर, आप अच्छा ये
वो मॉर्निंग वो जो लड़की बैठी थी वो तो जल्दी जल्दी कर रही थी भाई. वो हैबिचुअल हो गई थी. So this I was talking about the Allahabad. How many of you have visited Allahabad? And number of people they have not visited. So I'm inviting all of you. And this Allahabad, you know, in this university, youngsters they will be definitely knowing Allahabad because of Amitabh Bachchan. His father was in the English department there. Harivansh Rai Bachchan there. I am dedicating this talk to the Lord, to my parents and my guru, Professor S.N. Dikshiji, who is no more now. My mother is no more now. Only my father is there. I am getting his blessings. He is 95-year-old now. And Professor S.N. Dikshita, he was a student of Sagar University. And then Professor B.M. Jory took him to the Delhi University, and he did PhD under the joint supervision of Professor B.M. Jory and Professor Panchanan Maheshwari. So in that way, whatever I am today, it is all because of their blessings. So I am dedicating this talk. Next one. Next one. If every time this thing will be happening, then my only with dedication everything will be finished. ये क्या प्रॉब्लम हो रही है भाई? ऐसे कैसे काम? So, topic is management of pathogenic fungi. How we can manage it with the botanicals. So, with the objective of providing eco-friendly disruption. अरे आके देखो यार. अब तो प्रॉब्लम नहीं आएगा. आप पहले एक बार और देख लीजिए. You just move it.
देख लो ना इसको फुल. हाँ, now it's okay.
Okay.
It's okay now.
पीछे चलिए. Back. Reverse. So with the objective of providing eco-friendly botanical antimicrobials, different aspect of diversity in terms of richness of secondary metabolite in various groups of plants, that is from algae to angiosperm, as well as their potential towards management of pathogenic fungi of diversified categories such as unicellular and mycelial ones will be elaborated.
So the biotechnological tools have also been used to make the research economically viable. Product development mechanism of action as well as patenting issue will also be addressed. Next one. So I have just told you from algae to angiosperm. So these are the few examples of antifungal algae. These are the you can see yourself here because of the I have already captured some time. Then next one, the antagonistic fungi. इसके पीछे चलिए जरा, back one. Algae. Then we are coming to the switch over to the fungi. Next one, antifungal algae. Fungi, they are Trichoderma harigianum, Trichoderma hematinum. These are the different species of the Trichoderma and Glycodidium variants. They are, with these fungi, we can manage these target pathogens. Next one. Likewise, antimicrobial, we have worked on lichens also. Very good results. They have been noticed in Heterodermia, Ramelina, Sterochelon, Aspnia, Parmelia, and Evernia. We have collected these lichens and extracted the secondary metabolites and tested against these pathogens. Next one. Likewise, the antifungal bryophytes are also there. Why I am telling you, depending upon your, because this conference, it is being held in botany department, so the youngsters who will be having a different interest in different disciplines, algae, fungi, bryophyte, pteridophyte, they can use them, and their secondary metabolites can be used for managing these pathogens. So we have a very good result have been noticed in Trichanthes striatus, and we have collected Professor Dr. Essential Vaso, who was the student of Major N.S. Parihar, who was an authority. I collected the samples from him and got a very good result in managing these pathogens. Next one. Antifungal pteridophytes, the Teres, Pteridium, Lycopodium, Selaginella, etc. They are also, because in these plants, in bryophyte, you will never notice any fungal infection. It means there is, in nature, they have synthesized such a wonderful compound which can be used for controlling these pathogens. Then, likewise, in pteridophytes also, very little infection we can notice. Next one. Antifungal gymnosperm, I did PhD. The main plant was at that time, Cidrus desera was there. So way back in 1976, I worked on this. In 1980, I've been awarded a PhD. And in the morning, Professor Lakhanpal sir has shown the photograph of Professor K. Singh. He himself was my examiner. He conducted the Viva after coming over from Punjab to that place. And one has gone to the Canada at that time. So this is how my supervisor tuned me for working in these directions. Next one. Antifungal angiosperm. Later on, I also worked on antifungal angiosperm from dicot, Trachisperm MA, Rhabdosia melissoides, mentha. And these are the aromatic ones. You can see. Then the Acacia, Eucalyptus, Artemisia, and Equus. Likewise, in monocots also, in Sembipogon, gingiva, while working in the CIMEP, I worked on these monocots also, like lemongrass, citronella, etc. And we got secondary metabolite patented also. Next one. The various techniques have been used for assaying the antifungal screening, the dual culture method, It is only for the youngsters. I am telling you, modified spore germination inhibition, modified paper disc technique, poison food technique, inverted petri plate, but these techniques, they took a lot of time in getting the getting the final result. So with broth micro dilution method, which was recommended by CLSI, formerly it is known as NCCLS method, with this we can get the result within 2 days time. So within 24 hours, in the case of the bacteria, in the case of the fungi, we can get the result within 48 hours. Next. Then the flora, you have seen. Next one. Why we need biotechnological intervention in plants? Because the population rise is there, enhanced disease, less land for agriculture is there, malnutrition, desire to learn the secret of the nature. So what is the demand? We need more plants, we need quality plants, we need healthy plants, we need to conserve plants, we need more natural product-based medicine. So we must design novel plants, superior in trade, productivity, upgrade the genetic machinery through novel gene combination in otherwise acceptable genomic background, add value to our existing crop uses. So what dictates a cell to divide follow a various developmental pattern of growth, how cellular tasks are performed through the dilates of genetics and metabolic chemistry, how can the fate of living cells will be modulated experimentally. Next one, sir. So these are the metabolites, the primary metabolites and secondary metabolites. Primary, they're universal compound found in plants that are known as sugar, protein, amino acids, purine, pyrimidines, etc. And secondary metabolites, on the contrary, are other plants chemicals that vary in plant species and also do not appear to have any essential role in metabolism, like alkaloid, terpenoid, phenols. Secondary metabolites are said to have a first been a photosynthetic byproduct or chemical waste, proving fit those plants creating them. Next one, sir. Next one, please. So these all are details about the secondary metabolites. Next one. Next. Secondary metabolism is a highly dynamic process. About 88,000 plant secondary metabolites are known today. They are formed, stored, and catabolized under developmental and environmental influences, often synthesized and stored in a specialized cell tissue, synthesized via rigorously controlled fluxes of metabolites between the several cross-linked routes. Next one, sir. So these are the secondary metabolites: alkaloids, saponin, isoprene, terpenes, flavonoids, glucosinate, and phytoesterols. Next one. Next one. So medicinal plants used by traditional community are in Ayurveda about 1,700 plants, in Siddha 500, Unani 400, in Amchi about 300 plants. They have been known to be used. Next one. The aromatic plants from which the essential oils they have been extracted, like these are the essential oils, citronella to basil, and these are the business volume. You can see the word market of these essential oils. Next one. Estimated world production of essential oil in tons, you can see these. Next one. Estimated production of essential oil value in the US $1 million. Next one. The collection of volatile plants from different regions of India. I'm just giving you the example. Like Agel mormula, cinnamomum, eucalyptus, mentha, Phoeniculum vulgare, and Trachis eme, and majority of them, we are using it in one way or other. Next one. So these volatile plants collected from the present history, these are the different plants. And in Allahabad, in Prayagraj, the guava is very famous, but we have not used that. The waste leaf of guava, they have been used and got a very good result. Likewise, in that in turmeric also, the only the haldi which is being used for that rhizome, but aerial parts they have been the waste one. So we have collected those essential oils, and they are very good antifungal properties, and we can use them in a various way in managing the fungi. Next one. So extraction of essential oil, we have collected this, and in our botany department since the time of Professor Pant, Dividarshan Pant, he established there the culture of the excursion. So after joining there in 87 in University of Allahabad, we developed this biological product lab there. But later on, when the senior persons were not there or they were reluctant in going for excursion, so since 1999, the department gave the responsibility as for managing the excursion. So about 150 students of MSc, they've been taken to different parts of the country, and we have gone even to the Adokram area in Nathula area also in Sikkim, and you see they have been trained. And this is the Avdesh Kumar, a very efficient collector at that time, not administrative collector, the plant the students they have collected, and because of that enthusiasm, he got a degree, and now he is an assistant professor in Mizoram University. So through Clevenger apparatus, we got this essential aromatic compound, extracted essential oil, and we got it in this way, and we kept it for longer duration, and they are even active even for a year also. Next one. So NCCLS method, I am telling you, this is the microtitle plate reader. And with this, we can get the results in 96 well plate within no time, within 48 hours, we can get the result. And of course, for this, we have to write a project to DST, and only then we could able to get this, because the university was not going to sponsor this. Next one. So with this, we have got the IC50 and minimum effective concentration like that. Next one. So this is how we have collected the data. It is for the investor, I am telling you. Next one. Technologies for agricultural as well as human health. How for harmful pathogen. There are fungi, because this is the mycological society. The fungi, they are field fungi, some are storage fungi, or this few fungi, they will be coming through insect also inside. Then the human pathogenic fungi. So in that biological product, we are developing the biological product for agricultural health as well as for human health. Next one. So field fungi, these are the causal organism of these diseases. You can see it. Next one. Like wilt root. Then tomato seed caused by rhizoctonia. Next one. Sheath blight of paddy, rhizoctonia. Next one. Damping off of a brinjal seedling. Next one. In guava wilt, you can see this is caused by Fusarium actually. Next. Wilt of Farrer, Fusarium odum. Next. So Fusarium oxysporum and Fusarium solani, how they can, you can see how they are growing in a petri dish. Oh, this is for the youngster, I am telling you. Next one. the method for disease management, the chemical, how these are chemicals which are non-renewable, non-biodegradable, petroproducts are there, sometimes phytotoxic and commonly costlier and with many side effects. On contrary, the biological control will be, they are renewable, biodegradable, they are plant-based, no adverse effect, and economically viable and eco-friendly also. Next one. Here I can give you the example. When we have gone with our product to the village area in the farmer, we have seen that they were spraying the cypermethrin on the radish, on the potato crop. Like, why you are? It is a carcinogenic chemical. You see what farmer told? He said, "Okay, sir, this is for selling, for our purpose. We are having a different area for our own consumption. So they are playing with the health of the common people. So we have to educate them." And one of my students will be later on giving the talk how the illiterate people, they will be educated on these issues. Chemical pesticides have caused reduction in biodiversity of natural enemy, outbreak of secondary pests, pesticide-induced resurgence and degradation of the natural resources. Next one. So this is the consumption of pesticide in India and other developed countries. You can see we are using how much? Very little. Next one. Since this is synthetics of the control of harmful fungi for human pathogens, these are the synthetics, azole, ketoconazole, glycofulvin. When they will be taken orally, they are having a side effect. They are even immunotoxic also, econazole and fluconazole. And for plant pathogen, we are having these dithiocarbamate, Vitavex, Plantavex, Bevestin, etc. And aluminum phosphide, this is also called sulfos, and it is called the black poison. Next one. So they are having the residual toxicity like endosulfan, etc. That is why some chemicals like aldrin, etc. They have also been banned by US people because of the residual toxicity. Next one. You see, even vultures, they are talking. They मुझे भी रासायनिक उससे टॉक्सिसिटी से पैरालिसिस हो गया, लगता है अपना वंश ही समाप्त हो जाए. They are discussing like that. So it is all because of the DDT mixed with that non-vegetarian component. They are when they are consuming it, they are dying. And you see everybody, youngsters, they are for Pepsi and Thumbs Up, for cold drink. And what preservative they have been used, they are having a residual toxicity. Next one. Next. So this Trichoderma viridae, this is the common biological control agent. So number of soil-borne pathogens, they can be managed with this. Next one, Trichoderma helgianum, that used as a biofertilizer. Next, and biopesticides. This is the mechanism how they can be, this filament, mycelium can be controlling them. Next. This is the mechanism of action of rhizoclinians sclerotium hyphae coiling with Trichoderma helgianum. Next. Next. The mode of action, previous one. mode of action by producing secondary metabolite. You can see the dual action, dual culture method, inhibition of fusion by trichoderma strains. Next one. And you can see by using trichoderma hereditary, we are getting a very good crop. Next. This is the biofertilizer as a glucuridium variance. They can use. That is why in Allahabad we have developed this. Allahabad is known for Sangam actually. So we have developed the product where biofertilizer come biopesticide. Why we give that product name Sangam. Next one.
Kindly unmute kindly.
Next.
There is a disturbance. Kindly unmute.
Next one. आगे बढ़िए भाई. So this is the mass culture of glucuridium variance in sorghum grains. Next. इसको आगे बढ़िए. So for managing the field fungi, we can see the wind complex, the field, next. Next.
Kindly mute yourself.
You can see there are good Next. Intimate pathogenic fungi, the Beauveria bassiana, they can be used for bark, caterpillar, fruit and shoot, borer, etc. Methylene, these are the bioagents which can manage these insects. Next one. So what are biopesticides? Biopesticides, that is the microbial pesticide and biochemical pesticide. This is the integrated approach. Next one. What can biopesticide do? A fungus that kills a fungus, disrupting chemical signal, stopping development, and same as chemical pesticide. Next one. They are less toxic, only affect target pest rather than the large spectrum, very effective in a small quantity and decompose quickly. Next one. So they have been used like this. Application varies from spray, powder, or genetic injection. Next one. So cell wall degrading enzyme, lyses, proteases, chitinases. So next. They are producing a toxin specific to pests, causing disease, preventing establishment of other microorganisms through competition and other mode of action, interfere with the growth or mating, and pheromones that repel or attract. Next one. They are effective against aphid, green plant hopper, like that. Next one. They are also effective against these. Next. Beauveria bassiana cultures one can see and how microscopically one can observe it. Next one. So this is how this biopesticide or biocontrol agent can be made and they can be sold to the farmers. Next one. Next. So Biological Product Lab will provide a trained research scholar, eco-friendly technique and training facility. And through non-governmental organizations, self-help group, manpower and extension worker and financial support. This is how that lab research can be reach to the land. Next one. We have developed this AU derma at that time. Next. The ketonil. Next. We can escape because I have already explained it. Next one. So these are the insects with which the conidia and its mycelium or its spores with which they can go inside the stored grains. So and they are causing the mycotoxins and they are very injurious for our health. Next one. Next. So one thesis was there with Dr. Amartya Shukla. He worked on this.
And another person, he is Ramesh Singh. He is in Sardar Patel University. He was next one. Then we have developed the fumigation technique, how that natural fumigant can be tested. So with this method, they tried it, and later on we have also tested in the warehouses also, and developed the substitute of the self house also. Next one. So this was only to show how this insect is inoculating that conidia and spores inside the grains. Next one. These are the side effects of the synthetics. Next, we have gone to the warehouses where the testing have been made. Next one, for managing these pathogens. These are formulated products, active consists citronellal, and this is the chemical formula, and cost was very little at that time. Next one. Next, we have also tested the organoleptic test, how that natural fumigant, whether such treated gain can be consumed or not. For that, that organoleptic test was conducted. Next one. You pass it on. Next one. Now I'm coming over to the human pathogenic fungi. These are the dermatophytes, nothing but three genera: Epidermophyton, Microsporum, and Trichosporum. Collectively, it is called dermatophyte, and they are called causing superficial infection. While deep mycosis is also there. We have tested all these, and when I started this work in the Central Institute of Medicine and Aromatic Plants, when I was directly working with Dr. Akhtar Hussain, the director of that institute. When he noticed that deep mycosis pathogen also I have tested, he immediately came to that lab. Where are those pathogens? You will die. You will not be. What would be your fate? So he immediately got it autoclaved. Okay, you have to kill it. I got it from AIMS at that time. So only he allowed me to work on the superficial pathogen so that nothing will happen to our health. So this cytilidium, it is also called causing the infection on the nails. That Mrs. Shai, she is sitting here, Mamta Patra. So she worked under me at that time. She worked on onychomycosis. Then next one. So medically, these dermatophytes, doctors, they call it as a tinea. So depending upon the site of infection, it is called tinea capitis. You can see. Next one, on the scalp area. Then on those who are having a beard, like you, Palsa. So they should be, they will be very cautious, because nowadays it is a fashion with कोहली की वजह से सब लोग रखने लगे. So because chances are there, conidia and mycelium, you can also be very careful about it. In your Barbie. Next one.
Sir,
So,
kindly summarize, sir, please.
Okay. I'll follow your instructions, sir. So these are the dermatophytes. Next one. So how we have developed the formulation, and with that, how the treatment after the application of that formulation. This study we have conducted in collaboration with Dr. Professor A.K. Bajaj. He is no more now because of the corona, and we have a very big loss during that because he was a medical. Because dermatologists, they use much time for getting the money from the patient instead of the wasting time in research. But he was very much dedicated for that. Next one. So, and you see, Malassezia dander. It is very important. Malassezia unicellular yeast-like fungi.
they are causing either hypopigmentation in the skin, and that is why whenever I was using in the guest house or in the train, never I used the bedroll. I always kept my sleeping bag with me. Here also I am using that because that unicellular is like fungi. They can either affect our skin or the dandruff may be caused. I'm not able to see Professor Aneja here, and my student Bohi Shukla, she noticed that he is having a lot of malaria or dandruff on this. So I'll tell if he would have been here. She told me that I can get those samples to culture them. Next one. So there are different species of malaria are there. About 19 species are there, or out of which about 10 species they are in our lab. We are maintaining, and for maintaining this culture, for cultivation of this malaria, I have given the problem to Siddharth Datta when Bengali fellow, and I had gone to King George Hospital. London at that time. Dr. Medigili, she was expert in this. So I learned the technique for cultivation of that. But those were having media non-vegetarian component. That is why some foul smell was there, and lab was not able to. Lab person they cannot work. I told Siddhartha, nothing doing. You have to modify it. Ultimately, we succeeded in modifying that medium, and that medium we also got patented. Next one. And that Siddharth Datta was a non-vegetarian, मछली खाते थे, तो उसको कोई प्रॉब्लम नहीं होती थी. But बाकी even I don't take egg, तो प्रॉब्लम होता था. Next one. So these are the different species of malaria. We have to be very alert. Next one. Next. So how we can test it? We are using the sellotape for pasting the papers, etc. That sellotape, if we will put it on an infected area, and after that, if we will taking it and placing it on the slide, directly we can observe. We can observe the different species of malaria. Next, sellotape method. Colony of malaria in phase contrast microscope. Next one. So this media was patented in 2002. And next one. This is about the dandruff. I have already explained. Next one. I have to follow the instruction of chairman. Please, sir. I have to hurry up. You see the infection of the dandruff here. This is the severe case of malaria. Next one. So it is the internal cause, and this is the external causes for this. The people, they are using the shampoo, they are using the hair dryer, etc., and poor hygiene. These are the important things for which we should take the precautions. Next one. So ladies and the sick people, they should take a lot of precaution for this. Next. Then management of the majority of the available synthetic antifungals due to the fungistatic nature are not able to control the diseases. In other words, recurrence is very common in such cases. Next one. So we have developed this skin guard, nail guard, skin pro, and pityriasis versicolor, this semua, which is caused by this malaria. Next one. So in silico approach for prediction of lesion binding, the bioinformatical tool we have also used for this. Next one. We will escape it. Next one. Next. Next. Pass it on. Next one. So this is how we have the lesion binding protein. Next one. We got a thymol from the Trachyspermae, which is the active molecule there. Next one. Next. Next. Pass it on. frequently pass kariye. Yes, these are the mechanisms of action, extraction of DNA from the fungal pest as well as the oligonucleotide design. Next one. We can discuss if somebody will be there interested in that. This is how that DNA band can be noted, amplified DNA band of Aspergillus fiber treated with the active constituent at their respective MIC. Next one. Next. Next. आगे बढ़े. There is no record of any earlier report of such type of bioefficacy of the plant constituent against stored grain, fungal pests as well as insect pests. Hence, this study represents this. It is because of the fungicidal efficacy, insecticidal efficacy, broad pesticidal spectrum, long shelf life, non-phytotoxic. So these are having an edge over the synthetics. Next one. Next. Next. Next इसको आगे बढ़िए. So one can see here. जब पीछे चलिए जाए. Next पीछे पीछे. You see the IPR issues. It is mentioned here. So everybody must have done the PT. You must remember only these things. PT, GC, then NIIT. If you will be remembering this, PT, GC, and NIIT, it means you are well versed in the patenting, IPR. P for patent, T for trademark, G for geographical indication, C for copyright, and for neighboring right, integrated circuit, industrial design, and trade secret. So it is for the youngsters how this is how I got them to remember these things. Next, आगे बढ़े. So you can see the organizing secretary here, Sushil Shahi. He was the student of mine at the time. Amritesh Chandra Shukla, myself, and Dr. A.K. Bajaj from Medical College, dermatologist, and Sushil Kumar, the then director of CMAB, and one chemist was there in is the first patent of University of Allahabad in 2001. We got it, UK, Japan, and his patent. We have developed the formulation for the dermatophytes. Next one. So that Siddharth Datta, he published this paper with this on Milezetia. Next one. Next. So I'm telling you, for the youngsters, when this patent, its message came to directly Vice Chancellor, and then Vice Chancellor wrote to me this note, congratulations, Dr. Dixit, please meet me. When I met, he gave this certificate to me, and when I showed it to my next one, to my students, next one, everybody will be in a jubilant state, so they can work more hard to get more patent in future. So in our lab, the people, they are working even at late night also. There is no time-bound program. 9 to 5 or 10 to 4 is not there. Next one. Next. This is another. We have conducted this. We are training the farmers also in that way. Next. Next. We are having this. This is Rohit Mishra. He's. I'll be telling later on. Yeah. Next. This is our lab where we are getting the PCR, real-time PCR and gel documentation. So now I am retired. I have so far produced only 33 students. And Dr. M.C. Pandey was my first student. And presently he is financial controller in Dr. Ram Manohar Lohia Hospital, Lucknow. Then Amritesh Shukla, he is currently in Lucknow University as professor. Dr. S.K. Shai. He was heading the department here, and everybody knows because of his active involvement, we all are here. And
Sir, kindly, please.
Then Dr. Ramesh Singh, he is in Sardar Patel University, Meerut. Then Dr. R.C. Mishra, he is Vice Chancellor now in Mahakaushal University. Then Dr. K.P. Pandey, he is the principal in one of the college. Siddharth Datta is in Pune in R&D firm. Then she is there. Mamta khadi hai yahan pe aap. हैं? You can इधर आ जाओ. हैं? हैं? So she is Mamta Patra, now Mrs. Chai. So she worked on onychomycosis और बड़ी मेहनत से काम किया था, बहुत अच्छी थी इसकी हुई.
Professor Dixit, please.
Yes, I'm but only 1 minute. So, Dr. Rohit Mishra is Associate Professor there in Center for Science and Society. Next. So, he is in Bundelkhand University. He is in this Mizoram. And she was a DST scientist. She is retired now, Rita Gupta. And likewise, he is also assistant professor in Allahabad. So this is the team. Next one. So the two students, they are Piyush Srivastav and Saket Jha, they are in University of Illinois now. They are working on cancer. And Mohi and Rohit Shukla, they are sitting there for their presentation. And she worked on termite and she worked on biofertilizer. So next one. So eco-friendly. Thank you very much for patient hearing, and special thanks to Professor Atri sir for chairman for allowing me to complete this, and to all the audience here who, because I have received so many things. If anybody will be interested, I'll be talking, discussing later on. But those who have, if they wish to Allahabad, you can write me. I will be a very good host for you people. There will not be any problem. You can have it only deep in living.
Thank you, sir. Thank you very much. Dr. Pradeep, please.
Yes, sir. Thank you, Dixit sir, for the extensive work on the presented on this platform, and it will definitely beneficial to all participants as well as especially to those younger mycologists. So thank you for your wonderful presentation, sir.
Thank you. Thank you very much. Now, straightway we move on to the third lecture by Dr. M. Clayselvam, Faculty of Marine Sciences, Annamalai University. He'll be speaking on marine fungi for the future prospects. Is Dr. Clayselvam there?
Yes, sir.
Kindly, sir, bear with us and restrict to the time.
Definitely, sir.
Please.
Thank you. Okay.
Yes, sir. You can straightway share your slides and start your talk.
Yes, sir. Slides are sharing, sir. So, a pleasant good afternoon to all of you. Respected chair and co-chair. Distinguished mycologists and my dear fellow participants and student friends. Really, I feel very happy to share my research experience with you people. So all of you, I know, very hungry. So without, just within 15 to 17 minutes, I finish my presentations. All of, to the world,
to the world, India is beautiful. To the world, India is beautiful. In India, Chhattisgarh is beautiful. In Chhattisgarh, Bilaspur is beautiful. In Bilaspur, Department of Botany and Biotechnology is beautiful. In the Department of Biotechnology, and the students of Department of Botany is always beautiful. And we, the participants of the National Conference on Fungal Diversity and Bioprospecting, participants all are beautiful. Everybody beautiful. Also, I'm also very beautiful. Go to the next slide. Next slide. So the marine environment is beautiful. So in the marine environment, there are so many ecosystems. We are having the marine environment, that is coral reef ecosystem, marine ecosystem, mangrove ecosystem, coastal ecosystem, salt pan ecosystem. There are so many ecosystems. They are all very beautiful. They are all not only beautiful, they are all resourceful. Resourceful in the sense, there are two important resources. One is living resources, other one is non-living resources. The living resources are, which includes starting from microbes to mammals, are living resources, whereas the non-living resources we have plenty. There are so many non-living resources. So the living resources, they breathe, they eat, they grow, and they multiply by way of reproduction, and they have all the senses. Whereas the non-living resources, they do not have anything. But the living resources are renewable, whereas non-living resources are non-renewable. So these resources, the living and non-living resources should be sustainable for future generation. So here nowadays, the sustainability of the resources is very big question mark. Next slide, please. So here, our environment, nature is.
Sir, your slides are not shared.
Kindly share the slides.
Oh,
Kindly share your slides, please.
Yes, check it. It's shared already, no? This one should be already shared, right?
Now it's visible, sir.
It's okay? Right.
Yes, okay, sir.
Shall I?
Yes, please, sir.
Yes, sir. Thank you. So our environment is just like our mother, whereas the development activity is just like your wife. So which one is more important? Whether mother is important or wife is important? Both are important. So the mother nature we have to conserve. So we are disturbing the mother nature for the development activities. But due to the development activities, we are especially the exploitation as well as other things. We are destroying our environment. So we have to preserve our mother nature with. The mother nature is to be cared with love, whereas the wife, that is the development activities, is to be loved with the care. Next slide, please. Next slide. Close it and next. You close this one. So I'm closing. Oh. What happened? Oh, every time some issues there, right? Next.
Slides are not moving, sir.
Yes, I don't know why. Yes, sir. I don't know why it is so. Just allow them. Allow all the three.
If you can see your slides there, you can proceed further. Extempore, no problem.
Yes, sir. Yes, sir. Just to use the cursor. I mean, that is forward, revert this one. This one, you see. Oh, there. Oh, what happened? Better English. Stop it, Richard. Sorry, sir. So some technical issue, please. Hold for 2 minutes. Like this. Yes.
Yeah, it's okay. Now you can make it full screen.
Punching for future prospects.
Or you can straightway start. Yes, okay.
Yes, sir. Yes, sir. Next slide. Next slide. Yes. As I mentioned, so the marine environment, we have so many ecosystems. Just press one by one. So this is lagoon ecosystem. Next one. Next slide.
Sir,
This
again,
one. Coral reef. Next one. You just keep on press. This is a mangrove ecosystem and the estuarine ecosystem, coastal ecosystem, and the salt pan ecosystem. So these are all normal ecosystem in the marine environment. So all very beautiful. Next one. So apart from this, we have extreme environment like hydrothermal vent, glacier, thermal spring, as well as salt trench. So these are the different ecosystem in marine environment. Hello, please. Next slide, please. Next slide. Next slide. So in the marine ecosystem, to estimate the microbial load in the marine ecosystem, a team of scientists from under the leadership of Dr. Frederick Graceland is the chairman for that Global Census Committee chairman, and nearly 500 scientists from 70 countries participated in this cruise. So they made a 10-year survey to estimate the microbial power, especially the diversity of diversity in marine ecosystem. One girl, like Dr. D. Chandramohan is our alumnus, is the only Indian participated in this COL program. Next slide, please. And another team of researchers, they made a survey, a team of researchers from the University of Georgia. They estimated the realistic estimate of microbes on the earth. They estimated that more than 5 million trillion and trillion of microbes in the marine ecosystem. That means you can add 30 zero in the after 5. That means if you be comparing the African elephant, the biomass of microbes, more than 240 million ton elephant of the African elephant, that was biomass we are having in the marine ecosystem. Next slide, please. So there are more than 90. So next slide, continuously just proceed. So just proceed. So here, since from Anton von Leeuwenhoek, that is father of microbiology, then modern microbiology, then Robert Hooke, and he is Dr. Chandramohan is our alumnus. He is the father of marine microbiology, and he is my teacher. Dr. A. Purushothaman is our teacher. He's only taught me the basics of microbiology in my career. And he is our alumnus again, Dr. Kasturi Venkateshan, he did MSc in 74, 76 in anomaly in marine science, and after that he completed PhD. Then he moved to PDF for Japan. He worked there for more than 10 years. Right now, he's working as a senior principal scientist in NASA in USA. And what they did, they collected sample from space. From the space sample, they isolated many bacteria and fungi, and he wanted to name it 2 bacteria. For that, he wrote a letter to President of India, got the permission. After getting permission, he given the name to that bacteria, that is Bacillus Abdul Kalami. Such a distinguished alumnus we are having. He also developed another, why I'm saying all those things, since from Anton von Leeuwenhoek till today, more than n number of scientists working the diversity of marine microorganisms. Excel, please. But the scenario is like this. Courtesy to Dr. Professor Chandra Manoharachari, only 1% of microbes so far described. When we take the fungus, only 0.8% of fungus are described or culturable. Next slide, please. So it is for terrestrial. With respect to marine ecosystem, only 0.1% of microbes so far described and identified. So remaining 99.9% to be identified. That means the reason is, next one, please. The reason is our understanding on the marine macroeconomic diversity is very not clear. There are so many questions we are having. The first question is, so what are the microbes present in the ocean environment? We do not know, and what they are doing in the marine ecosystem, there is no clear idea, and how they interact with all the higher organisms, we do not have answer for the questions, and how do the microbes affect other marine farms? So these are so many questions we are having. We do not have answer for all those questions. Next slide, please. So the reason is why 99.9% of microbes are not described. The reason is, the first reason is we do not have adequate monograph or manual for the identification of especially microbes. Next one. And the another interesting reason is one of our senior national professor, Dr. T.J. Pandian, used to mention that taxonomists are endangered because nowadays, especially youngsters, they are not willing to come for taxonomy studies. They are directly going for molecular taxonomy and other things. But we are very fortunate. We do not have basic taxonomy, especially our the distinguished, just like our Lakhanpal and other people, Manoharachari and Bhagiraj, they are all distinguished taxonomists. Now the taxonomists are endangered. I think my humble request to the young budding scientists, please take taxonomy work in your basic studies, especially in MSc higher studies. And the other interesting reason is most of the microorganisms are dormant because their dormant nature, they do not grow because whenever the suitable conditions are there, they start grow and multiply. So these are the main reasons for we are unable to culture the unculturables. Next slide, please. So for that, we are having biggest challenge: how to culture the unculturables. Even then we are having sophisticated instruments like a confocal microscope, flow cytometer. We are unable to culture the unculturable. Next slide, please. And we have the answer for how to culture the unculturable by way of studying the genomic studies. So genomics is one of only way to how to culture the unculturable organisms. Next slide, please. Apart from that, we have the molecular taxonomy. We are having so many molecular markers. By using the markers RAPD, RFLP, we have so many markers. By using these markers, we can culture the uncultured organisms by way of rDNA for bacteria, ITS for fungi. Next slide, please. And apart from that, we have metagenomic studies to study the uncultured organisms. So metagenomics is excellent techniques to study the genes isolated directly from microbial communities without culturing in the laboratory. We have two advantages. One is through the metagenomics, we can understand the physiology and role of unculturable in the ocean. Likewise, based on the metagenomics, the gene from environmental DNA extract has helped to characterize the phylogenetics in nature. We have one limitation, that is through metagenomics, we does not give any information on physiology, biochemistry, and ecological function of uncultured microorganisms. Next slide, please. So far, more than 200 marine microbes so far sequenced. From our center, so far we have isolated 78 fungal strains from different ecosystems. Out of 78, 17 strains we have sequenced and deposited in the NCF bank. So next one. So with this fungal diversity, so what are the bioprospering aspect of fungus for human welfare? So I have to be aware of that when we see the global population. Next slide, please. So next. When we see the global population in 1970, the global population only previous only 3.6 billion, whereas in 2022 the global population is more than 7.8 billion. That means the population keep on increasing. That's why we have to focus 4 important areas of research. One is, next one please. The first focus area where we are going to do that is food security. So the demand for food security is keep on increasing because the available cultural lands keep on reducing due to industrialization as well as real estate. That's why our farmers are cultivating our paddy and vegetables, but they are facing so many losses due to crop losses, due to drought, salinity, disease problem, insect and post-crop loss. These are the reasons for we are getting this product. Next one, please. So welcome this issue. The next one. So according to World Health Organization, more than 75.6% children are anemic. Next slide, please. Next slide. Likewise, more than 36.5% of children are stunted growth. Next slide, please. Likewise, there was an estimate that more than 45% of women aged between 15 to 45 are either anemic or their body mass index below normal. So that's why we have to overcome the malnutrition problem. That's why we took a next slide. We take especially in marine environment, we have both finfish and shellfish. These finfish and shellfish have a rich source for nutrients. It has protein, carbohydrate, lipids, minerals, water, essential amino acids, everything. Apart from that, they are having rich source of PFAS, that is polyunsaturated fatty acid. So it is rich in seafood. But we are using this seafood, especially next slide. We are using the seafood waste, especially the fish waste, shell waste, seaweed, seagrass. So these are the four important marine waste we have utilized for the cultivation of mushroom as a substrate. So these are the innovation technology we developed under the projects of the University Grant Commission as well as DST, NFDP, and the Ministry of Human Resource Development. They funded projects for developing the cultivation of mushroom using the marine waste. Next slide, please. So for the cultivation of mushroom, we used four different substrates like seaweed, seagrass, fish waste, and cell waste. Please click. So these are the four different substrates we used for the cultivation of mushroom, and these are the standard methodology we followed for the cultivation of mushroom. Next one. So we successfully cultivated the mushroom by using the marine waste as a substrate along with the paddy straw. So this is the success story of cultivation of mushroom in our department by using the marine waste as a supplement substrate. Next one. So after developing the technology, so we estimated the approximate composition of mushroom cultivated in control as well as negative control, positive control. We estimated moisture, next week, moisture, and we estimated fiber, then vitamin, then protein, then carbohydrate, lipids, and amino acids. So we compared the mushroom cultivated using the marine substrate as well as without the marine substrate. So we found that especially the mushroom cultivated by using the fish waste got an excellent source of all the essential nutrients as well as especially all the essential amino acids present in the mushroom cultivated by using the marine substrate. Next slide, please. So with this, we started. So the cultivation of mushroom is strictly. It's a very easy maintenance and the minimum area is required. Next, we have a meager investment for the cultivation of mushroom, then less manpower, the quick return. Especially mushroom is nutritious, has a good market and highly profitable. Based on this criteria, we approached the government of India, got the projects. Next one. So we transfer the technology to the fishermen community as well as self-help group in and around Karur district. We successfully more than 750 NGOs got trained in this mushroom cultivation using marine substrate. Right now, 9 different NGO people, they have started their own business in the mushroom cultivation as well as they are running their mushroom shop. Especially they are preparing biryani, mushroom pagoda, mushroom soup like that. They are getting more than 4,000 rupees extra income from this training. So next, this is the first achievement we made. For this only in the 2014 MSI, I got the first oral president award. I thank Mr. Dr. Mulrich Kaur as the chairman as well as Dr. Yashwant Sharma is the co-chair. Thank you. Next one. Next slide, please. So next important aspect for the growing population, we are having second important area we have to focus that is biodiesel production. So biodiesel, all of you aware of the next slide, please. So biodiesel is in need of our. The reason is we are exploiting one of the non-living resources. Especially we are getting the petroleum product from the fossil fuel region, but the fossil fuel is exhausted due to the over exploitation. And apart from that, it will provide economic energy security as well as economics, and the prices keep on increasing for the fossil price, especially petroleum products. And most importantly, if we are using the fossil fuel petroleum product, it will lead to production of high amount of carbon dioxide level. It leads to the global warming, again climate change. For that reason, we have to find out alternative diesel from the bioresource. Next slide, please. So here, the biodiesel having so many positive aspects. I'm to the time constraint and pass on to the next slide. So what we did through the process, there is a process called the trans-acidification process. It is nothing but it's a chemical reaction for the conversion of triglyceride into biodiesel as well as alcohol. So this is a mechanism by which we started the production of biodiesel by using the marine fungi. Next one, please. And next slide. And these are the mechanisms, these are the biochemical reactions next week. So these are the, actually we have the triglyceride is the compound. It was converted into biodiesel as well as glycerol but through the chemical reaction by using the sodium hydroxide as well as methanol. Next slide, please. So next slide. So these are so we isolated mangrove associated rhizosphere sediments. From that, we isolated the alluginous fungi, and the actually 28 fungi we have isolated from the mangrove forest, mangrove environment, and one such fungus that is Rhododendron muscigerosum having exhibited high lipid content in the mass culture. So we got the mass culture of particular Mycological, and the study was we optimized the mass culture, and we by the process of transcription process we we produce the biodiesel by using these oleaginous fungi, and it is very cost effective when compared with the other ingredients used for production of biodiesel. So to conclude that project, it is very feasible and effective method for the production of good quality these biodiesel from each single cell oil could be a competitive and conventional diesel for their fuel in future. Next slide, please. So this is the second aspect. Third important aspect for the growing population is the production of drugs and chemicals from the marine sources. Next slide, please. So nowadays, especially we are having so many drugs and chemicals we are facing, especially the growing population facing so many diseases like cancer, arthritis, heart attack. For that, we have developed so many drugs and chemicals. Other purpose, our center. Next slide. So we focus. These are the area we have developed. We have produced both primary metabolites as well as secondary metabolites from different sources of marine fungi isolated from different ecosystem. From as I mentioned last. Next slide. So these are the area we work. So these are the untapped area. We are working only in the normal environment, but these are the extreme environment. From the I see all of you aware of that the hydrothermal vent. This is called the hydrothermal vent. The other is called Bilaspur. From that only the bacteria Thermus aquaticus was isolated. And more than 11,000 meter depth. More than 11,000 meter depth. The temperature is more than 350 degree temperature. So in this 3 degree temperature, certain organisms are living. So from that, especially the Jane worm in the hydrothermal vent, they isolated one bacteria called Thermus aquaticus. From that only, we are having that fat polymerase enzyme. It was very much useful in the PCR reactions. So likewise, we have so many extreme environment. If you are focusing in this environment, definitely we'll come out with very good drugs and chemicals for the future generation. Next slide, please. And the last one, the last important area for the global population, that is called next slide, please. Next please. So in the marine ecosystem, more than 97% of salt water, 97.5% salt water, we are having 2.5% only fresh water. So these are the 2.5% fresh water we are using for all our domestic as well as industrial purpose. But report says, especially the recent report says, the scarcity of water is major threat to our growing population. Next slide, please. So this next slide. So this is the issue communication from the Times of India paper. So for that, according to Sundar Bahuguna, he mentioned that already we are facing two different world wars. But in future, if you are going to face third world war, it is only because of water. Because water is a major threat to the growing population. So they are, unlike the other world war, this world war has two sides. It will be now several inter-country and intra-country. So water is life. So that third and the ultimate war of humanity will start over side of the life, which means water. So that's my next place. For that reason, we have to, our center, we are concentrating on we are doing the mass scale tree plantation by using the NSS volunteers as well as social people. So the especially mangrove is one of the best source for how to maintain the global to get rid of the climate change as well as mitigation of the global warming. So what actually the mangrove is the superstar as compared to terrestrial forest. Why? Because of it absorbs high quantity of atmospheric carbon dioxide, and also it has, especially in the mangrove ecosystem, we have sediment soil. In the sediment soil, the carbon storage is millions of years we are having. So another important reason. And also more than 93 million tons of carbon dioxide removed daily by mangrove forests. For that reason, we are planting the mangrove forests almost all the east coast of India. So we are especially, I think recently the G20 in Bali, Indonesia, all the world leaders, they are giving awareness to the people. We have to protect the environment by way of by way of plantations. So mangrove is one of the best source to protect our environment, especially coastal. It acts as a bioshield. It acts as a bioshield in our coast. Actually, our department situated in the coastal village, very coastal village. From our department, just within 300 to 400 feet, our Bay of Bengal is there. So during the 2004 tsunami, more than 200 people died in the coastal area. But we have artificially developed mangrove in our department, and it will protect the more than 1,000 people. So the mangrove is acting as a bioshield. I request all the people, especially young minds, please try to do mass tree plantation to get water. Next important solution is developing technology for desalination process. So desalination is nothing but removal of salinity from the water. Very simple technique, but it's very costly. So my humble submission to the audience: if you are developing certain technology, how to convert the saline water into potable water, definitely it is very good. Very good. Contribution to the science. So contribution solution for the scarcity of water. Next one. So with this research, social, previous. So research is not an ivory tower, but the power of help to poor people and poor knowledge. So research should not end with appreciation, but it should be with application to the society, community, and humanity. Thank you. Next one, please. I thank my university, Anambra University, one of the largest university in Tamil Nadu, second largest next to Madras University, the second largest university in India. We are having more than 10 different faculties and 54 departments and more than 33,000 students doing this. Is a residential university. More than 33,000 students doing their master degree in our university right now, and more than 3 lakh students they are doing degrees in online distance mode. And I mentioned one important things with you. Our alumnus is become a president of India. Alumnus become a president of India, and one alumnus become the governor of Tamil Nadu, and many Padma Bhushan from our university. Thank you, and I have thanked the MSI for giving this wonderful opportunity to share my experience with you people, and especially my special thanks to Department of Botany, especially Sushil Kumar and his team, and especially MSI, Professor Akbar Ahmed, for President of MSI, Secretary Dr. Raman, and my friend Dr. Maldivanan and Malarvi and Avnit Pal. Thank you, one and all. Thank you for your listening.
Thank you very much, Dr.
Thank you, sir.
For
Thank you, sir.
Highly informative lecture.
Thank you, sir.
You have presented a lot of information and innovation using marine resources,
yes, sir.
Especially linked to the society, socially relevant research. You gave a lesson in your presentation. I'll request Dr. Pradeep to please.
Thank you, sir. Thank you.
Thank you, Kalim sir, for your nice and informative talk on how best we can use the marine resources to solve our human crisis, especially in the food, medicine, malnutrition, biofuels. So many aspects you have touched, and it's wonderful talk. Thank you for your wonderful talk, sir.
Thank you, sir.
Thank you.
Thank you.
So with this, we complete this session. There were only 3 lead lectures, and all the 3 are completed now. And I'm extremely grateful without wasting much time, because already people are hungry and it is lunch time. We have already exceeded some time. So I'll straightway put a vote of thanks to the organizers, to MSI, and to my co-chair, Dr. Pradeep, for providing us this opportunity, and to the enlightened speakers, the lead lecture presenters, Professor N. Mathivanan, Professor Anupam Dikshit, and Professor M. Clay Selvam, for sharing their research and presenting excellent information.
Right. Thank you, sir.
Over to the organizers, please.
Thank you.
Thank you.
Organizing secretary.
Thank you. Thank you. Nice. Thank you so much.
Nice presentation.
Nice. Thank you. Thank you so much. Thank you. Thanks. Thank you. I'm not. Huh? Thank you. Thank you. Thank you. Thank you. Thank you. You can find that I have to go to the
So this session is over for lunch. And thank you all again for listening. The audience, uh, three speakers speaks in this session on different topics, and their lectures were no doubt very useful and very scientific for the study point of view as far as youngsters who will enjoy with these lectures and contact the persons if you are conducting the research on the same line. Thank you all, and I extend my hearty thanks to our chairperson and co-chairpersons who has given the valuable time and chair the session. And again, I invite you for lunch. Thank you.
So, thank you all, and now we will break for lunch. We will assemble after lunch at 3:20. 3:20, sir. Thank you. Thank you all.
If anybody has any question, please.
Yeah. If no questions, then we can move to. Yeah, if you want to ask, you can ask.
Okay, please.
Sir, Professor Dilip P. Hande. See, if you have any insights on this presentation, sir, maybe you can share with the delegates as a
Thank
co-chair.
you. Thank you, Professor Kango, for the unique work presented here. And he is basically worker in the enzymes, bioactive properties of the fungal enzymes based on the oligosaccharide referring to prebiotics and the probiotics. He has a workout on the production of fungal enzymes by using the oligosaccharide, and force were found to be enhanced growth of the prebiotics and the probiotics. So he has focused on the generation of mono oligosaccharides, and the mannans are most important, and he has used also the copramyl mannose production fungal strains using there are some different fungi like Aspergillus voraci and all these things. I hope this work for the budding scientist is very important, and it is he has also published the good quality papers on the enzymes. Thank you so much. Thank you, Naveen.
Thank you very much, Professor Hande. Now the second presentation, second lead lecture is Professor Manruchi Kaur. Manruchi Kaur is the editorial board member of Kavaka, Mycological Society of India, Department of Botany, Punjab University, Patiala. And the title of the presentation is Unearthing the Hidden Agaric Potential of Northwest India. So now with this, I request Professor Manruchi Kaur to present this one.
Yeah. हम कौन से फाइल पर देख लिए
Yeah.
Chhattisgarh.
You don't have to copy it. I'll go it from there only. Okay.
This one.
Yeah. Nothing. No, this one. One more thing. Let me do it.
MSI 2022.
No. Another thing करके है.
Madam,
Yeah.
you have to copy it. Otherwise, and the
Okay, copy it. We'll delete it later. Okay. I need to wear this. It's already working.
That is working.
The pointer.
No.
You have a pointer?
No, ma'am. Sir.
I need a pointer. Share it. Share it.
Asked one time, but it is showing or not.
Oh, yeah, but put it on a thumbnail first here. So, a very good afternoon to all present. On the onset, am I visible and audible to the online audience?
Yes, yes, ma'am.
Okay, thank you so much. So, a very pleasant good afternoon to all present in this hall and the scientists who are there on an online mode. So, this side is Dr. Manruchi. I come from a university that is named after, that is the second university the world over to be named after a local language, that is the mother tongue of Punjab, that is Punjabi. So usually you pronounce it as Punjab University Patiala. No, it is Punjabi University Patiala, and it is named after the language Punjabi. So today I will be delivering a general lecture with due regards to the organizers of the conference and MSI. And I bow my head to the senior fraternity that is sitting in front of me and the ones who are there who have mentored me all throughout, Dr. Atri, Dr. Lakhan Pal. I bow my head to them because they introduced me to this world of mushrooms while I was growing up, and I started my research. Uh, and my talk would be rather focused on the audience that is behind this senior faculty, because I want to put something into your mind. If I can touch somewhere in your heart or your mind that you would like to take such research, because I'm a hardcore taxonomist. So I believe in biodiversity and interpreting into different kinds of genera and species. So that is my job. So agarix is a word which would represent a kind of mushrooms which are lamellate, and they grow in the wild where we are working in. So my area of research has been the northwestern part of India, because we are located in Patiala, and the Himalayas are northwestern part of the Himalayas are quite near to us, and of course the uh, plain areas, the states also are included in the research. So first of all, why do we want to go in for a research in any kind of diversity studies? So for taking up any kind of diversity studies, as I said that I would be talking on how you can use them. So first of all, it is very interesting and very important, and the crux and the base of the research, that is to identify, to know what are our bioresources. Once we know, we have collected and identified them to the correct part, then we collect along with some ethnomycological data, so that we would know that, okay, we collected at that time and we talk to the people there, and they use it in this manner, so that we can further take our studies into the domestication studies of the wild agarix. So this is where this diversity leads to domestication. Then other than that, there are this mycorrhiza species. We have been listening to this mycorrhiza study by my seniors, and this of course would be aiming at improving the plant health, especially the nursery seedlings. So my map is not visible here. So the major states in this area are the following. So these are the kind of areas that we usually visit on foot and for the collection of the material. This is usually in the state of Punjab and Haryana, that is the plain areas. Then we move to the subtropicals like, where we get tree needle pine and other mixed endospermic vegetation. And then we move to the temperate forests, which are the hub of such mushrooms, from where we collect a more number of specimens, because here the monsoon, once it touches the northern part of India, and the mushroom collection starts. So these are the various types of forests which we visit. Then we need to understand the growing type. That is to be noted down. See, beautiful fairy rings we have been talking of. This we pictured somewhere back while on excursions. Where does it move from? Where is the arrow like? Okay. So this is a beautiful fairy ring that we have been talking of, which we pictured some time back. Then these are the cespitous clusters that they come up in, usually solitary or maybe in cespitous clusters like solitary. Then on cow dung or any kind of dung. These are coprophilous mushrooms. One of my students will be presenting very soon after the talk. These are the leaf litter agarix, the marasmius genera. Then these are the lignicolous ones. And then, of course, the wood rotting or coming out from the living bark also. So like that. So for any taxonomist, we published way back, Atri et al. 2005, we published a paper which has this field key. And it has been nurtured all throughout the experience of working in the field as Dr. Atri was doing his PhD and then further going on with us. And this key, if once filled truthfully in the field with the from the fresh specimens, you are, my dear, very close to your identifications once you go into the literature. And then some of the macrochemical tests also are there, which needs to be performed. And one thing more, while you are noting down these characters, along with, you will be noting down, there is another substitute to it, that you would be noting down by interacting with the local people there, because while in the field, you would meet a lot many people, or maybe rarely so, those who collect the mushroom from the field for their, either for sale in the local market, or they just take it from the forest for their own nutritional requirements. So this key needs to be done, and there are glossaries available to understand all the terms that are there. For an amateur, this key, if it's understood, taking a glossary into consideration would be of great help. And the कहते हैं ना कि जितनी डिटेल में इसको भरोगे, जितनी शिद्दत से इसको भरोगे, उतने अच्छे से आप अपने स्पेसिमेन को पहचान पाएंगे. So now, we have been into the agarics since long. We have published a number of new reports from India, new to science species we have published, and diversity goes on. It is never ending, and it's a passion with me, and it is a passion in a lab to be in the field to collect specimens. But then there has to be an aim behind them. That aim goes on along with hand in hand. These agarics, we have been working on the identifications, publishing them, then the ectomycorrhizal agarics, then my lab has done something on edible agarics coming from the wild, not those which are already available. Then some medicinal aspect, that is nutraceutical aspects, we try to infer them. Then we are just trying to start on the magic mushrooms, but legally in India, it's not very welcome. And then on litter degradation, we recently did some work on. So these are the areas, along with diversity, you can further your studies into. So ectomycorrhizal fungi, we have agarics, we have heard about. So this is a long list of various taxa which have been found in putative ectomycorrhizal associations while on excursions in the field. So we collect the roots along with, and then those roots, they are cut into different sections, and we try to look for the fungal mycelium in the roots. And then isolate it maybe, and then go in for culture studies, and then re-inoculation of these into the tree from under which in the forest we had collected it from. The successful studies are there. And then, these inoculations would improve the survival of the seedlings when they are taken from the nursery to the forest for replantation. Otherwise, there's a lot of death rate in those, survival rate is less. Those which are inoculated, Dr. Atri's student did this study with Shoria ravasta and a Rasula species. And it was a very happy thesis when we saw the results. There were a lot of survival in the seedlings, other than other results, but this was the crux of that thesis and a success. So these are the various species which we have collected, and in putative ACM association. And dear students, this is how you work out a species. Once you fill up the field key, taking these characters into consideration, then you go for freehand section cutting. Just in a cotton blue or a Congo red, you stain them. Then you make these drawings. These are the final drawings which have been traced from the rough drawings which were done. Actually, the original drawings are different. Then they are traced onto a scholar sheet. Then an inking is done. This is a very interesting kind of work, nothing that is laborious. And this is the ectomycorrhiza which has been inferred. And these are the internal microscopic details. They obviously match with the kind of characters that we have drawn here. So this is Cottonarius in association with Cedrus deodara. Then we have Pinus wallichiana with Tricoloma paridium. Then we have, of course, these associations I'm talking of after the identification part, which we did. So this is Sister Lipiota Yersinia with Cedrus deodara. Then in Pinus wallichiana, we have Lectarius rubri lacteus. So this way, Amanita pantherina, it is usually found in a putative ectomycorrhiza. Once, not twice, not thrice, number of times you find it under Pinus wallichiana. Then with Quercus semicarpifolia, we found Amanita rubro-valvata. Beautiful specimens in this field. They are eye-catching. And this is another species, Amanita sesroides, which was found in ECM with the Rhododendron arboreum. And these are the beautiful stages of the development of the carpophore which have been captured. And you can see this and how close proximity it was going in with the tree. And later on, the roots depicted similar kind of characteristics as were seen in the carpophore. So this is Amanita candida, which is a species which has already been published. I should have done it the other way around in ECM with Quercus semicarpifolia. See, whenever you are publishing a species, usually the journals they do not take it. Up to they do not take it without molecular, but at that time they used to say it's based on one single character, on one single solitary carpophore, we will not publish it. So we waited for 5 long years to get another specimen from another state of a similar kind, and then we published it from both the places. So this is another one which is in association. So this list goes on. Amanita fritillaria with Roxburghi, Pinus Roxburghi, and then Benziniana with Roxburghi. So then the second aspect starts. That is unearthing the wild agaric potential in the field of nutrition. That is from the wild what we can get, and let us reduce the pressure of the collection that the local people do. Let us give them back some technology to culture them on some kind of agro waste that is already present in that area, so that we interact at different points with the various. This is the data which was being collected all throughout. This is my PhD, right? And that's my father, late Professor S.S. Sani. He used to accompany us, me and Dr. Atri standing here. So then these are my students interacting differently at different times, and that's me again, a lot of transition. So these are the list of edible taxa we have actually collected from the field, and a number of them are listed here. Anybody wants to know, I can share the list. This is one of them. Again, identifying, it is very important to identify these taxa to the correctest of the possible pinpointed identification, because any misidentification would lead to be fatal also. Because to distinguish between these edible and non-edible ones is a tough job. It comes with experience, and even sometimes if it is an amanita to be handled, I would rather tell my students not to even taste it on the tip of the tongue. Why? Because you never know what is there in it. And I can share an incident while we were working in Pathankot. Dr. Amandeep, she was here just now, she's somewhere else. She was doing her PhD at that time, and 3 or 4 of my research scholars were with me doing the collections. We brought the collections to our place, and then, we were working out, tasting them, writing, filling up the field keys. I'm just trying to share this because these things do happen. And suddenly, my students are not very, I go with them, we are friendly, but they have a little distance to carry, and that should be maintained. Suddenly 2 of the girls, they started laughing for half an hour or so. They kept on doing that, and that was quite difficult for me to digest. Then I interpreted it in a way that we were working with Peniolus at that time, a genus, and maybe they tasted it and something worked with their mind, and for half an hour they had that trip, they had that trip of the drug. So, but luckily they sustained that, but it could have been anything other than that. So do not taste anything which you do not know of. So these are the various, this Agaricus blazei is another one, which is, pharmaceutically quite being in use and is being worked on. Then Bisporus, Campestris, Scamulosis. Flocularia is one which we described for the first time from India, this species, and from Kashmir Himalayas. When I saw this specimen for the first time, I was lost. I could not make out as to what genus it would be. Later on, we did that. Then these are various species. Then, yeah, Volvirella. Volvirella beckeri, Bombaycina. Then another variety we proposed and published, Diplasia, Arestia. Then another genus was separated from Volvirella as Volvopluteus. And these are the various edible ones, Volvopluteus. Then Chlorophyllum. Then Lacaria bicolor. Then Satrella also, Candoliana is edible. Neolentinus, then Lentinus. This is one species, Standistis, which has been published by now. It's an edible one because how it is edible? Because from literature we did not find anything, but the local people, they used to collect this and take them home. So Lentinus scarosulus, it has been well under cultivation, and a thesis on this is there in our lab. Dr. Atri mentored that. Then Hemimorpha. This is one, Amanita we picked up from Narkanda. While moving from the higher roads, while looking down into the ditches of the mountains, we saw ऐसा लगता था जैसे किसी ने बड़ी बड़ी रोटियां बना के फेंक रखी हैं. So that once we walked up to them, it's an amanita. So later on it was deciphered, and they come up gregariously on the slopes. So Vaginata, इसके lookalikes बहुत होते हैं. Vaginata को पहचानना थोड़ा मुश्किल होता है. Benziniana, see how beautiful it is. Very tempting. Chambiangiana. Then Pluteus, Pedastatus. We started work on this Pluteus Pedastatus, but somehow in the process of work, we lost the culture. We're looking for another one. Constricta, then Paniolus. Anything that blues out is dangerous to taste. And Paniolus is one such genus which gives a lot of bluing reactions once it is handled or so. Then this is Pluteus. Then, after knowing so many edible genera from our general fungal forays and the domestic studies, we thought that let us go in for some domestication studies. So we started raising the pure cultures of the edible taxa. At that time, we would know that it is edible from the locals, but then after identifying and going into the literature, we would be more further sure that these are edible. But then at that time, in the field, we used to raise their cultures, the pure cultures. Yes, sir. So this is how the domestication studies, they started. This is how we got the food, but all the physico-chemical parameters and the agro-waste trials, they are being done. And the success story has been there with Lentinus stagninus. Macrocybia giantea we are still working with, but we have got the fruit bodies in the test tubes, as you can see, and we have got the test tubes even on the agro-waste with various combinations. So the nutraceutical potential, as I said, we have collected a number of them. Blazei is another one. Macrolepiota, luteus. So the magic mushroom, the work has just started. We have a number of collections of these magic mushrooms on us, and we can vouch upon that. We can identify anyone brings it to us. But the compounds, if they need to be worked out, I am in talk with my pharmaceutical department, but still nobody's really agreeing to be with it because it is not illegal. Otherwise, it is an LSD drug, which is very legalized in Canada and other countries. So Gymnophilus is another. These are all psilocybin creating genera and the species. So we started work on later decomposing agarics. Most of them were belonging to Marasmius. Other species were also there. So these are the various Entolomas, marasmias. I have to hurry through them. So we did a comparative analysis of the leaf litter just to know as to what the actual thing happens with the litter, how they decompose it. Although it was a qualitative analysis of the cellulose and lignin degrading potential was carried out, the experiments. So with that, these are the various aspects which we can touch, and I can go on with the litter agarics also as to how the nitrogen carbon nitrogen ratio would decrease or increase with the occurrence of the fruit body, and not the fruit body being there, only mycelium being there, and the mycelial mat where no mycelial mat was there. So these comparative studies were very encouraging, and we are further planning to endeavor into these. And I thank my lab on the onset, my father, Professor S.S. Saini, and then Professor N.S. Satri, my mentor, and all my lab mates, my PhD students, without whose hard work, because I can only formulate a research problem, but with them, they actually take it into the practical part of the things. And here I am presenting their work. Thank you so much. So any questions?
Thank you very much, madam. Your presentation is very good, and you have given the field key for the identification of mushrooms, and you also listed the full diversity of the mushrooms that you have done throughout your career, and you also shown that edible, medicinal, nutraceutical utility of the mushrooms. So, if audience is having any questions, maybe you can ask one or two questions.
I would like to answer that.
Madam, is there any single characteristics to differentiate edible and non-edible?
Not really, sir. But as a layman, what you can do is that avoid anything that is brightly colored, has a hanging skirt below, and a vulva below.
Because in Chhattisgarh, we get so many varieties of this one. They call it as puttu. Once rainy season comes, then you can see in the number of mushrooms in the market. And we are afraid. We are from south. We are afraid by seeing that one different color will be there, dark color will be there.
And anything that you pick up and it changes its color, avoid that.
Thank you very much, madam. And Professor Dilip, if you have any observations in the lecture, maybe you can. Yes.
Thank you, Professor Manuruchi Kaur, ma'am, for the unique presentation of classical taxonomy, which is very important today now. We have described the hidden agaric potential of the northeast India, and this is an excellent work of agaric systematics and the unique taxonomic work for the Himalayan region. There are several parameters we have presented here, like the growth types, field. Key for the identification is the very important task for the new researcher. They don't now have the how to measure the microscope and all these things. So the presentation, which is having the agarics and the edible medicinal magic type of the mushrooms and the ectomycorrhizal type of the mushrooms, and you have meticulously studied like the various type of the camera lucida drawings and the micrometry which is carried out, which is very important now. You have also presented here the species of Amanita, the taxonomy of the Amanita species and the agaric species. There are so number of the genera like Floceraria, Holularia, Chlorophyllum, Lentinus, Pleurotus, and the another type of the mushrooms we have described here. This is a very outstanding type of the classical taxonomy work. Thank you so much, ma'am, for your presentation. Thank you. I think next presenter is Avneet Pal Singh.
Yes, sir, Avneet Pal Singh. Yeah, the title of the presentation is Diversity and Evaluation of the Period Boroid Fungi from the Northwest India. Sir, please. Yeah.
Thank you very much, sir.
So you know the timing, sir?
Yeah, sir. Yeah, I'll finish in that. I'll finish in 8 minutes. No, you need to finish. Yeah, it's here. Just Which one? Yeah, MSI 2022. Yeah. Just yeah. Yeah, the second. Okay. 5 minutes. Okay, I'll finish. No issues. Which ones are yeah, yeah. So at the outset, I am extremely thankful to the honorable organizers and MSI for this chance to present the work what we are doing at our place at Patiala. Professor Manduchi has just shared the account of one of the groups of higher fungi, the agarics, which are edible, and of course we have so many applications. There is another group of fungi which are belonging to the higher Basidiomycota, and they are basically associated with the wood, and they have that excellent potential of decaying lignin and cellulose, and they have been named as the wood decaying fungi. And in them, we have got a unique character. They have large size fructifications, and on that those fructifications we have the presence of these very minute openings, the pore-like openings, and after which they are called as polypores or deep poroid fungi. And they are a very, very diverse group which has been placed in 17 families within phylum Basidiomycota. So this talk is based on the work that we could do in the last almost 12, 13 years from different parts of Himachal, Punjab, Uttarakhand, and some parts of Jammu and Kashmir. And in this region, we get that complete range of variation as far as the range of host diversity and the altitudinal variation. So from that subtropical to the subalpine, we have the entire range, so that chance is available to have that complete diversity range. So from those beautiful these coniferous and mixed coniferous forests to the scrub vegetation of Punjab, we could collect the samples with very simple technique from their wooden substrate. And this is how these fungi, they are very specialized as far as their ecology is concerned. They prefer to grow on the shaded parts of the wooden substrate. Now, parts of northwestern India, and till now from our lab we have described 190 taxa, among which 16 they are new, and many of them they have been recorded for the first time from India. Just to give you a glimpse what we do, we started when I was a PhD student. I started with my work on a corticoid fungi which do not form these pores, and when I got that chance to be associated with the university as a faculty, so then it attracted us to explore another group of wood decayers. They are the poroid fungi. So initially we started our descriptions based on the morphological characters, and based on those morphological characters, we could come across some of the diversity and some of the specimens which could not be related with the earlier published, and we proposed them as the new species which were described from time to time. That Chinese traditional medicinal system, you come across two very important groups. One is Ganoderma, second is Phalinus. They are the two genera which have been exploited by the Chinese medicinal system for their medicinal and so many other properties. So in this, we have that unique brown colored surface of the fructification, which is one character to indicate you that your specimen could be placed in the family Hymenocataceae. And when you examine them in detail, you can come across some unique features, and anytime you may come across a new taxon. So with this, we could describe a large number of good number of new taxa within this polyperacy. Since these groups, these fungi, they are associated with the host, there was an interest to explore what kind of interaction do they have with their host, what is the kind of level of penetration of their hyphae, and how do they associate themselves with their wooden substrate. So that also gave us a new dimension to work with, and we could see how the association is there through those section cutting techniques. And in some of the cases, we could very clearly trace the course of penetration of the fungal hyphae. Then we explored this family Ganodermataceae, and this is a kind of proud movement that probably in the country we have got the best collection as far as the genus Ganoderma is concerned. One of our theses exclusively that has described 39 species of Ganoderma based on morphological characters. So this is a genus which can be distinguished in two broader groups, one with shining basidiocarps, other with the dull basidiocarp, and here the subgenera they do separate. So right from the collection in the field, you have that mindset clear in which direction you are going to apply your taxonomic key for their identification. You can supplement your work with the help of those microscopic techniques. You can get that idea how the spores they are looking like, and that really helps you to place your specimen in a particular species. So based on this, we could come across that huge diversity as far as this genus Ganoderma is concerned. That thing that goes on. This is one collection which we collected somewhere in Chandigarh, and it was growing on a dead, decaying this tree. And initially we described, we could not place it. We worked out its details, traced the course of fungal hyphae. Here you can see the hyphal penetration, how they penetrate, and that gave us a beautiful publication in that that very reputed journal, Mycotaxon from Mycology. This, as Dr. Manruchi has highlighted, initially you could publish only on the basis of your morphological characters. Then eventually we came across that intervention from molecular phylogenetic studies. So you have to change your course. You have to supplement your studies with that input also. So we could collect one specimen, very minute specimen from Kashmir area, and we examined its morphological characters. We went for multi-gene phylogenetic studies, and this was again giving us that beautiful publication which recently appeared in Nova Hedvigia in 2022. So this proved that your morphological identification, if you have correctly done, it will 100% be supported by the molecular data also, if you are firm with that. So this was the first part where we explored the diversity, and then this group gave us another chance to evaluate for their fructification, since in literature we have a lot of reference there, different kinds of properties. We started with some proximate composition and element content analysis, initially with some 4 species of genus Ganoderma, and we came across good quantity of different kinds of metabolites, even some of the essential elements we could get it, and it was published in 2022 in one of the leading journals that has been there. So from thus, I'll conclude in 2 minutes, sir. This gave us another idea. Let us explore their activity part. So we could do some extract. We prepared some extracts, and we could notice very good antioxidant potential as far as the aqueous extract of these fructifications that is concerned. So we could get those similar kinds of results in number of these specimens based on different assays. The mass spectroscopic analysis that gave us an insight of presence of those rich flavonoid polyphenolic compounds in the fructifications of these fungi. So that was later on taken up, and we could publish it in International Journal of Medicinal Mushrooms. And then we also had very initial studies on the central nervous system CNS activity of one of these extracts of genus Ganoderma, where butanol fraction was giving very promising kind of results. And finally, recently we have further extended these studies and taken it to the level of some anti-cancer studies based on the cell lines. And in this case, again, the results, they were very good as far as the aqueous extract of some of these fungi they were studied about. We could do and perform some of the microscopic studies where the cancer cells, they were being observed at different stages of apoptosis. So those cancer cells, they were targeted by the extracts of these fungi, and we can see pre-apoptotic, apoptotic, and post-apoptotic stages. They were clearly examined, and we could also see the changes in mitochondrial membrane potential, which could be one factor for that apoptotic action. And again, based on UHPLC analysis, we again came across very good quantity of those polyphenolic compounds. So this gave us an idea that there are so many kinds of aspects which you can explore with as far as these poroid fungi they are concerned. So I started my work with my guide, Dr. Gurpal Singh Dhingra, and we were lucky to get that support from various people who are working across the globe. Professor Nils Hallenberg, Professor Lee Preverden, Professor Ewald Langer from Germany. So they were very generous enough to share resources which were available with them. So one of the proposals that was funded by Sir. I'm extremely thankful to my parent institute, the colleagues from pharmaceutical sciences, and these research scholars who really made this presentation to be possible in front of this August gathering. Thank you very much. Hope I kept the time.
Thank you very much, Dr. Avneet Pal.
Thank you, sir.
And the next presentation is Amandeep Kaur.
She's chairing a session, I suppose. Meanwhile, Vineet can.
Yeah. The next presentation is Vineet. Please, 5 minutes for the presentation. 5 minutes for that.
Sure.
No. नीचे करो MSI होगा देखो MSI 2 नहीं 2 Hi. Thank you.
Sir, am I audible, Dr.
Yeah.
Hande?
Yes, Philip.
Okay, thank you, sir. A very good afternoon to everyone, and I thank the organizers to give me time to present my work. So I'm working currently now in Israel, and I work with endophytes and how they can help in resisting the biotic and abiotic stresses in cucurbits, and I'm majorly specifying in melons right now. So, as you know, why melons? So melons is one of the most consumed fruits in the world, which is loaded with a lot of minerals and vitamins. And in Israel, it is produced towards majorly in the south part because it is more favorable condition there. Much of drier, very less precipitation, and it can go up to 14 to 47 degrees centigrades, and a very suitable condition for survival of melon fruits. So, and how melons can, how endophytes can help. So, as we know, and a lot has been talked about the endophytes, that these are the organisms that colonizes the inter or intracellular spaces. Sorry. Intracellular spaces of the host tissues without showing any symptoms of their presence. They're ubiquitous and are found all around the world. Whenever if there is a plant, there is an endophyte colonizing it. What these do, they provide various kinds of fitness, including abiotic and biotic stresses. And usually, what has been done till date is that the people have used medicinal plants for utilizing the potential as a pharmaceutical or either for plant. Now, what we are trying to do, we are not using the host plant. We are not taking the endophytes from the host plant, but we are taking it from belonging to the same family. So we are taking from wild cucurbits that are there in the desert area, and we are exploiting them to be used as the source of abiotic plants in melon plants. So our project is divided into two parts, with biotic and abiotic stress. Biotic stress, we are dealing with fusarium and charcoal rot disease, with fusarium wilt and charcoal rot disease. And for abiotic stress, we are dealing with salinity, drought, and heat and cold stress. So I'll be just talking about the salinity today. So for that, what we did, we collected plant samples. These are the wild cucurbits that are growing in wild in this tree region. This is the southern part of Israel. This is where I live. Here, this is the holy city of Jerusalem, and this is the southern part, which is around minus 600 feet from the sea level. It's completely desert there, and these cucurbits are collected from this region, and following a standard surface sterilization process, we obtain
different kinds of fungi from different parts of the host plant. This and then there was a purified. So these are the fungi that we obtained from the host plant that belong to different genus. And from this, this is the host plant. This is the distribution of endophytic fungi that are isolated from different plants. So maximum isolates were obtained from Ecubelium, followed by wild melon, and then Memordica. So when you see the holistic feature, maximum plants were colonized in the roots, followed by the stem, and then leaf and fruit tissues. So first, as everyone suggested, go for the taxonomy, and we perform classical taxonomy, and we found that there are Fusarium is the most dominant species that is there, followed by Alternaria and Cladosporium. Also, we found Acromonium and Bipolaris species. Then we did molecular identification. For molecular identification, we used first of all extracted DNA, followed by arbitrarily primed PCR, which is cutting at different points, and then ITS based sequencing to identify up to genus level. So following this protocol, we identified this is DNA. This is the DNA bands, and then we followed APPCR. So you see, these are different fungal isolates showed similar bending patterns are considered as one isolate belonging to the same group. So based on that, classified or grouped them into 14 different classes. Then we went for identification, that is ITS based, and you can see what we found in the classical taxonomy. It was repeating here also. So Fusarium, Bipolaris, Aspergilli, Mortaria, Cladosporium, Bipolaris. It was the same, just the same result based on the ITS basis. Then we wanted to check the activities, different kinds of activities that it has. So first of all, we checked for the phosphatase activity. So you can see when you grow it in a bromophonal medium, minimal bromophonal medium, it changes the color, the isolate from blue to brown, which means it is solubilizing phosphate. Then we check for hormone production. So it is producing hormone, indole acetic acid. And these 5 isolates are producing indole acetic acid hormones in the in vitro condition. Then we check for cell degrading enzymes. So it is producing cellulase, protease, and chitinase activity. And the fungal endophytes were then checked for cytophore production. And this isolate ER24, which was producing all other enzymes, was also producing cytophore activity. So based on our screening assays, we know that these are the 5 or 6 endophytes that are showing different plant growth promoting activities. Now, we wanted to check the in vivo effect of the endophytes in the plant. So first of all, we germinated the seed following this protocol, and then we treated the endophytic fungi in the roots of the melon plant, and then checked for the stress tolerance. Sorry. So you can see here. This is just the control plants. This is without any treatment. This is with treatment, and this is increasing salt treatment. So as you see, when there is 100 or 200 millimolar concentration of the salt, the treated plant or the colonized plants are in much better shape as compared to the non-colonized plants. You can clearly see here, it shows a higher plant fresh weight, dry weight, root weight, all the morphological characters and the weight are much more in comparison to the non-colonized plant. Then we did the greenhouse assays, and we found the similar characteristics, that the endophyte colonization is increasing all the morphological characters as compared to the non-colonized plants, which is much more significantly. Further, we moved to the physiological character, and we found that the relative water content in the colonized plant at 100 and 200 millimolar concentration is significantly better than the non-colonized plants. Also, the stability, the plasma membrane leakage, it is much more in the non-colonized plant as compared to the colonized plant. Followed by the electrolyte leakage, you can see the electrolyte leakage both in the roots and the soil of the non-colonized plant is much more higher at higher concentration of salt as compared to the colonized plants. Then we checked what is the effect of the endophyte colonization in melons when it comes to the photosynthesis. So you can see the transpiration and stomatal conductance both goes down as the in non-colonized plant as the salt concentration is increasing. But when you see the colonized plant, it is much more stable in comparison to the non-colonized plants, which suggests that the endophytic fungi is helping the plant to maintain its original structure so that it can perform normal photosynthesis in the plant. Then we checked what is the ion uptake. So the endophytic colonization reduces the sodium uptake and increases the potassium uptake, so that it means it can maintain the sodium potassium homeostasis in the plant so that it can perform normal functioning. But you can see here the chloride concentration is increasing in non-colonized plant as compared to the colonized plants. Then we checked for the biochemical parameters, and in colonized plant, you can see the chlorophyll content is much more higher with the increase of salt concentration in comparison to the non-colonized plants. Then we checked for the phenolic content, and you see as the salt concentration increases, there is an increase in phenolic content in the colonized plants, which shows that it is not only providing antioxidant activities through enzyme activity, but also non-enzymatic pathway. Similar with the flavonoid one, you can see clearly that it is having much more flavonoidal content in comparison to the non-colonized plant. 2 minutes. Okay, then we checked for the lipid peroxidation, and you can see the non-colonized plants here are in much more stress as comparison to the colonized plants. Also with the proline is a you can see the proline concentration is increasing, which means it is providing a supplement to the plant with the colonization. Also with the catalase activity, the peroxidase activity, and the superoxide dismutase activity, all these antioxidant activities are increasing when the endophyte is when the plant is colonized with the endophyte in comparison to the non-colonized plant. We also did some histochemical staining, and which also suggests that there is a these are non-colonized plant. These are non-colonized plant. These are colonized plants. So there is less of cross production. Also, this is the mortality. So there is less mortality, and also the lignin deposition. You can see the lignin deposition is much more in the colonized plant as compared to the non-colonized plant. Then we take the endophyte and see how it colonizes. So you can see this red mycelium with the red fluorescent dye. So it is colonizing the roots as compared, and no fluorescent signals were obtained in the non-colonized plants. Okay, then we measured how much it is growing, and with each concentration the colonization is increasing. And then we did the molecular identification using multigene sequences, and we found that it belongs to Fusarium flavum species, which is well supported by the classical taxonomy. And then we did the field trials, and we found that under the field trial also not significant, but the endophytic colonization is increasing the food grading and total suspended solids soluble in the plants. And just now we published this paper in this journal. You can find it online. It's available free online. Thanks to my group that I'm working with. That's my supervisor, Dr. Freeman, And that's our research engineer and two of my colleagues. Thank you.
Thank you very much, Dr. Vinny.
Thank you.
Thank you, sir. So Dr. Amandeep Kaur is available here. Okay.
What about schedule from 3:20?
Sir, this is of seventh session, sir.
Second session.
Seventh session.
Yeah. When it will be, I'm not aware because I was just called for this. We can,
Sir, you can ask, sir. Sir, you can ask.
सर इसमें तो आप ही देख रहे हैं ना, मैम का भी उसको भी कर दीजिएगा।
मेरा भी होगा MSI 20।
मैं करता हूँ।
Yes, sir.
और ये अनर्जिंग वाला मैम।
सर कुछ गुजरात को देखिए।
दिन में से भी कर देते हैं। ये शुरू होना। दिन में नहीं, एमटी साइकिल में कर दीजिए डायरेक्ट। हाँ, ये तो ठीक है। यूजलेस ही होगा। हाँ,
मेरे काफी चले गए होते हैं।
Thanks, buddy. Thank you so much.
मुझे पता नहीं। So the next presentation will be by Dr. Amandeep Kaur. The title of the presentation is the occurrence of Caprophilus mushrooms in Punjab. Yes.
Copy the PPT. Folder name, please. Folder name, please.
It's good. बंद कर don't sir.
नाम क्या है?
PPT छत्तीसगढ़
MSI?
yes.
Here is one. Yeah.
Madam, you have 5 minutes time for your presentation.
Yes, sir. Yes, sir. Good afternoon to everyone. The topic of my presentation is the occurrence of coprophilous mushrooms in Punjab. So coprophilous mushrooms are saprotrophic fungi which germinate and grow on dung, herbivorous dung. They are specifically adapted to dung of herbivores. And during the present study, dung localities of Punjab state were investigated. And we worked out 172 collections. And the collected material belongs to 20 genera and spreads over 7 families of order Agaricales. This slide shows the area surveyed along with year of survey. These are the various types of coprophilous habitats of Punjab from different places. We have collected various wild animals of Punjab and various herbivorous animals of Punjab. The coprophilous investigator taxa belong to 7 families of order Agaricales. These are Bolbitiaceae, Agaricaceae, Cephalaceae, Strophaceae, Entomoletaceae, Luteaceae, and Lyophiliaceae. From Bolbitiaceae, 2 genera were discovered, Bolbitius and Conocybe. 6 species were discovered from in the genus Bolbitius and 16 species in genus Conocybe. These are some of the pictures of species which we investigated from growing on different types of dung, mixed dung, cattle dung, or cow dung. In family Agaricaceae, the gender reported are Coprinus, Chlorophyllum, Leucoagaricus, Leucocoprinus, Lepiota, and Agaricus. So we reported 2 species as new, Agaricus stylatus cuticus and Agaricus flavis typus. These are the pictures of Coprinus collected from dung, Chlorophyllum, Leucoagaricus, Leucocoprinus. These are the Lepiota species which are growing on herbivorous dung like Lepiota xanthophylla, Lepiota thrombophora, Lepiota apicaris variety occidentalis. Agaricus species were also reported, and the first one is Agaricus dilatus cuticus, which is new to science. Then in the family Cephalaceae, 6 species are reported in the genus Cephalella and 16 species belonging to genus Paniolus. New varieties like Paniolus africanus, variety diversitabus, and Paniolus speciosus, variety Pilocystidiosus were reported. Then in family Cephalaceae, Parasola, Coprinopsis, 12 species were reported in the genus Coprinopsis, and 3 species are reported in the belonging to genus Coprinellus. These are some of the habitat pictures of Cephalella, Paniolus, Paniolus cynoannulatus. Some of the pictures of belonging to genus Coprinopsis, Coprinopsis sedonivia. In family Strophareaceae, the gender reported are Stropharia, Agrocybe, and Psilocybe. So Stropharia semiglobata, variety Punjabensis. This is the new variety which we reported. Then Agrocybe adides. Some of the pictures of Antolomotaceae. In family Pluteaceae, we reported 2 genera, Volvarella and Volvopluteus. 2 coprophilous collections were made in the genus Volvarella and 2 in Volvopluteus. In family Lyophiliaceae, only 1 species, Termitomyces adicatus, was reported. So as we investigated the genera with respect to distribution, ecology, and seasonal availability, it is found that the genera belonging to family Cephalaceae were the highest, and Entolometaceae, only 1 species were discovered. Then genus-wise distribution. This figure shows a species-wise distribution. Maximum species belong to Cephalaceae and then Agaricaceae, and afterwards Bolbicaceae. Then this shows the seasonal occurrence of investigated Coprophilus. Maximum species were reported in monsoon season, followed by summer and then winter. Only 0.6% were reported in autumn season. This is the month-wise distribution. No species were found in the months of April and October, as this is the transitional season in the state of Punjab. Then this shows the species-wise distribution of investigated taxa in different seasons. Species-wise, 58% species belongs to monsoon season. This is shows the frequency of occurrence of Coprophilus mushrooms on different dung type. Maximum species were reported on mixed dung, then on buffalo dung. We reported small percentage of species growing on deer dung and rabbit dung. Then some species were reported on sheep dung and elephant dung. Then occurrence of investigated taxa in different growing habits. Most of the species were found scattered, and only small percentage were observed in fairy rings. This is the genus-wise distribution in different growing habits. Then percent distribution of Coprophilus taxa on the basis of their economic utility. 59% species were status is not available, and only 16% species were found edible. So we worked out 172 collections, and new species, 5 new species were reported, and 7 new varieties were reported. So the study shows that dung is an important substrate for the growth of Coprophilus mushrooms. Thank you. I'm very thankful to my revered guide, Dr. N.S. Atri, and Madam Dr. Manruchikar. Thank you.
Thank you very much.
Thank you, sir.
I request Professor Navi to come onto the dais.
Thank you, respected participants, all sir and ma'am. I sincerely thank you for enriching us for your knowledge. And then we sincerely appreciate the presence of Professor L.B.K.S. Bhaskar sir for ensuring the session that runs well. Thank you all for participating. ये अपने पेज निकल रहा हमारा
Thank you very much. I declare the session is. Thank you, sir. So good afternoon, everyone. I am audible. Hello. Am I audible? No response. सर देखिए तो नहीं हो रहा है क्या?
Yes, yes, you are audible.
Ah, yes, sir. So good afternoon, everyone. So very bad. Yes. Yes, sir. So I'm Dr. Muriasan, the chairperson for this session. So this session 7 is dedicated to the Dr. M.J. Tirumala Chair Young Scientist Award Lecture. So in this session, we have totally 16 lectures, both online and offline. Offline, we have the 6 lectures, and online, we have the 10 lectures. For each lectures, we have about 10 minutes presentation. So within the 10 minutes, 7 to 8 minutes for presentation, and then remaining time for the.
There is a revision because of time constraint by the organizers.
Okay.
So,
How much?
We are already delayed by 2 hours.
Yes, sir.
Just a suggestion if we can cut down the time to 5 minutes.
Yes, dear participants, due to out of time, so we reduce the time of your presentation time. So just make a 5 minutes presentation and 1 or 2 minutes question and answer session. So be strict with the time because we are out of time, and also our organizing secretary asked us to restrict the time. So now I request the first presenter, Kamhandas Bhaskar. How many are in offline? Yes, sir, please.
Do we have Kamban Das Bhaskar?
Yes, sir. Yes.
There. And then we have Rohit Shukla. We have Mohi Shukla. We
Yeah,
have
you can.
Srishti Verma, Deepika Mahobia, and last we have Chandrahas Devangan.
Okay. So I also request all the online participants, presenters, be ready for your presentation. So you will call one by one. Yeah.
presentation देना है या अभी नहीं उसी समय से
So first we have 6 offline and then followed by 6.
इसमें एक फोल्डर बना दो, ठीक है, डेस्कटॉप पे सेशन के नाम से सारे प्रेजेंटेशन हर पर्सन के नाम से बना, ठीक है, ताकि कंफ्यूजन ना रहे.
You put the number also 1 or 2 like that.
Can do that. Yeah.
Meanwhile, the presenters are requested to kindly load their presentations so that we can save the time. Kindly hand over to the person dealing with, so that we can get all of
Sir, voice is not coming.
ஐயோ எனக்கு கால்ல நடுங்குது எல்லாரையும் அதுதான் சேர்க்குது நம்மளே சேர்ந்து யாரும் இல்லையே
என்ன வேலை பண்ணிருக்கீங்க ஏதோ வேற ஏதோ லிங்க் கூட்டு பண்ணிருக்கீங்க போல வெளியில வேணாம் நியூ டேப் ஓபன் பண்ணி தான் கொடுக்கணும் திரும்ப லோட் ஆகும் நீ கூட பரவாயில்ல கூட ஏதாவது வந்துச்சுன்னா பாத்துக்கலாம்
Hello, sir. Okay, presentation. Presentation. Hello, sir. Hello. Hello, dear sir. Hello. Dear sir, can you hear me? Hello? Hello? Hello, sir?
We can hear you, but I think the chairperson is not at ready. Dr. Murugesan?
Okay, sir. Okay, sir. Sir, my screen is it visible, sir, now?
Yeah, it is sharing.
Okay, sir. Is it movable, sir?
Yeah, it's moving.
Okay, okay, sir. For you, sir.
So, Dr. Murugesan? Dr. Avneet Pal Singh?
Okay, sir.
Dr. Harijah or Dr. S. Banerjee?
Dear sir, hello sir.
Good evening, Dr. Murugesan. Can you hear me? Anybody from the organizer side online for this session? It shows Dr. Harit Jha, the coordinator for this parallel session, and Dr. S. Banerjee as a reporter for this parallel session. Anybody in charge of this parallel session from the organizer side? Professor K. Murugesan. Seems to be a lengthy break.
Dear sir, dear Sharma sir, sir, can you yes sir,
Yes.
sir, can you contact Murugeshan sir, because sir it is delaying too much sir, because many of them are there.
No, maybe in the offline mode, they are waiting for people to assemble.
Okay, okay. Sir.
Oh, no, I got a message that there is a power cut there.
Okay, okay. Sir.
So we have to wait until the power is restored there.
Okay, sir.
He says there is no PowerPoint due to power cut and network is not there. So maybe they are waiting to sort it out.
Okay.
I received a message from Professor Murugesan that they are going to shortly sort it out.
Okay. Thank you, sir.
Yeah, welcome.
Dear Sharma sir,
Yes.
Sir, you can if offline lectures are over, so you just ask them or request them to go ahead this one, sir. Otherwise, sir, it is getting
No, no,
delayed.
no. He just messaged me saying that problem is being rectified. So
Okay.
they are going to connect it anytime now.
Okay, sir. Okay, sir. Thank you, sir.
நமக்கு இப்ப கடிதம் வந்தாலும் திருப்பி பேசுவாங்கல்ல
வாய்
இதுக்கும் முடிஞ்சுதானே இருக்கும். இது இந்த
என்னதான் காட்சி ஹலோ
நல்ல தப்பு.
Good evening, sir.
Good evening. Good evening. Good evening. Good evening, Dr. Sharma.
Yes, sir.
Yeah,
Some power cut in சொல்லிருந்தாங்க.
Okay.
So I think they are rectifying it.
Okay. The rest went on well.
I think so. Yeah.
Yeah. Okay. Okay. Right. Right. Yes. Very nice.
This is night time the morning, no, for you?
It is morning 6:45.
Okay.
Yeah, it is 11.5 hours difference between India and US time.
You had a good sleep?
Yeah, no, I was there up to almost 12 o'clock. Then I went to bed and 5:30 I woke up.
Okay.
Yeah. So congratulations for the associate managing editor for the next 3 terms.
Thank you, sir. Thank you.
Okay, okay. How is your work is going?
Yes, sir. It's going. I have 2 projects.
Okay, good, 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 good.
We are doing both the molecular and morphological diversity.
Diversity. Okay, okay.
Of marine fungi from Andamans.
Okay, okay. And you can also bring out a issue. So every time the section editors they are bringing one editor, they are editing one issue, no?
Yes, sir. Yes, sir.
And you managing editor and as well as the but of course the managing work will be more. Apart from that, if you want to bring one issue, you can tell me. I can put the slot.
Okay. But already Professor Kalaiselvan is going to do one marine fungi, one volume.
Not necessary on marine fungi. Now we have not restricting to the theme.
Okay.
Because sometimes the theme, some papers may not come. So what we are doing is, if the theme is coming, otherwise we are getting other papers also.
Okay.
you have to receive manuscripts and send to reviewers, and then if it is good paper, you can edit it and you can accept it, and then you can 10 articles per issue you have to bring. Yeah, you can think about it. Okay.
Sir,
Yes.
one special issue is going to be released next year by Professor Attri, one book.
That is the biographic sketches of mycologist of India,
Yeah.
past and present. That will be released in the Golden Jubilee celebration.
Yes.
It is under preparation and he is doing it. You have sent your biograph?
Yes, sir. Yes, sir. I sent Professor Vittals also.
Professor Vittal, you sent. Okay, okay. Yeah, you prepared that this thing, no? What is that? The obituary for Professor Vittal.
Yes, sir.
And this biography sketch, how many pages it is coming?
No, actually, Professor Attri said 3 pages. So we try to restrict to 3 or 4 pages for each
Okay,
person.
okay. Okay, you can send that also so that I can put it in the file. So whenever the award lecture starts, we are reading the biographic sketches, no?
Yeah, okay, I will send soft copy. Whatever
Okay,
that I have.
yeah. Yes, the session is still going or what?
No, it has been interrupted because of this power cut.
Power cut. Okay.
More than 45 minutes.
Oh, okay. Still, it is not resumed.
Okay. And due to power cut, there is no internet.
Internet, yeah. But probably other sessions are over. Or only this session?
I think so. Because I just contacted Professor Murugesan. He said they are rectifying it. Any moment it will come.
Okay. And I contacted Dr. Susil. He told that only this session 2 like that.
Okay.
Because of the interruption. Dr. Panniselvam is there?
Seems to be.
I will check whether other sessions are going. But if the power supply is gone, means then all
It
the
may be
sessions.
for all other halls also.
Yeah.
I sent a message to Professor Madhivanan. He said they are still attending.
Yeah, okay. Yeah. There are some, they have technical problems. There are a lot of problems are there.
But if they have internet connection, we can make everything online.
Yeah, everything make. Another thing this time, I didn't compel him to club it. Too many parallel sessions will make it difficult for them also.
To gather the crowd.
Because the 4 halls, they need the 4 halls.
Yeah. Any idea to give it to Springer or anybody, sir?
Yeah, I am. I asked Professor because they are in Delhi. Earlier, we Springer we could not materialize because they told the number of members are less.
Okay.
So that's why we could not materialize during that time. Now we have raised our member above 500, and now we are bringing 4 issues per year. So the next 2 pending are one is the Scopus index. For that we are working on it, and I have requested Professor Rupam Kapoor, and you also explore the possibilities. If some agencies they can assist us to send the application for a Scopus. So it will be easy for us to get the indexed.
I will ask some of my colleagues here how to do it.
Yeah, MSI can pay the nominal charges which they are charging for their assistance.
Okay.
I think once Professor Atri tried, but it was not materialized.
Because of that lower number of
Lower number and the issues per year. At that time, only one issue. I think one issue.
Okay.
So now, then later we made into 2 volumes, and then now we have 4 issues per volume. And the response is also good now. And then now we made a mandatory that every those nominations with for fellowship, they should have minimum 3 papers.
Okay.
So that's why we didn't get any nomination this year.
For fellows.
Because people will speak many things, and they will publish in other journals, and they will give only suggestions to MSI only. Just do that, do this, like that. So we thought of making it five publications, but initially let us try for three publications. Probably next year we get some nominations, because this year many people have published in the Kavaka.
And once it is Scopus listed, many people will come forward to publish.
Yes. And I asked Professor Rupam to explore the possibilities of
getting listed in Scopus.
Scopus, as well as the Springer publication, like that. Springer publication, like the managing editor, she has some experience. I think she told Professor Rupam Kapoor. Yeah, Valia. And she is good at with the journals. So that's why she prefer from Delhi and her. And it is easy for them also. No? Managing from the same place, it will be very easy.
Delhi.
Yeah.
Sorry, maybe you would like to contact Professor Madhivanan or somebody there.
I have already contacted him, and they told he told the same thing. This thing, power failure. And also I contacted Susil. So he also told, he didn't say about power failure. Simple. Hello, that Professor Madi, இன்னும் power வரலையா? இன்னும் 20 minutes ஆகுமா? Oh, 20 minutes. But this only one session and other sessions are completed. மூணு செஷனும் இப்போ இன்டரப்ட் ஆயிருக்கு. ஓகே ரைட். செஷன் 1 போயிட்டு இருக்கா? ஓகே. ஃபினிஷ்டுன்னு நினைக்கிறேன். சரி, பார்க்கலாம். ஓகே ரைட். ஓகே, குட். ஓகே, தேங்க்யூ. ஓகே ரைட். ஓகே. ஒரு இது ரெஸ்யூம் ஆயிடுச்சான், பவர் ரெஸ்யூம் ஆயிடுச்சான்.
ஓகே.
இன்னும் ஒரு செஷன் போயிட்டு இருக்குனாரு. அங்கே போயிட்டு வரேன் நான். ஃபர்ஸ்ட் செஷன் என்னமோ போயிட்டு இருக்குனாரு.
ஓகே,
போயிட்டு வரேன்.
Hello.
Hello, sir.
Sir, about to start. Dr. Patel is going to start now.
Yes, sir. I think so, sir. They are working on it. I think so.
Okay.
Hello. Yes, sir. Tell me, sir. Shall I start my? Shall I share my slides, sir?
May I know your name? Arjun Lal Yadav?
No, Dr. Niranjan. The first oral presentation online.
Niranjana M.
Okay, so you can give that name also. Okay, so I think they didn't send that PPT earlier, and then they can put. Okay, you are online now. Okay, you can proceed. Yeah. Hello.
Yeah. Thank you, sir. Yes, sir. I'll share the.
So just a moment. So this is due to some issues here. Just we cut short the time to the 5 minutes presentation. So just focus on your objective, and then key findings, and then the conclusion. So 5 minutes and 1 minute question and answer discussion.
Okay.
So this is the same for all the other participants. All the participants in this session only 5 minutes. So you try to complete within the 5 minutes. Yeah.
Sure,
Thank
sir.
you.
Sure. Thank
Okay.
you.
So you can proceed. Share your PPT. Yeah.
Sir, is it visible now?
It is visible here.
Yeah. Is it speaking H class?
Shall I begin, sir?
Yes.
Okay.
So then now totally 6.
Sir, can I start?
I just a moment, sir.
Shall I start, sir?
Just a moment. Yeah, we are getting ready now, and just a moment. I will just ask you to present. Then I would like to know how many are online. Is there Arjun Lal is online? Hello.
Connection issue.
Just wait, sir.
No, Arjun.
Okay. Sir, Chetana is online. Yuvarani. Hello. So let me confirm that Yuvarani, are you online?
Hello, sir. Good evening, sir.
Okay, okay. You stay online, ma'am. I will call you. Okay. Then Anushri Tiwari. Valli.
Yeah, sir. Good evening, sir.
Okay. Okay. Good evening, ma. So you just stay online. The one by one we call you. Okay.
Okay, okay, sir. Sure, sir.
Sir, I just speaking online. Yeah, you can put. You can.
Laptop.
Yeah. Yes, you can put it there, sir. We don't need to.
Shall I start, sir?
Yeah. இல்ல அங்க வந்தா கூட பரவாயில்ல இங்க இருக்கு வந்து நின்று பார்க்க So no issues, sir. We can put it here. No. அங்க லேப்டாப் இங்க வச்சாலும் அங்க வரும் சார். Yeah, vector. That's okay. Let's see your on. Oh. Just we need to see it. Okay. For audio, we need to get it. Yeah. Yeah, we can we move this stability? Yeah. Yeah. Okay. Yes, sir. Dr. Niranjan, sir, you can
Yeah.
start your presentation.
Okay. Thank you, sir. Thank you, sir. Okay. Good evening, everyone. I thank you for the Mycological Society of India for inviting me to have a oral presentation. So today I would like to present my PhD work. The title of the presentation in MSI, Diversity of Hysterials in the Andaman Islands. I'll quickly start. So in my abstract, I presented 10 different species belongs to the order Hysterials, in which 4 new species I'm going to present now. And the key characteristics of Hysterials are basically the Hysterials are belongs to the Dotidiomycetes or bitunicate fungi, belongs to the phylum Ascomycota. The key features of these Hysterials are mostly are in saprobic in nature, occurred in twigs, barks, and wood herbaceous plants in both terrestrial and aquatic environment. The ascomatal characteristics of Hysterials are mostly carbonaceous ascomata, longitudinal statures, bitunicate assay, hyaline or brown phragmo or muriform ascospores. So among these 10 species, I am going to, they are belongs to the 5 different genera. The first genera, I'm giving a brief characteristics of each genera. As you can see in the slides, the first genera, genus called Hysterium, they are having most of them phragmospores, that means the horizontal septata, mostly that are 3 septate, and very few are more, but without swollen supermedian cells. And the second genera, Stereobrevium, it consists of dictyospores and hyaline in nature, and they may or may not have the gelatinous sheath or pigmented. And the Gloniella, which is having very few species, and the spores are belongs to Gloniella are mostly hyaline phragmospores, and very few are dictyospores with red pigmentation. And again, the rated Hysterium is almost closely related to the type genus of the Hysteriaceae or Hysterium, closely related to the Hysterium. The major difference of these Hysterium and rated Hysterium differences is in ascomata containing the linear cracks. In the case of rated Hysterium as the parallel cracks. And the last one is Asterocanan. It is again the carbonaceous feature, but still the spores are mostly having the one septate and having the longitudinal cells, oblong cells, having the obtuse ends with or without sheath. So the first species is Hysterium hyalinum. As you can see in the slide, linear slit in the ascomata, and cylindrical assay, which are having the overlapping ascospores, and pseudoparaphysis, which mostly having aseptate linear without branching. And most of the spores in early stage, they are in hyalian in nature, where it grows in mature becomes dark brown, and which having consist of the three septate with central septa constriction. The second species called Hysterium rhizophore, it's almost similar to the Hysterium as in the case of ascometal features. The major differences in the ascospores and their length and breadth, which are not having few constricted and non-constricted septa. And the third species belongs to the Hysterium is Hysterium village. The most of them, these spores are having several septations rather than having the three septate. In this particular species are having the 13 to 14 septate, in each septate having the constriction. And the second genera Rhytidisterium, as you already told, that which consists of the linear cracks. And the spore features are most similar to the Hysterium. Our initial hyalian turns to dark brown when it matures with constriction in the each septa. And Rhytidisterium rubrum, this is the new species which we described in our study. Except from the ascometal features, the ascospores are turned to hyalian to dark brown or reddish brown sometimes. So they are very peculiar characters belongs to the Rhytidisterium. Therefore, we have been described as new. And the last species, Rhytidisterium versicolare, which is very known and new to India, which is consists of again three septations with central septa constriction. The third genera Hysterobrium andamanicum, we describes based on these spore septations. Most of these spores are having the dictyospores, which horizontal and longitudinal septations. Here in our new species having two different linear septations. This is the peculiar character of this species. And again, the second species is Hysterobrium mori, which is new to Andaman and India. This is again spores having the brown, having the longitudinal and horizontal septate, central septation, strong constriction.
Dr. Niranjan, your time is over. Just
Yes, sir.
kindly.
Gloniella aptinia, which is longitudinal cells which are having the 3 to 5 septations with obtuse ends. The last species is Asterocanan bijakoshe. We presented as a new species because of the presence of the sheath over the ascospores. And among these 10 species, we try to raise the culture, and one species we successfully able to raise a pure culture, that is belong to Rhytidisterium ruflum, and we made a phylogeny based on the 3 different softwares, RaxML, POP, and Mr. Bayes. And the culture which we get and herbarium specimens we deposited in NFCCI, and the descriptions were submitted into Mycobank, Faces of Fungi Index, Fungorum, and the tree-based software we submitted in the phylogenetic tree, and sequences we submitted into NCBI. Thank you.
Okay, thank you. So finished within the time. So any question from audience, participants, online participants? Also from the
Any question?
participants from here?
Dr. Niranjan?
Yes, sir.
What kind of septa are present in these ascospores?
Sir, mostly phragmoseptate, sir, horizontal septate.
Are they primary or secondary septa?
No, sir, they are the very prominent septa, sir.
No, I'm asking, are they primary or secondary septate?
I couldn't able to understand, sir, what it means primary, secondary.
Oh, in that case, that let's leave it. Okay.
Okay, thank you, Dr. Niranjan, for your presentation. So now I call the second presenter. You can leave. Okay, now I call the second presenter, Arjun Lal Yadav. There.
Arjun?
No.
Okay. So now move to the Chetana.
Young break. Do we have Chetna? No, Yuvraj.
Okay. So now,
Hello, sir.
now
Hello,
yes,
sir.
you're online now?
Sir, my name is Basim.
Team, let's make the team.
Wasim, yeah, okay. So you didn't send your PPT earlier?
Sir, actually, unfortunately, I have joined with another link.
Hello, sir.
So I
Oh,
didn't
okay.
join, sir.
Myself, Ivrani, sir.
Ivrani, just wait, ma. We will call you. Okay, so now,
shall I present now?
Wasim, no?
Yes, sir.
Okay, so you can present your topic. So,
Thank you, well, sir.
Wasim Agram is going to present the diversity of endophytic fungus associated with the Costus species. So you can proceed your presentation within 5 minutes, and then 1 minute or 2 minutes. The question session, yeah.
Thank you very much, sir. Sir, is this
You can
slide
share your
visible?
PPT, yeah. Please share your presentation. Yeah?
Sir, I'm sharing. Is this visible to all?
No. You're not getting.
Sir, kindly make me a presenter.
So we already.
Kindly make me presenter, sir.
Now she has asked him to.
Without file, there are many of them are non-in.
Sir, I'm unable to share. Kindly make me presenter.
Just try now.
Now?
Wasim, try now. You have been made the presenter. Try it.
Yes, sir. Yes, sir. It's showing now.
Yeah.
Okay, it's coming, yeah.
So it is visible now?
Okay. Yeah, you can go to directly to the objective and no need of the background, yeah.
Sir, I'm a research scholar at Science College, Durg. My diet is Sir Dr. Sriram Kunjam. I'm working on the diversity and external enzyme activities associated with fungus, associated with Costus species. Now, sir, this is the plant. Costus species is the endangered plant of Chhattisgarh, belonging to family Costaceae, and it has many of the
Wasim,
common genera belonging to
hello,
these
Wasim?
Alternaria, Aspergillus, Fusarium,
Wasim?
Parishes. To comply
Hello?
these studies, an isolation of fungus has been done, and followed by this fungal
Okay.
isolate were treated for their biochemical activities.
Wasim?
Yes, sir.
You can skip that all the background information, so you can go directly to the objective and then key findings and yeah.
So I'm
You
showing
can say
the endophytic fungus isolated by me from the Costas plant. That is, the first one is a master plate, and the both second and third is the isolates. So moving from our Aspergillus species isolated from the Costas. So the left one is the Aspergillus niger, and the right one is Aspergillus. These are the microscopic photographs of the conidiospores. Okay. And these are the Fusarium species under the light microscope. You can see, sir, the micronidia of the Fusarium species. Now moving towards the, sir, screening of endophytes. So these are the enzymatic activities result. So positive charge for positive for cellulose activities and positive result for pectinase and amylase activity. Now, sir, showing the conclusion of the results. So these are the species isolated from the species, and these are the combined studies results according to amylase, pectinase, and cellulose activity. Sir. So the maximum studies are shows penicillium maximum positive test for amylase, cellulose, and other species shows the moderate level results. Sir.
finish.
So slide is not changing.
Yeah, it is from your end, no?
I don't know what is happening. Slides are stuck.
The file is on your control. You are the presenter.
For your system.
No, he is present. He is from East.
No.
Yeah, we got it. Yeah, okay.
Yeah, now it's changing.
Go to the
Go
conclusion.
ahead.
Yeah. Conclusion.
Yes, sir. Now this is the conclusion, sir. The study concluded general penicillium showed maximum positive test for MLA cellulase and test for pectinase was negative, whereas the other species showed moderate level of enzymic activities. So thank you, sir, for giving me
Okay,
a
thank
chance
you.
to present this work.
So, thank you, Akram. So, any questions from the participants, online participants? Basim,
Yes, sir.
what is the difference between penicillium and aspergillus? How you can differentiate?
Sir, we can differentiate by the spores, conidial spores, sir. If you see, sir, the spore of aspergillum, it will be a single and bird shape, sir.
Any difference in conidia for morphology?
Yes, sir, definitely, sir. So penicillium have the finger-like projections, which is not seen in the aspergillus.
Thank you. Okay, thank you. Yeah. Okay, the next one. So you can leave that meeting.
Kindly stop sharing. Kindly
Yeah.
stop sharing. Yeah. Thank you.
The next one, Chetana.
Hello, sir. Hello.
Yeah, you can. Yeah, you
I
can.
am Snehal Biranje, sir. After Arjun Lal Yadav, I think that there is my name.
What's your good name?
Snehal Biranje.
Okay, yeah.
Okay. Go ahead.
Okay, should I?
You just remove him
Remove
and
them.
make
Remove
her
them,
present.
Okay.
Wasim, please stop sharing from your side. Wasim,
Okay, sir.
stop
Yeah,
sharing
yeah.
your screen. Thank you.
Sir, allow me to share the slides.
Yeah. Wait.
Okay.
Washim, can you leave the your PPT?
I am leaving the PPT.
Snehal Sudhir Biranje.
Snehal Branchi.
Stop sharing.
Yeah.
Yeah.
Okay.
Next one, yeah.
Snehal, go ahead. Snehal, go ahead. Share your screen.
Okay, sir. Is it visible,
Yeah.
sir?
We're getting. Yeah. Maximize. Yeah.
Okay. Shall I start?
Yes. Okay, you can continue now.
Okay. Yes, sir. I will complete it within the time. Good evening, all. My name is Snehal Biranje, and today I am presenting here the research entitled First Report of Black Volumyces pseudomilitaris from Western Ghats of Maharashtra. Here is the hypothesis. When we collect the material, we found that the material was morphologically Black Volumyces pseudomilitaris. After we done the ITS sequencing, we confirmed that it will be a new report to the fungi of India. How to approach the work? We with the help of different methods like collection of specimen to up to the molecular identification. Purpose of work that the species Black Volumyces pseudomilitaris and the group is an entomogenous fungus, and many of the species from this group mainly used as a biocontrol agent as well as in the pharmaceutical products. So in this present work, we are analyze the fungus as a probable source of biopesticide or pharmaceutical product. So here are the material methods. Here are the material methods. We collect the specimen from the forest region of Kolhapur and brought to the laboratory, and the further analysis will done by the morphological and microscopical analysis, and further process with the preparation of different media like a PDA and the saberdextrose agar with yeast supplement. And it is followed by, after the isolation, we prepare the DNA extraction, PCR, and the sequencing by the method Amir S. Sutar, Amires et al. In 2015, and it further the sequence process for the blast for the identification.
Yeah, move. You can move your slide. Okay.
Here are the results, the morphological and microscopical analysis result. So the specimen we collected from the field on a Lepidopteran larva, which is type is about 25 to 50 mm and the stomata is about 12 to 25 mm long. While we're taking the section of the stomata, we found that there are perithecea more or less superficial or immersed at the base. And this perithecea apex is prominent, elongated and ellipsoid or elongated to the ovoid in structure. While the next, that is the ascus. In the perithecea, ascus is present with a prominent ascus cap, which is a key character of this group. The cultural characteristics. We found that the culture, when we isolate the specimen into the pure form of the cultures, we have a whitish mycelium. After the some time, the whitish mycelium turns or shows the little pigments, but the major pigments secreted in the media, and it is clearly visible in this picture that the media is completely full of with the pigments. But after the mycelium, after the dark period, when the mycelium grows, we expose it for the dark period of about 8 days. And after that, we expose it to the light, about the 300 lux. And we find out that the mycelium start compacting and formation of the fruiting bodies was there in the test tube. And final result was there. The exact formation of the stromata is found in the cultures. So after the successful isolation, we go for the molecular identification. And in the molecular identification, after the DNA extraction and the sequencing, we have this result that our sequence clearly matched to the Cordyceps pseudomilitaris, which is a previous name of the Black Velomyces pseudomilitaris. And then we go for the FTR analysis. Here we use two methanol, so methanolic extract for the fruiting body which we collected from the nature, and as well as we use the cultures for the analysis. And here we got the same peaks in both in natural sample also and in the culture samples also. That is CO stretching was there, CN stretching is there, aromatic ring was there, amides and aldehydes also present. And these the results we match to and compare to the previous workers, and which is I included in the result significance. That result significance of my work, total that collected material was morphologically and microscopically analyzed, and the fungus was successfully isolated in the pure culture up to the fruiting bodies successfully development, and the molecular analysis matched with the morphological identification, that is Myces pseudomilitaris, and it is have a quite similarity with the Cordyceps militaris. The FTI results in the studies were quite similar previous research where the same functional groups were detected in the Cordyceps species like a Cordyceps militaris as per the Joshi et al. As per in 2015. Cordyceps contains the amino acid, fatty acid, nucleotides, and the as per the biochemical analysis, it shows the potential sterols, mannitol, polysaccharide, and many other components, and in which oleic acid and D-mannitol are high in a content as per the Liu et al. As we also have the same result for the FTR analysis, that is D-mannitol is present there. And after the work, I will conclude that the Black Velomyces pseudomilitaris is a first report from the Western Ghats of Maharashtra. And these are some references.
Okay.
Thank you.
Thank you. Thank you. Thank you, Brinja. Any questions from the online participants on her presentation?
Snehal,
Yes, sir.
what is the difference between black velomyces and metacordyceps?
Sir, metacordyceps mostly have the color difference or the stromata. Mostly the metacordyceps not having the fleshy stromata. It is quite pliant in structure, while its color is whitish lilac in the structure. But in the black velomyces, it is a bright colored orange to yellow, and its key character that is type is whitish in color, and it is also shows difference than military, sir.
Okay, the title which you have communicated to the abstract for the abstract volume, that was on metacordyceps link. Yeah. And you are
Yes, sir. Yes,
presenting.
sir. But I already present this in the poster, rapid fire poster, sir.
So you submitted two abstracts for today's presentation. Huh?
Yes, yes, sir. Yes, sir.
Okay, thank you.
Okay, thank you.
Okay,
Okay, you
thank
can
you, sir.
leave your. You can leave. Okay, the next one. The next participant is person that is Chetana.
Do we have Chetna?
Yes, yes,
She's there.
okay, if she's not there, then Yuvarani. Yes, she's there. Yeah.
Hello,
Yuvarani, you
sir.
can do. Yeah, you can make your presentation. So the next person is Yuvarani. She is going to present the diversity of litter decomposing agaricus from the Pondicherry.
Yes.
Yuvarani, you can go ahead. There are your slides and briefly present your objectives, key findings, and conclusions. Yeah, within 5 minutes.
Okay, sir.
We take time.
Next, okay. Yeah.
Connecting problems to the ஆகுதா இல்ல இது முன்னாடியே நோட் பண்ணிக்கணுமா
Yuvarani, yeah.
Sir,
Slide is loading, sir.
Okay, okay, yeah.
Sir, is there any possibility to show the slide from your end, sir?
Oh, we have his
Can you present here? Yeah.
Okay, just hold on, beta.
Just.
Yuvrani, kindly stop sharing your screen. Then we will be able to share from here.
Okay, sir. Okay, sir.
Just stop sharing. Do we have our presentation?
Yeah, open it.
But first he has to stop sharing.
Yuvarani, you can stop your sharing, your file sharing. Just play.
Hi,
Okay, waiting. Close, but still here you are unable to share because it shows that you are sharing.
Yeah. Now she stopped.
Okay. Share your presentation.
Two only, sir.
Two more. Three. Including it. No, many joined.
Just joined.
Yeah.
Now Chetana is not available. Now Yuvrani is. We have Anushri and Vali.
Yes. Yuvarani, you can present.
Okay, sir. Good evening, sir. I am Yuvrani. Topic of my presentation is diversity of litter decomposing agarics from Puducherry.
Okay.
Next.
The next one, yeah. You need to stay here and. Okay, go to the objective. Go to the objective. Yuvarani, go to the directly objective. Okay.
Mushroom diversity studies are being done to document mushrooms occurring in southern India. Not much on agaric diversity has been carried out in Puducherry.
Yuvarani, so go to the objective. I think you are another PPT. Okay, you are getting our slides.
Yes, sir.
Then objective. Are you seeing the objective? Second slide, yeah. Background you can skip, ma.
Yes.
Yeah. Present the objectives.
To collect the litter decomposing agarics around the region during rainy season. To observe and to record the macroscopic and microscopic characters of collected agaricus as present and scintillars and cumbration. DNA extractions of DNA from the fruiting body of the agarics by setup method and identified by sequencing internal transcript base. Next time. I collected 11 species of litter decomposing agarics belong to 6 genus and 4 families. Marasmia species is the dominant species. Next time. Here we can see one by one the micromorphological structure of agaric species. First one is Gymnophus gyposus and second one is Mycena notiolens. Next one. Third one is Marasmus emetocephalus. Fourth one is Marasmus midnophurensis, was first reported in southern India. Next time. Marasmus palmivorus and Marasmus Tetraphyrogus species. Next. Troja bengalensis and Fluorotus cystiosus. This phylogenetic tree shows the evolutionary relationship among the isolate different Marasmia species that is confirm the identity. Next slide. 11 agaric species belong to 6 genera were collected, which belong to family-wise Marasmiaceae, Mycenaceae, Ophiolophyceae, and Flutaceae. Among these 6 species belong to Marasmias and Flutus, Tetraphyrogus, and Tragia. 9 species of agarics are being reported for the first time from Puducherry. Studying micromorphological character internal transcript base. Thank you, sir.
Okay, ma. So nice presentation, and we completed within the time. And any questions, sir, from the participants on our presentation about the litter fungi? So from your side, sir.
Yuvrani?
Yes, sir.
How Marasmius differs from Agaricus?
How the Marasmius is different from the Agaricus? No?
Tiny size.
Is it?
Tiny size, white spore, sir.
Okay, thank you. Go ahead.
Okay, ma.
Anushri.
The next one is Anushri. So, Anushri, you can join. You can present. So Anushri is going to present their topic on the biodiversity studies on mushrooms found in Achanakama Tiger Reserve, Chhattisgarh. Anushri?
Do we have Anushri? No, she's not there. Go with Bali.
Okay. Then Vali?
Yes, sir.
Yeah, you can present. You can share your slide, ma. So Vali is going to present the topic on the diversity of leaf endophytic fungi in mangrove plants, Kodikara in Nagapattinam district of Tamil Nadu. Share your slide, ma.
Sir, I'm trying to share. Sir, I'm trying to share, sir.
Okay.
Five years. Five.
Okay.
4 presented and we are the last one.
And only 5.
See, that's done. Otherwise, now tomorrow we will be put to start into this.
Sir, I'm not able to share. Please allow me, sir.
Did you send already your
Yes, sir. Yes, sir.
Okay. You will share that slide from our side, Ma, then you present, okay?
Okay, sir.
Valli. Now you will get Ma the slide, then you present, okay?
Okay, sir.
Then ask the next slide. Next slide we will move here.
Good, beta. Thank you. We are doing a lot of.
Shall I present, sir?
Yeah.
Good evening to all. Today I am here going to present the diversity of leaf hydrophytic fungi in mangrove plants of Kodekkarai in Nagapattinam district of Tamil Nadu. Present by me. Next slide, sir.
Next one.
Introduction, material and methods, collection of plant samples. The leaves of 3 different plants were selected. The plants Sueda martima, Avicennia marina, and Siliconia brachyta were collected from Kodekkarai forest. Healthy and mature plants were selected for the sampling. Next slide, sir.
Next one.
The healthy leaf materials were thoroughly washed with running tap water. The surface sterilized by Raviraja method. After the surface sterilization, the leaves are cut into pieces approximately 1 centimeter length and plated on the culture medium. Normally used potato dextrose agar medium. Streptomycin is added to prevent the growth of bacteria. Then the plates are incubated under sterile conditions and monitored every day for the growth of endophytic fungal colonies. Next slide, sir. The fungal growth out from the plate tissue were subcultured on the fresh antibiotic-free medium for identification. The identification is based on morphological examination and phonetical characters. Colonization frequency was calculated as directed by the Suryanarayanan et al. And then the slide, lactophenol cotton blue mounting. The loop full of culture was picked with the help of sterile inoculation loop, and the slides were prepared. The slides were gently heated. If any air bubbles is present in the cover slip inside the cover glasses. Results, the isolation of endophytic fungi from Avicennia marina plant. Then next slide, sir. Isolation of endophytic fungi from Sueda martima plant. Isolation of endophytic fungi from Salicornia brachyta. From these 3 different plants, I have identified 18 organisms, 12 from Avicennia marina and 8 from Sueda martima and 12 from Salicornia brachyta. Next slide. Discussion, conclusion. The endophytic fungi are one of the most unexplored and derived group of microorganisms that make symbiotic association with the higher life forms and may produce beneficial substance for host. Endophytic fungi is an important source for bioactive compounds and have a wide range of applications in medical fields. So hence the present study will help to dedication of potential drugs from endophytic fungi. Thank you, sir.
Thank you, Ma. Thank you, Ma. So any question on our presentation from the online participant? Yes, sir. Yes.
Okay, thank you, Bali.
Thank you.
Thank you.
Yeah, you can skip. Yeah.
Thank you, sir. Thank you, sir.
So we have come to the conclusion. So in this session, we have totally the 16 presentation. Out of the 16, we have 10 presentation online, 5 offline, 5. So the mostly this session focus on the diversity aspect of the fungi, many different kind of fungi, and also the metabolites and their metabolitic profiles of that fungi presented by all the presenters. And I think all the presenters are presented very nicely within the time. And due to some inconvenience, the time delay. So anyway, we completed within the time. I thank all the presenters for this, those who presented in this session. So also I thank the MSI office bearers and MSI for giving this opportunity to share this session along with the Sir Dr. Avnish Paul Singh. Thank you, sir.
Thank you very much.
Yeah. Thank you, sir, chairperson, co-chairperson, for conducting this session wonderfully. Even though we had a lot of interruptions in power cut and internet and other issues, but you have conducted it in timely fashion as well as given sufficient time to all the candidates. There were online as well as offline sessions. I thank you again, sir. And I'd like to also appreciate your support with a token of appreciation. Sir.
Thank you so much. Thank you. Really, we are thankful to the organizers, sir, and mostly these students have put up a lot of effort to make it happen. Really great. Thank you so much. Thank you. Really cheers to you.
It was the most eventful.
Yeah. Thank you, sir. Thank you, sir. Thank you, sir. Sir, thank you.